Good morning, everyone, and welcome. What a spectacular day we had yesterday, and I'm sure it's going to be well matched today. I'd like to start by acknowledging and paying respects from all of us to the traditional owners of the lands on which the Academy operates and where its fellows and employees live and work, especially the Ngunnawal people, where we're meeting this morning. Welcome to you all, and to those online. Greetings. We had a spectacular day, and to have a terrific event in the evening, and to see one of our own fellows being awarded a very special award. For those of you who couldn't attend last night, and for those online, here is the wonderful news that Professor Eddie Holmes, of course an Academy Fellow, was last night awarded the GSK Award for Research Excellence. He made such a difference to how the world tackled COVID and has made enormous contributions, sharing last night that perhaps we can take a single sample of blood and within a few hours determine any virus that's in there and speed up treatment amazingly. What an extraordinary achievement. And so can you join me in a thank you and congratulations to Eddie. So now we move to the final group of our fellows' presentations. And to start off today, I'm delighted to introduce Professor Rajeev Varshney. Rajeev, you've been instrumental in developing and applying large-scale genomic resources to crop management. You've determined the genomes of so many species, and it's made a huge difference to broadacre and horticultural crop industries in Australia and around the world. Farmers and people are now eating and thriving in Africa and other places because of your work. Congratulations. I know we want to hear more about it.
Good morning, and thank you very much, Lynn, for nice introduction. First of all, I would like to thank Academy for inducting me its fellow. It's really an honor and privilege for me. And now I would like to deliver my talk. So, before I present our research work, I would like to mention briefly that from where I have come, I did my BSc and Master's degree from AMU India, then PhD from NCCS University Meerut. Then I had a stint for about 5 years in IPK. This was the time I started to have the collaboration here in Australia, because one of my postdoc project was in collaboration with Professor Peter Langridge from University of Adelaide, and of course, Professor Andreas Graner from Germany. Then I moved to India, where I served ICRISAT, Global Research Institute, in various science and research leadership positions for about 17 years, and continued to have the collaboration with Australia. Then moved to Murdoch University in 2022. During these last 25 years, I had a privilege to work with very eminent scientists, colleagues, and very hardworking people who are the part of my teams as colleagues and collaborators. So I'm thankful to each and everyone, and also had an incredible mentorship from large number of individuals during last 25 years or so. But very quickly would like to mention some of key colleagues: Professor P.K. Gupta, my PhD supervisor; Professor Andreas Graner, my postdoc supervisor; Professor Wajahat Hussain, one of the great teacher in BSc, MSc; Dr. William Dar, Dr. Peter Carvery, Dr. David Worthington from ICRISAT, and Professor Peter Davis, Professor Eastwood, and Professor Andrew Diggs from Murdoch University, and of course my family. And would like to also thank Professor Jan Small and Professor Steve Poblis, who are also sitting in this room, for nominating me for this fellowship. Now, as I'm an agricultural scientist, and we know that agriculture and food is facing lots of global challenges, and some of them, as we know, that overpopulation, ongoing conflicts and geopolitical issues, these makes agriculture much more important in the current scenario from the perspective of the food supply, and of course climate change. All these issues, they pose a serious threat for the food security in developing countries. The good news is that we here in Australia, we play a very key role to contribute towards food and nutrition security around the world. Though in agriculture in Australia, we are having this thing on that on the belt, we call it agriculture belt. In this side, so our arable land, we are having around 25 million hectares, but agriculture worth is about $44.5 billion. So what agricultural scientists in Australia are doing, they need to keep the Australian agriculture climate change ready and internationally competitive. So what we have been doing, when I say agriculture, not only the geneticists or breeders, but all the entire agriculture community, including agronomists, pathologists, etc. So what we have been doing, that we are working towards enhancing yield, quality, and production area. And if we are able to do anything increasement, either in yield, quality of production, we can always enhance our value of agriculture production, can benefit growers in Australia, and even just 5% in any direction can give that advantage of about $2.25 billion or so. Now, here I would like to take you back in 1960s when many countries in Asia, they were struggling to provide enough food to their population. At that time point, Dr. Norman Borlaug, Dr. M.S. Swaminathan, Dr. Gurdev Ghosh, and Dr. Yong Longping, these gentlemen, they provided high-yielding wheat and rice varieties, which saved many millions of the peoples in Asia through the transformative agriculture by contributing genetics and breeding. And I had a privilege to meet Professor Norman Borlaug in 2003 in Italy, where he challenged the next generation of scientists to embrace modern gene technology to create innovative and sustainable solutions. And these words, they really impressed me. At that time, I was working in Germany. When I went back to lab, and at that time, genomics was in its infancy. In 2005, we had the first crop genome in the form of rice sequenced. This was the same year when I worked with my mentors, Professor Andreas Graner and Professor Mark Sorens, and we provided a framework of genomics-assisted breeding. And then, what we believe that if we can integrate genomics in the crop improvement, we can really deliver the superior varieties, which can be disease-resistant, high-yielding, etc. But to do this thing, we need to understand the genetics of those different agronomic traits. With this objective, what we have been doing during last 15 to 20 years, my lab has been contributing leading sequencing of genomes of large number of crops in collaboration with different colleagues and collaborators around the world, including here in Australia when I was working at ICRISAT, but now at Murdoch University, we are having genome sequencing center kind of thing, and we are contributing a lot in this direction. So after that, and then we have been using the genomic resources to understand the genetics of agronomic traits and development of the better varieties. I cannot provide everything in this short presentation, but would like to give an example of a chickpea crop, which is a very important crop from India perspective, Australia, but drought is a serious production constraint. So what we did, that in 2013, we developed the chickpea genome. Then in 2016, we sequenced around 100 varieties. Then in 2019, we sequenced the germplasm accessions, so like landraces, the genetic diversity, which has not been used in the breeding program. And then eventually, we also decoded genomes of more than 3,000 chickpea lines. We presented this genetic variation map. And recently, we have completed the genome sequencing of about 10,000 chickpea lines. So what we have done, that based on these sequencing efforts and phenotyping in the field conditions, we found a QTL hotspot in chromosome number 4. We have dissected it further. But when I was working at ICRISAT, we wanted to integrate this thing in the chickpea varieties. At that time point, we realized two serious constraints in the breeding program. One, cost on the genotyping. So we reduced it by establishing a low cost and high throughput genotyping platform, which is being used right now in more than 28 countries, and breeders can make this, they can make the best use of this platform. Second was the skill set in the breeding program in Africa and Asia. So what we did, we organized different training courses, at least 10 training courses. Through these training courses, we trained breeders in modern genomics and breeding skills. And then we have used the different kind of molecular breeding approaches in that crop improvement program, and our breeders, they developed improved lines based on those genomics integration. And then after rigorous testing in the different locations in the respective countries, several climate resilient high yielding nutritious variety have been released, delivered to smallholder farmers. For instance, in this slide, you can see 5 varieties of chickpea, which is having 15 to 20% higher yield in the case of India. And also in Africa, like in countries like Ethiopia, Kenya, Tanzania, etc. And not only for the drought, we also deliver the varieties for several diseases, Fusarium, Ascochyta blight, etc. Now, when I moved to Australia, we thought, can we replicate similar kind of storage here in Australia? So what we did, we started working with the GRDC, and we developed a chickpea pangenome for Australian varieties. And then what we did, that we did this pangenome analysis here in Australia, and we found that Australian chickpea varieties, they are missing that QTL hotspot, which was responsible for higher yield. So what we did, that together with GRDC, Chickpea Breeding Australia, we started to introgress this QTL hotspot. And we have developed the improved lines, and they will be evaluated next year here in the different locations. Wheat is very important crop in Australia, and what we are doing right now in wheat, that I think last year or so, we have developed a pangenome of 17 wheat varieties, and we try to understand those regions which are responsible for agronomic traits. And right now, we are leading an initiative, and this initiative will deliver wheat pangenome for 100 wheat varieties, and also the haplotype catalogs for 1,000 wheat lines, and many of them, they will be included from Australia, and they will going to change that breeding pattern, what we are doing here in Australia. And what we are doing, that we are using these resources to enhance nitrogen use efficiency, which is a very important trait here in Australia, as well as the heat tolerance in collaboration with large number of partners. Similar kind of efforts we are doing in the horticultural crops, which is also very important industry here in Australia. And what we are doing, that together with Hort Innovation and with several partners here in eastern states, we are developing the genomic resources, low-cost genotyping platform in large number of crops. And also recently, we also initiated a project together with Queensland University of Technology in collaboration with Hort Innovation, where we are using the genetics approaches, AI and machine learning approaches to create next generation orchard. So in summary, genomics technology has been very powerful. We have been making the best use of these technologies to develop the new generation of breeding lines. In my opinion, national and international collaboration and creation of next generation scientists is really required for sustainable agriculture. And at this stage, I would like to thank large number of collaborators, partners from Australia, as well as from the international organization. And finally, my sincere thanks to hardworking people from my group's PhD student, postdoc, etc. Thank you very much for your attention. I will be happy.
Thank you so much. This is just global scale research, and it's great to see it happening here in Australia. And I know there are going to be lots of interest in this, and I know Steve Powell's is in the audience amongst others, so I'm looking for hands going up. And while you're thinking, I want to know more about your training program, please, because one of the great things about agricultural research is how quickly you translate it into action, working with farmers, growing them, often experimental crops. Could you just share a little bit about that training program?
Very good. Thank you very much. So this program was part of that Gates Foundation as well as Ministry of Agriculture and Government of India. So what we were doing, that we were organizing these 2 weeks long training courses. We were bringing the breeders from the different countries. And in these training courses, we were not training them how to generate the experimental data set. Rather, once we get the data set, how to make the best use of this data set to create the next set of the varieties, working with the farmers, providing the better agronomy packages, because genetics and agronomy, they go.
Did they have enough science background when you
started? So they were basically working as a breeder, but in the traditional breeding approaches in the different countries. Yeah.
Thank you. I think almost every research program should be including that community.
Yeah, I think so. Yeah. And this has made the impact on that aspect.
Yes. Yes, question, Francis.
So that was amazing. I just cannot believe that there's over 10,000 types of chickpeas. But this is a wonderful example.
Your butter chicken will never be the same again.
No, it's a wonderful example of international collaboration, but it must have taken so much logistics to get access to all of those. Yeah. Could you just expand a little bit on how you went about it?
Thank you. Thank you very much. Good question. So in agriculture, what happens for each crop, we are having these germplasm collections. They're stored in the different gene banks around the world. So what is happening, that in ICRISAT, where I was working, this is a global research institute. Even in chickpea, we had around 20,000 accessions, 20,000 different kind of chickpea varieties coming from 65 different countries. And these germplasm, they were collected before 1993. So at United Nations, there is a convention, CBD, Convention of Biological Diversity. So any germplasm collected before that, this becomes that germplasm held in trust, and now this can be contributed to anybody. When I was at CRESAT, I had a plan to sequence the entire germplasm collections, but then I moved here, but good news is we already completed the sequencing of around 13,000 chickpeas. 7,000 left, somebody else will do that one. Thank you.
And as Foreign Secretary for the Academy, you would be very heartened to hear that response, I'm sure. Thank you. James, a question.
Thanks, Rajeev. And yeah, also, I didn't know that much about chickpeas either, so fascinating. I suppose extending that question, how's the current geopolitical environment impacting your research, and what threats do you see emerging?
So, yeah, so I think that the geopolitical issues, they pose challenge in two ways. One is this exchange of the germplasm from one country to other country. Second is even in terms of the funding. So even many of these, my earlier collaboration, we used to have a lot of funding from that internationally. But in the current scenario, number of regions, nowadays the funding from one country does not move to the other country. So what people are doing, they are working in their own countries. And then we are working through the data analysis and collaboration like that. Yeah.
Gosh, it's an extra challenge.
Yeah.
Thank you so much. Join me
Thank you
in
very much.
thanking for
Thank
a
you very much.
wonderful
Thank you.
presentation. Now I hand over to Francis.
So thank you, Lynn. It's my pleasure to now introduce Professor Guoxiu Wang. Guoxiu Wang is internationally acclaimed for his pioneering research in advanced battery technology, including by setting records in lithium air battery performance and significantly improving the safety and efficiency of lithium batteries. So please welcome Professor Guoxiu Wang. Thank you.
Thank you. Good morning, ladies and gentlemen. I'm very honored and also very excited to be elected as Fellow of Australian Academy of Sciences. Today, I'm going to talk about battery technologies, which I think and also well recognized as a cornerstone for clean energy technology, clean energy transition. As you know, climate change and global warming is a reality. So this is mainly caused by the burning of fossil fuel for energy. And you can see the burning of fossil fuel causes not only climate change, but also heavy pollution and deteriorate the global ecosystem. If we consider our society, our modern society, without electricity, we cannot survive. Without mobility, we cannot survive. So currently, we heavily rely on fossil fuels. If you combine the renewable energy with the battery storage, we can create a sustainable future. Actually, the battery technology is not a new technology, it's an old technology, has been developed for more than 200 years with many different kind of battery system, including non-rechargeable battery we call primary battery, and rechargeable battery we call secondary battery. And in the history, it's really the invention of lithium-ion battery bring us a dramatic change to our modern society. That's why these three scientists were jointly awarded the Nobel Prize in 2019, and they were praised to create a rechargeable world. Lithium-ion battery laid foundations for portable electronics and also make a fossil fuel-free world to be possible. So if you look through the development history, it went through almost 3 decades. In 1970s, Professor Whittingham in the United States, he proposed and invented called lithium metal batteries, using lithium metal as anode materials and titanium disulfide as cathode materials. But this product commercialized but failed because the safety problem. And later, in 1980s, Professor John Goodenough, he was a giant in physics and also material science. He developed a cathode materials called lithium cobalt oxide, which can dramatically increase energy density of lithium battery. Because it still uses lithium metal as anode, has not yet been commercialized, and in early 1990s, Yoshino in Japan, he figured out an idea using the petroleum coke graphite as the anode materials, plus adequate electrolyte system, and then later Sony Corporation commercialized lithium-ion batteries. So nowadays, the lithium battery system is still under dramatic development for diversified applications. So here I would like to introduce lithium-air battery, which is regarded as the ultimate goal of lithium battery technology because of very high energy density with many potential applications, particularly in the aviation industry and also defense industry. So in my group, we developed several chemistry, the molecules we call redox mediator to boost the performance of lithium-air batteries. So here, this work, we developed ionic liquid bearing redox active, or we call TEMPO molecules. We invented these molecules and we added in to the lithium-air battery system. We found that we can increase the energy, the discharge capacity of lithium battery for 33-fold. And here you can see not only we can, with these new molecules in the electrolyte system, we can decrease the overpotential of batteries, and also we can increase the capacity, specific capacity, and the energy density of lithium-air batteries. And then later, we further developed this technology. We invented a new molecules called PTI TEMPO. These molecules has a very special molecular structure. When we put in the battery system, the lithium-air batteries exhibit 46-fold increase in capacity, and also we achieved very long cycle life. So these molecules has a steric hindrance the structure with a TEMPO functional group on the side of the backbone of PDI. And then my group is the first in the world. We utilize the proton molecules to apply to the battery system, particularly lithium metal batteries, to protect lithium metal anode. So here we use firebromine, or called silk protons, with the helix structure. When we put in the electrolyte, and when these proton molecules contact lithium metal, it becomes a 2D sheet structure. And then efficiently, we found that this strategy can efficiently protect lithium metal anodes. Here, from this figure, you can see we can extend the cycle life of lithium metal anodes for building the lithium metal batteries. So next, I will talk about battery safety. Nowadays, with a wide spread of EVs, electric vehicles, and large-scale energy storage, the safety is a big concern. When you drive Tesla, actually the battery, the car, the EV, the current version is not really safe. And this is happened in Sydney airport. The EV burns and destroyed luxury cars in the Sydney airport 2023. And particularly this is a typical case in Geelong, Victoria in 2021. when Tesla big batteries during the commission process cause fires. Once it's burned, you cannot extinguish the battery fires. So this is a big challenge. So in my lab, we figured out a solution. We developed a new electrolyte system based on the fireproof materials we call phosphazine. And we use this rational thinking. We figure out if we use a fireproof polymers, we can develop fireproof batteries. So this is the testing. You can see when utilize our newly developed, we call gel polymer electrolyte, you can see if you ignite the electrolyte, it cannot be burned. That means if you put this electrolyte in the battery system, it's absolutely safe. Then we construct these batteries, and we achieved very nice performance, electrochemical performance. Currently, I'm utilizing my ARC Industry Laureate Fellow, try to commercialize this technology. And then with the widespread of EVs and home, household and large-scale energy storage, and after 10 years, 15 years, at the end of the battery, you have recycled the batteries. Here, we developed a technology can very nicely recycle batteries, particularly extract lithium, and simultaneously we can generate hydrogen. So next, I will talk about our rich resources. Australia is very rich with critical minerals, particularly for battery industry. Currently, we export 50% lithium minerals to the global market for battery industry. And also we own substantial critical minerals such as nickel, cobalt, and manganese, which are critical for battery industries. But our current practice is not sustainable. We just dig and export minerals. So we should construct plants and process those minerals to the battery materials and supply to the global battery market. Currently, I'm leading a team at UTS. We try to build advanced battery manufacturing facilities and provide a platform for the industry to establish battery industry in Australia. In my vision, if we combine solar, wind energy, and battery storage, we can build up a sustainable future. I would like to acknowledge funding support from ARC, CRC, and ARENA, and thanks for.
Thank you very much. That topic is certainly rich in questions. Do we have any questions from the audience? And I can see one up the back. You always ask questions, Lee. Just up there. Yeah. Oh, up. Oh, they were pointing at you. All right. Okay. Thank
you for sharing your amazing work. We talk about recycling at the end of the life of these batteries, and do they have a long life? I'd love to know that. Can we actually then take them apart and use the constituents over again, please?
Yes, current practice is a landfill, which will cause severe environment problem. Using the science and the technology, we can 100% recycle all components in the spent batteries.
Briefly mentioned commercialization of your technology. Are you able to expand on that at all?
Yes, because this technology we co-developed with scientists in Israel. We are currently jointly commercialize this technology, and we talked with many different battery manufacturers globally.
Could I just abuse my privilege as chair and ask a question? So with those large Tesla batteries in Geelong, and they're burning, right? What about using sodium for those large ones?
Sodium-ion batteries is a newly emerging battery technology, and is also currently under intensive development in my lab, but not commercially available, not yet.
Well, thank you very much.
Thank you.
So I won't ask you again to thank Kivshar, but I'll ask Lynn to come to the stage. Thank you.
Thank you, Francis. I'm now delighted to introduce our next presenter, Professor Nicole Webster. Your work, Nicole, has spanned from the Great Barrier Reef, the west coast of Australia, right down to Antarctica. So it was a great grounding for you to become a global leader in marine microbial symbiosis. And you've generated a transformational insights into coral reef microbiology and pioneered the application of molecular tools to the sponge microbial symbioses, all those bugs living inside things we didn't know were there. Welcome, please share your research with us today. We'll be very happy to hear it.
Good morning, and I would like to also extend my thanks to the Academy for welcoming me into the fellowship, and a special thank you to Professor Helene Marsh for nominating me and making all of this possible. So today I've decided not to actually share any hard data, but I thought I'd take you on a little bit of a journey into the wonderful world of marine microbes. So as a coral reef microbiologist, my research has really been focused on trying to understand the myriad of ways in which microbes contribute to reef ecosystem health, from their roles as symbionts and pathogens of marine invertebrates, their roles in biogeochemical cycling and higher food webs, their roles in inducing the settlement and metamorphosis of different marine larvae, and then trying to understand how all of those processes are impacted by environmental change, particularly climate change. And in piecing together all those different roles, I also became interested in understanding whether marine microbes could be sentinels, alerting us to stress in the reef environment long before we see visible signs of reef decline, and that would allow for early management intervention. So to sort of approach that, we sequenced the microbial genomes of over 5,000 bacteria and 800,000 viruses collected along the length and breadth of the Great Barrier Reef to establish a really comprehensive database of marine microbes from which we could then use that to assess changes in microbial communities along space and time. And from that analysis, we found that actually marine microbes, because of their sensitivity and their predictability, they have extraordinary potential to be used as early indicators of environmental change. Now, while microbes can tell us something about coral reefs at a really broad scale, most of my career, as Lynn just outlined, really zoomed in to understand how microbes form partnerships with different marine species. And this particularly focused on marine sponges, which are not only some of the most beautiful life forms on the planet, they are also the oldest living multicellular animals. They're incredibly ancient. They evolved during the Precambrian explosion 500 million years ago. So this provides a great model system for understanding how microbes also contribute to host evolution. And marine sponges are incredibly diverse. There's 15,000 species of sponges globally, and of those, 2,500 occur on our very own Great Barrier Reef. So Australia really is a global hotspot for sponge biodiversity. And they're incredibly ecologically important. They consolidate the reef substrate, they provide habitat for a wide range of species, and they also form this really critical link between what happens in the benthic environment at the bottom of the ocean and what happens in the pelagic zone. And that's because of their incredible filtration rates. So in fact, sponges are really quite remarkable filter feeders. They're able to process up to 50,000 times their own body volume in seawater every day. And it was also recently discovered that sponges are part of a really highly efficient recycling pathway for dissolved organic matter. They're able to convert it via their rapid cellular turnover into detritus that then becomes the food for reef consumers. And in fact, dissolved organic matter transfer through this so-called sponge loop actually approaches the gross primary production rates thought to be required to sustain entire coral reef ecosystems. So possibly contributing to why coral reefs can persist in what's the equivalent of a marine desert, these really oligotrophic seas. Now, as part of this filtration, they use these flagellated choanocyte cells that you see there to efficiently extract out all of the particulate organic matter, the microbes, and the viruses. And so that leaves the outflowing seawater essentially sterile. But it's because of this filtration efficiency that sponges can also be particularly impacted by water quality stresses like elevated nutrients and sediments, as well as the global pressures of ocean warming and ocean acidification, which I'll also just touch on at the end. But of course, the reason marine sponges are such a wonderful model for a microbial symbiosis researcher is that they harbour extraordinarily dense, diverse and specific microbial communities. There can be up to 10 to the 9 symbiont cells per centimetre cubed of sponge tissue. You can't even see a host cell on that micrograph on the left there. And each individual sponge can host thousands of genetically distinct microbial communities. And these microbes are really specific to their host. They're completely different to the microbes that we find in the surrounding seawater, which the sponges are sitting in. So we wanted to understand what these microbes do in the sponges, so we did a whole range of different molecular analyses, and we found that there is extensive taxonomic redundancy and functional equivalence in the microbial communities for pathways associated with carbon fixation, nitrogen metabolism, sulfur metabolism, and then the production of a broad array of different vitamins which the host can't synthesise for itself. When we undertook a meta-analysis where we sequenced the symbiont genomes of over 1,200 symbionts, we found that these microbes are definitely not passive passengers. They are actively recycling the reef organic matter. They have genes that can degrade the sponge tissue, they can degrade coral mucus, and that can also degrade macroalgal exudates. And so these sponge symbionts, they also have genes that really allow them to attach directly to the host tissue. So there's a range of genes there. They have very active defence systems, so things like CRISPR and restriction modification systems. They are really enriched in these eukaryotic-like proteins, which we think actually allows the symbiosis to be maintained over a time. And then all of these genes are actually being shared amongst the different members of the symbiont community, indicating that they really do provide a competitive advantage for the sponge microenvironment and that they likely play a really significant role in host ecology and evolution. So how do these microbial communities get established then, and what maintains them over time? Well, there was some beautiful work a number of years ago coming out of Roman Stocker and Justin Seymour's group that showed that many marine species, like corals, actually have a really chemically rich microlayer that can attract specific microbes and deter others. So we were really interested in understanding whether this chemoattraction could actually be used to trigger the attraction of microbial cells from the rare seawater biosphere. So these symbionts are below the detection limit in the seawater, but maybe they're just there at really low abundance, and this chemical attraction could be a mechanism to attract them towards their host. So this concept of chemoattraction, I showed you a short video there of microbial interactions with a piece of marine snow. Because we track the microbial movement, you can clearly see that movement isn't random. It was largely occurring in response to the chemicals that were being released from the surface of the particle. So to test this in sponges, we use this artificial platform called an in situ chemotaxis assay, or an ISCA. It's only about the size of a credit card. It's manufactured out of PDMA, and it's manufactured in such a way that you can load this device with different chemicals, and they slowly diffuse out into the surrounding environment, which creates a chemical gradient that can trigger the migration of attracted bacterial cells into the ISCA wells. So to test this with sponges, we made a range of chemical extracts of the sponge tissue. We loaded them into the wells, and we saw a significant enrichment of some sponge symbionts in the ISCA wells that contain the sponge extracts, revealing a really remarkable active role for some of these symbionts in finding their sponge host. We've also done a series of work that shows that vertical transmission from the parent sponge to the larval offspring is another really significant mechanism for maintaining these symbioses. And this occurs in both brooding sponges that raise their larvae internally, as well as broadcast spawning sponges that release the eggs and the sperm into the surrounding seawater for external fertilisation. So here we see symbiont transmission occurring via the egg stage. So how are these remarkable symbioses being impacted by environmental change? Well, we've done a number of decades of research now using both experimental systems like the sea simulator at AIMS, as well as natural environments like these carbon dioxide seeps up in Papua New Guinea, to try and understand how the sponges were responding to ocean warming and ocean acidification. And from that research with many different species, we've found that just like corals, there's significant variability in the species tolerance limits. But what we see in sponges that are sensitive to ocean warming, not only using proteomics and transcriptomics do we see an immediate stress response in the sponge host to the elevated temperature, we also see an immediate stress response in the symbionts, and that includes reduced expression of functions that mediate that partnership. And then over time, this disruption to nutritional interdependence and molecular interactions between the sponge and its symbionts further destabilises the animal. We see the loss of our known symbionts, the introduction of microbes with a much more scavenging lifestyle, and then we start to see declines in the host health, including tissue necrosis. In sponges that are tolerant of ocean acidification, we actually see an increased potential for energy-efficient carbon fixation and nitrogen metabolism, yet in species that are sensitive to ocean acidification, we see this reduction in their ability to be able to use host-derived metabolites. So these findings really suggest that microbes do play a really important role in how sponges tolerate climate change. And in thinking about preparing for today, I actually reflected back to a period back in 2010 when I stood on this stage in receipt of the Dorothy Hill Medal. And at that time, I had an acknowledgement slide which had the figure on the left there. And I put this there because as a young mum in science, I was often really conflicted and torn because the time that you spend doing research, you do take away from your family. And I wanted to acknowledge the impact that that has on young family. I've reflected back, and 15 years later, you can see the figure on the right, and I hope that one of the things I guess I'm most proud of is that dedication and passion for science has, at least in part, played a role in their journey as well, with all three of them now pursuing careers in engineering, chemistry, and likely astrophysics. So, thanks.
Great to hear that personal aspect too. Congratulations. Absolutely extraordinary work and a new world, I think, for so many of us. Yes, James has a question, and then we're coming back to the front.
Thanks, Nicole. Amazing research. I suppose I have two questions. One is some of the work around kelp with the climate change and warming in parts. They're looking at moving sort of more robust kelp to different parts. Is that something that you're considering or is part of the research? And the second question is, I have at the foundation, I have a number of people that research what they call squishy fish, and they say it's really hard. It's it's really cool to search whales and sharks and all the really sexy kind of big things. It's pretty easy to get money, say, for whale sharks, but no one's really interested in funding research around squishy, ugly fish. So I just wanted to maybe ask you a little bit about the funding challenges you face.
Fantastic. I'll start with the kelp because actually in my new role as the Executive Director at IMAS at the University of Tasmania, that's actually a project that we're really involved in, is the transplantation of genetically tolerant kelp populations. And it works really well. If you get the ecological conditions right, if you stop the predation of the urchins, you actually see quite remarkable kelp growth, 10 metres in a year we're seeing. We've done a little bit of that work of transplantation with sponges. We did some of that work down in Antarctica as well. It can work. It depends on the species, how quickly they can do that. In fact, there was a lot of work that was done up in Papua New Guinea and in the Torres Strait around actually growing sponges commercially, actually with some of the indigenous populations. And actually you can get really remarkable growth if you get the right sponge species. Others are really sensitive. So there is potential there, I think, for transplantation. And there isn't much research happening because, as you said, they're not charismatic megafauna, but there's not much research happening to understand range expansion in sponges, so that's probably a future area. People often ask me why I speak so passionately about microbes, and anytime I'm given a soapbox, I will do that, because certainly having a career in a marine science institute where you're the only one working on sponges, and not only do you work on sponges, but you work on the invisible things inside them, it's been really hard. But there are unique funding. So we've just been awarded a really major grant by the Sanger Institute to sequence the genomes of 10 host sponges and all of their symbionts as well, to establish them as a model for understanding microbial contributions to host evolution, actually. So there are pathways, but they're probably more challenging than some of the other things that people can work on.
Congratulations. Yes, question, please.
And what does the sponges or microbes do with the nitrogen? Convert it to the N2? What's the nitrogen cycling in that system?
Yeah, they do the full cycle of nitrogen denitrification, because ammonia is a toxic waste product for the sponge. So they have to find a way to be able to eliminate the ammonia from the sponge. And so we have thousands of different microbes all contributing different parts of the whole nitrogen cycle within the sponge as well. And in fact, there is a quote that I absolutely love for a really eminent microbiologist, Tom Curtis, who said, if the last two, and it gets to this question as well, if the last 2 whales choked to death on the last 2 pandas, that would be disastrous, but not the end of the world. If we accidentally poison the last 2 species of ammonia oxidizers, well, that would be another matter. The tragedy is that it could be happening right now and we wouldn't even know because we're not exploring it. So,
gosh, you were nodding to that answer. Yes, sir, please, another question.
Thank you. Beautiful work, and I love the depth of which you've gone into everything. I'm going to ask a slightly obscure question. How do you think that AI and machine learning is going to completely change what you do? Because this complexity is so hard for us to understand. I can imagine it will revolutionize your world.
Yeah, it will absolutely revolutionise things. So in trying to really get, because of the diversity of microbes, thousands of different microbes within a single sponge species, it's been really hard to disentangle things like metabolic syntrophy, who's feeding who's doing what. And because they're not genetically tractable, you also can't do any kind of knockout experiments. With the application of AI, we'll be able to tackle some of these things that we just haven't been able to before. So I think you're absolutely right.
Thank you so much. Absolutely extraordinary work. And I hand back to Francis.
Thank you, Lynn. It's my privilege to introduce our final speaker for this session, and that's Professor Tony Weiss. So Tony Weiss is a world leader in molecular studies of human tropoelastin and its assembly to make 3-dimensional elastin protein materials. So please welcome Professor Tony Weiss.
It's a pleasure to be here. Let me first of all thank Francis for the introduction. I want to thank the Academy for this remarkable honour. It's a very humbling thing to be standing over here and to be recognised by peers as well. And of course, obviously, I particularly acknowledge and am thrilled by the people who nominated and supported this entire process. But that's not my entire presentation today. Today I'm actually here to talk about something that I'm calling an elastic journey. And for that elastic journey, I would like to really describe to you something that is, without any further pun involved, is very close to my heart. And that, of course, is the concept of elastin. And so elastin basically is a protein answer to the body's elasticity. And the best way to visualise this is that, of course, none of us are rigid robot-like objects. Basically, we have a lot of flexibility built within us. But for a long, long time, that flexibility has been inaccessible. And in many ways, it still is inaccessible at the molecular level in terms of building components. So just to drive this point home, for example, the lung that you can see on the left-hand side over here, as we breathe in and out, we require elasticity, and of course it's elastin that contributes towards that elasticity. Elastin, again, being the body's rubber-like protein that is required for that elasticity, as evidence that, for example, the lung needs that elasticity, which in this particular case, elastin just comprises 4%. When the elastin is damaged, for example, in emphysema, then, of course, lung function begins to shut down. Across the top there as well, you can see second from the left, we have a vertical cutaway view of a blood vessel, and as that blood vessel expands and contracts with every heartbeat, for example, in this case an elastic blood vessel, it responds to that, those waves of systole and diastole to smooth out those peaks and troughs to be able to construct a very precise range over which we then get the flow of those peaks and low levels of blood pressure. And you can see words like internal elastic lamina, external elastic lamina, and so forth. And that's because within the vessel wall, there are concentric layers of elastin that contribute towards that elasticity. In the largest vessel, for example, coming out of the heart, which is the aorta, it's estimated, depending on which particular part you look at, that there's between 1/3 and 1/2 of that just of elastin alone. We can go on, but for example, in the bottom left-hand corner, you can see skin, and as many of you know, elastin is lost as we age. You can actually do the pinch test, just pull up skin and then watch how long it takes to drop back down again, and that actually is your measure of how well the elastin is doing recoil. And of course, that elastin degrades, and of course other locations too, for example, elastic ligaments and joints. And so the body really then has many locations that rely on elastin as the body's elastic protein. But the difficulty, of course, is that elastin is made primarily before we're born, and then is really only provided in trickle form after the first few years of life. And so, of course, you can imagine it's necessary and to build lung before we're born, need to build blood vessels and so forth, and then just maintain them during growth, which is a very slow and very limited amount of production. And furthermore, the elastin is meant to endure. In fact, it's estimated to be the longest lasting stable complex in the body that's protein-based. So how is elastin made? Well, it turns out the best way to view this, and we've heard Lego in terms of presentations yesterday, so why not use Lego again? The way this is made is we start with a monomer, which in our case is tropoelastin. It's a 698 amino acid protein, and that protein is encoded by a single gene. In all vertebrates, it's responsible for elasticity, but in humans it's just a single gene. There are only one set of vertebrates that do not require elastin. Those are the weirdly lampreys and so forth, the cyclostomes, but all the rest of us do. And that tropoelastin is assembled to make these fibres and sheets and tubes, these very precise structures. Now, they're immobilized by a form of molecular glue that links these molecules together. So it's as though you actually stuck these with super glue to make these permanent structures. So to access that tropoelastin, to be able to build new elastic tissue, to repair damage in elastic tissue, means we can't go back to elastin from whatever source. So not only can we not go to animal sources, ethically, of course, we can't do anything with humans, of course, as you might imagine too. So therefore, we need to go to an alternative source. And so what my lab did as part of this foundational work was we said we'd like to make tropoelastin. No one else was able to do so. And the reason why was because it is enriched for key amino acids. There's a highly enriched level of about 2/3 of the protein consisting of just a rare collection of amino acids and codons. So we made what was then the largest tailored synthetic gene. This protein is not post-translationally modified in any appreciable way. So we then, and we then tucked that tailored synthetic gene into E. coli, and we matched the Epicurean requirements of E. coli to be able to make large amounts of this protein. And you can see the protein on the left-hand side just over here, and that is a protein that we're able to then work out its shape and its a whole range of properties. We actually were able to look at this for the first time. And so the shape of the protein is 20 nanometers in length. We worked out its shape using a synchrotron because, as you might guess, it's a wobbly molecule, so you can't crystallize it. And then in collaboration, not only with colleagues in the UK, but also the USA, we're able to also work out dimensionally what its collection of roughly half a dozen low energy forms were, and that they very nicely tuck into what was then the experimentally determined envelope that's been described over here. We could also show that we could tease that molecule out and extend it to 20 times its own length, and it would then retract without any evidence of energy loss at all. In fact, remarkably, much better than any other known elastic molecule or system that it is seen that's human-made. And that's not surprising because, for example, we need to be able to cope with expansion and contraction of, for example, the aorta about 2 to 3 billion times in our lifetime. So it's quite extraordinary. So now that we had this protein, what could we do with it? Well, apart from the science of understanding all these extraordinary features, there has to be the application, right? So I'll try and weave into this both stories, which are both the fundamental and the applicational. And so one of these, of course, must be, can we do something about blood vessels? And so we were able to construct tubes that contain tropoelastin, a sustained release material, and they're manufactured in the lab, just consisting of two components. And we stitch these tubes directly into replacement regions within the aorta. First of small animals, mice, and then we're able to move across to larger animals in other studies. And within those systems, we then implanted these with the theoretical expectation that those blood vessels, as implanted, would then gradually form new vessels, what we call neo-vessels, with organisation structure with these organised elastic lamellae that I described at the beginning of my presentation. And that is exactly what we saw here. You can actually see the organisation stylistically on the left-hand side, a series of the concentric lamellae, and no one else was able to achieve these with substitute blood vessels. And you can also see how it's arranged spatially across here by standing for the elastin. Now, this is very important because when, for example, you'd like to implant a blood vessel, let's say, into a young child, you want that vessel to be able to grow with that child over time. So you want something that is living and has dimensional and physical properties that match those that are seen directly within, for example, that child. And you can see as we slice through, sushi-like, through the vessel here, working our way along that tube, you can see these aren't just simply fibres, they really are concentric lamellar structures, which is all very exciting for us. And so we've clearly got an interest in expanding the story directly now across towards human use so that it can be used, for example, for bypasses amongst other uses, and of course obviously help kids out. And as part of that interaction, we're interacting with a nationwide children's hospital in the USA. One of the other scenarios, of course, is can we just simply use the assembly tropoelastin, which spontaneously assembles. We've learnt out the rules of assembly, which, by the way, exquisitely rely on assembly towards, towards giving assembled materials, and I will speed up my presentation within the next short while, and we can actually... when you get passionate about your own topics, time flies. But anyhow, so we're able to then generate seals across tissues. We could show these across heart and lung, and we can actually therefore give rise to locations where we can seal punctures within either heart or lung and give rise to materials which basically are much stronger but still elastic than anything which is sitting out there, including even suturing. Moving right along now, you can also imagine we can implant such materials directly or inject them as materials as well, directly deeply into wound sites, and we can encourage this repair. And there isn't sufficient time to tell you, but briefly, very briefly, we're actually able to generate a series of blood vessels. Tropoelastin triggers the formation of de novo vessel formation, which provide the highways for localised assembly, and there is an influx of cells and organisation that's associated with accelerated repair, so we see healing at about half the time. time. And indeed, that healing is shown by a demonstration very briefly over here, and I'll zoom straight across to day 14 for obvious reasons, and you can see the leading material that commercially does not heal nearly as well, at least within these model systems, and we've shown this within the human case as well, and that includes the formation of blood vessels as shown by the glow, and of course we see accelerated repair by the minimum formation of wound repair within these model systems. I will round out the story by telling you also that we can also inject a single bolus of tropolastin into heart, and we can actually restore and maintain the heart function as opposed to scar formation, and that's obtained through the formation of elastin. And indeed, you can see very briefly that by means of ultrasound within that system, that if we don't inject tropolastin, we see no wave-like formation ultrasound, but the echocardiography shows a restoration of that flow and restoration of a lot of function. And I can maybe answer that if there's any time for questions. And that's shown by means of reduced scar size. I'd also founded a company which was able to translate this. I'll go through to the bottom line over here, was formation of a generation of a large number of patents. We were able to draw in funding. We were able to take this through as an Aussie success story through to now 5 clinical trials, and this is being developed further still by AbbVie, which is one of the largest healthcare companies internationally, and that's been a very nice, very gratifying process. And the last, but by no means least thing to say over here very briefly is, of course, obviously I thank my family above and beyond, my wife Jackie, and wonderful family that I have, which I truly respect and love, and friends, many granting bodies, some of which are shown here, my colleagues in the lab, some of whom are shown here, the Charles Perkins Centre. Steve would appreciate the image, even though Steve knows this is a cartoon rendition, it actually does exist in real life. And of course, as you might see from one of the images down here, I thank the Academy not only for a previous recognition, but thank many other people for recognition over time as well. And Francis, I hope that was still okay. Thank you all very much.
So thank you, Tony. I must say, I didn't want to get out of my chair. I would have liked to have listened.
Thank you so much.
But I encourage you to ask Tony some questions, but I discourage him from giving too passionate answers.
Yeah, right. I'm always passionate, yes. Yeah.
So, there's a question.
Beautiful story, Tony.
Thank you very much.
Just mechanistically with tropoelastin, how does it know how to orientate itself? Because presumably in tissues, it has to do it in the right way.
Yeah, thank you. So, so keeping the response brief and less enthusiastic by the sounds of things, so tropoelastin assembly is partly spatially defined by the beginning and ends of the molecule. So the initial stage of association is a head-to-tail association process and then lateral, but there's also a deliberate process of anisotropy. So there is a delivery so you can stretch in almost any direction eventually on a macro scale. So it goes through log increases in terms of assembly from these tiny 20 nanometer building blocks to making something through intermediates that are roughly 10 times the size each time until you generate those large objects in the body. The external part of it has the ability to interact with cells by very precise orientation.
There's some good examples now of where genes that are expressed in the embryo and then turned off are turned back on again for therapeutic benefit. Is there a strategy here to look at turning back on, or increasing the expression of local?
Yeah, there is. So tropoelastin can be increased in amount, for example, through retinols and so forth. And in fact, that's the basis of retinol creams that are used to try and hopefully restore some level of elasticity in skin. The problem, though, is that the expression level is incredibly reduced over what you'd normally need to make a real meaningful impact on a larger scale. So expression drops to about 1%. The optimists say 2%, but it's that kind of level after about 3 years of life. So the rest of our life then would need to have this system persisting. So even if you were to increase it, you'd be increasing off that tiny base, and it's about a 3 to 5-fold increase, so you can see it's just insufficient. So therefore, we need to step in by supplying this building block separately, so you still need the Lego. Okay, that's fine. Thanks, Francis.
Thank you so much, Tony. That was great. And please thank Tony for inspiring. So that brings us to the close of our 2025 Fellows presentations. And I'm certainly inspired so much. We really do make the right choice. But there are so many other people who deserve to be in the Academy. And please, please do nominate them. So what are we up to? Morning tea, I think. I forgot to check my script. I have to remind all new fellows and awardees to head to the Becker Room today for your portraits. The Becker Room is outside one of those doors. It's not too far. And now we're going to go to morning tea, and I've been asked to invite all EMCRs to gather for a group photo at the main entrance of the Shine Dome. This is any MCR who's here with us today. Apparently there's about 110 in Canberra today or at the Shine Dome. So please move to the main foyer and gather for your photograph. Apparently we're going to refresh the theatre. It needs refreshing. And so I can please ask everyone to take your belongings with you to morning tea and come back here in about half an hour to hear the honorific awards. So thank you very much and see you soon. Thanks.
So why does science matter?
Science is what drives our advances in medicine and what gives us new treatments, new vaccines, new techniques in surgery. All of those need to be evaluated, developed, many of which come from crazy ideas from fundamental science. Other advances come from astute observations by clinicians. So science is so critical to advances in medicine.
And I think this was one of the most beautiful examples of science in action, where every biomedical researcher on this planet over the last 3 or 4 or 5 years has read a paper, and many have written papers, about a virus that they had never heard of. I find this mind-blowing to what degree we have, as a community of scientists, come and tackled this new threat.
I'm motivated to research antibody pharmacology because I see the human cost of deadly bacterial infections. I think science is fundamental to discover the mechanisms, and then we can use the mechanistic findings to develop novel therapeutics. This kind of translation is not come out from a simple thinking. It come up with a long-term research. So that's why science is so critical.
Science is just the way that we learn to think. You know, we learn to gather data, interpret data, make decisions based on data. And over the last few years, it's been really surprising and horrifying, actually, to see that not everybody thinks that way.
We live in a world where we need to make informed decisions about how we build our houses so that they use less energy. so that they're more comfortable, healthier, better places to be. Even just down to the simple decision of, whether or not you put insulation into your walls or into your roof, science can tell you the answer, which is the better of the two things to do.
If you think about thousands of years into the future, without science, we don't have a great hope of survival, I don't think. Science will underpin all of that. A better future for our kids, that's going to come from science.
I got 0 points when I applied for migration as an engineer to come to Australia. We went for the interview and the officer there said to me, I don't know why you want to go to Australia because you'll never get a job. My name is Marlene Kenga. I'm a chemical engineer. I'm a past national president of Engineers Australia, and I'm a past global president of the World Federation of Engineering Organizations, representing more than 30 million engineers around the world. I always wanted to be an engineer. I was very, very good at science and mathematics at school, but no one told me that girls don't do engineering. So it was quite a shock when I arrived at university and found that girls were less than 1% of the student body. I went as a very shy young woman, and I found I had to find my voice. It's interesting that a multinational company eventually hired me. I was only the second female engineer in that organization. Things have changed, I have to say, I'm pleased to say, and often I wish that I had some of the managers who are now leaders in many engineering organizations. But one of the issues for Australia is attraction. So we have very small proportion of girls taking up engineering as a career. And when they graduate, they often don't take engineering jobs. They go into other spheres like banking and so on, where their analytical and mathematical skills are highly valued, which tells me that we haven't done a very good sales job on engineering. I actually did something about that because I wanted to convey the impact of engineering. As president of the World Federation of Engineering Organizations, I initiated the proposal, a crazy, mad idea, to declare 4th March as World Engineering Day for Sustainable Development. It was an impossible dream. I had to get a nation from every continent to support the proposal. But I went around at UNESCO and knocked on the doors of all the ambassadors. And they said, yes, we need more engineers. We need more engineers to build the roads, to build water supply systems, to provide sustainable electricity. And people just love the idea that we talk not about the technical aspects of engineering, but what engineering does for people. Yeah. Now we're getting good comparative results. I chair Rux Energy. This company has a fantastic purpose where it's developing technologies for hydrogen storage. So what we have here is a crystalline framework that absorbs hydrogen into its pores like a chemical sponge. And we have specially built a machine where we have tested the resilience of this material for the hydrogen to be adsorbed and desorbed thousands of times to simulate an industrial environment. If it works, it can be the Microsoft of the energy transition. I'm really grateful to the Academy for recognizing my contributions, and I'm hoping that I can bring my energy and my resilience and my persistence to the projects that the Academy has. I like to say, you plant a tree. Science is the root, and engineering is the fruits of that science.
Should a good bottle of wine be sealed with a cork or a cap? There was a time when the high-end wine world scoffed at the mere mention of screw caps.
It might seem a simple thing, but that required an enormous amount of research.
It was in fact industry leaders in Australia who led the push, eager to increase consumer choice. Research has shown that screw caps offer a better seal, meaning oxygen can't sneak into the bottle. The result is a crisper and more well-preserved wine, suited to most varieties of white. Some winemakers insist that fuller reds benefit from a little oxygen in the bottle and therefore age better with corks. But screw caps have another benefit. They prevent the wine from ever being corked, a term used to describe the presence of undesirable chemical compounds almost always transferred from poor cork batches. Corked wine has a characteristic odour that some compare to a wet dog.
We understand so much about the underlying science in Australia that we virtually get it right every time.
Professor Peter Hoy is Vice-Chancellor of the University of Adelaide, but he was also Australia's first Professor of Viticultural Science and then Enology. In other words, an expert in the science of grapes and wine.
This is where science always comes out on top. It enables things because people trust it.
Hey, can you see it?
See what?
The wonderful possible. Imagine you see the world drowning in fast fashion. So you chemically dissolve old cotton clothes into something new.
Whoa.
Thought you'd like that.
What about a PhD at 80? What?
Still got it.
Pop-up law clinics anyone can access?
Nice.
All lifting others up through education. Yeah, I see it now.
Yeah, you do. Deakin University. See the wonderful possible.
This farm near the New South Wales city of Wagga Wagga is a long way from war-torn Ukraine.
These varieties here, they're all spring canola, and in Australia we mostly grow spring canola.
But it's familiar territory for Marina Tsu and Svetlana Omelchuk.
Big surprise for me that we have a lot of in common things in agriculture in Ukraine and in Australia. It's really was like, you know, Australia was something very fine, different from us, and when we are here we look and wow, we have the same thing. So we are very interested in
Marina and Svetlana research canola at the Ukrainian Scientific Institute of Plant Breeding in Kyiv.
For both countries, it's an important export crop, and for the globe, countries like Australia and Ukraine that basically provide the world trade of that commodity, what happens in those countries, in both of our countries, is important to world trade.
And it's what's happening in Ukraine right now that has brought these young scientists to Australia. When war broke out, the US-based Breakthrough Prize Foundation teamed up with the Australian Academy of Science to ensure Ukrainian researchers could continue their work.
Ukraine is actually a powerhouse, particularly in genetics and in plant science, so it's important for us to keep those links open. But whatever helps Ukraine also helps Australian science and international science.
Like everything, the work of scientists has been seriously interrupted by the conflict.
It impacted all spheres of our life, not only science, because you always are under stress, you're always worried about safety of yourself, of your family, your friends, and it's hard to plan your experiments and projects when you think just how to survive.
Але життя змінилося, але ми якось продовжуємо працювати. Дуже вдячні всім, хто нас підтримує, всім країнам, також Австралії, які надають допомогу.
One of their hosts in Australia was CSIRO agricultural scientist and fellow of the Australian Academy of Science, Dr. John Kierkegaard.
They told me sometimes they're travelling along roads which have mines. It takes them hours, 10 or more hours to travel safely to see some of their field experiments. Quite challenging, and I found them remarkable, but in their pragmatism and determination to sort of keep the work going.
Dr. Kierkegaard had hoped other Ukrainian scientists would join the Australian trip, but fighting-aged men have been barred from leaving Ukraine. Australia has made sure they still have support, including remote access to scientific infrastructure, including supercomputers, microscopes and telescopes.
Science and technology has clearly underpinned the growth of canola as a crop in both countries. The only way to sort of stay ahead of that game is that scientists around the world collaborate, share ideas, share information, and that's what conferences like this and exchanges like this can really promote.
Ukraine knows science will play a major role in rebuilding the country after the war.
And it's all about the human capital. We can rebuild buildings, right? It's not a problem, but we need to keep our human capital. It's our competitive advantage, the talent that we have. And we need to nurture the talent. We need to help the talent to grow, develop, and become better. And I think this will be of huge contribution to Ukraine's reconstruction eventually.
The support of Australia and Australian people are really important for us and for Ukraine now, because I know it's important to feel that we are not alone in this need. And we really appreciate all that you do for us.
I love doing cryptic crosswords. I love doing things like Sudokus and stuff. And doing maths research is like that, but souped up. I'm Aidan Sims. I'm a professor of maths at the University of Wollongong, soon to move to the University of New South Wales.
So 1 minus e to the 2 pi.
I'm a mathematician.
So now I guess
I research mathematics and I teach mathematics.
Even negative.
And I love both of those bits of the job. I guess the research that I do, I would call quantum mathematics. What we're trying to understand is mathematics that first came up when scientists were starting to see that things behaved very, very strangely at the very small scale of quantum physics. We need mechanisms, we need ways to picture it, we need ways to manipulate it to help us design, hopefully, quantum devices, quantum computers. But it's a long research pipeline, and what we aim to do is listen to the physicists, listen to the applied mathematicians, find out what it is that they need to know. But we also, I guess, are always conscious that there's a value in doing mathematics because it looks interesting and we're curious about it, because there's such a history of that leading to completely unexpected technological advances. That they couldn't have happened if we didn't have the mathematics, but no one knew that that was the mathematics we were going to need. That mathematical theory. The stuff that I started on when I was a PhD student, this was a new visual model for these quantum mathematical systems. And I guess it was something like a 12, 13-year journey from when I started to a paper that I wrote with a couple of collaborators from Denmark and from the States, where we answered a really long-standing question about the structure of these operator algebras using these higher rank graphs. That was a big one. And more recently, there are two sorts of algebras that we deal with, C-star algebras and abstract algebras. And we were seeing that in certain classes of them, there were incredible similarities between what was true about them, but the way that you proved it was completely different. We couldn't see the connection between them. And I had this idea that something that we had in our toolkit called groupoids might be the way to do it. And it was a bit of a long haul, but over about probably a 5-year period, that's what we did. And sure enough, in the end, you can use the groupoid as something that underpins both, and it translates the theorems back and forth. So that was a real proud moment. The people that I see elected to the Australian Academy of Science really are the leaders, like the top people in Australia, and I sort of find it hard to feel that my name belongs in that list of people, but it obviously makes me immensely proud. There's no such thing as just being good at maths or bad at maths. You practice and you get better at it and you struggle with it, and every so often you make a breakthrough, and for just a moment you feel, wow, maybe I'm actually okay at this.
Because that's really important. My job is working on making solar panels more efficient, affordable, and sustainable. My name is Xiaojun Hao. I'm a scientific professor at UNSW. The most used solar panel today are silicon. They actually works very well. But the thing is, they can only use part of the sunlight efficiently. We see some dead area. You can think of sunlight as the multi-layers of different energy, like the different colors in the rainbow. For if we can think of the silicon as a sponge, this silicon can only absorb part of the layers. If we can use two sponges, the top sponge can absorb the higher energy light. The lower sponge can absorb the lower energy light. This is exactly what the tandem solar cell going to do. So for the current silicon solar cells, their efficiency just about 27%. It's so close to approaching their theoretical efficiency limit. For the tandem solar cell, it can go beyond 30% already. And in the future, I believe it can go to beyond 40%. For the top cells, we want the top cell to pair with silicon. To pair with silicon means we need to have the similar properties to silicon: earth abundant, non-toxic, and last long, like cassette, making from copper, zinc, tin, and sulfur. So now sometimes people ask me, how many kids do you have? I have 2 kids, but I also have another one, cassette. We also invented a technology. That's what we call the TCA technology. That stands for transparent conductive adhesive. By using this one, we can easily stick the top cell and the bottom cell to form a tandem solar cell. The sodium and also the oxygen move from the bottom. My team, we have already achieved the world record efficiencies in a number of top cell material candidates. For example, cassette, antimicrocogenate, and perovskite. I want to say tandem solar cell is really going to open the uses in the places where space is limited. For example, building, vehicle, and even satellites. I'm incredibly honored to be elected as a Fellow of Australian Academy of Science. I feel deeply humbled to be recognized alongside a lot of scientists whose work I admire. But this recognition definitely also to my field of solar photovoltaics, which is so critically important and particularly important to shape the future to the net zero.
For many patients, chemotherapy is a bit like shifting deck chairs on the Titanic. That drives me every day to try to make new discoveries and to generate better drugs. My name is Payne-Scott, and I'm a medical oncologist and a clinician scientist. Lovely to see you. Have a seat. The patients in my clinic really form a crucial part of what has directed my research over the last number of decades. And I've focused on targeted therapies, and particularly for rare gynecological cancers, including ovarian cancer. I really became a bit obsessed about cancer and the way in which cancer cells were controlled. DNA repair is a normal process in every cell, but in many cancers, these repair systems are broken. So by understanding DNA repair, we understood that a drug called a PARP inhibitor was incredibly important. We introduced the PARP inhibitor initially for women who had a boutique setting. They had what's sometimes referred to as the Angelina Jolie BRCA genes. It was the first really good example of an inherited cancer susceptibility being used as a target for cancer therapy, and was profoundly successful. We set up the WeHi Stafford Fox Rare Cancer Program to be inclusive of all people in Australia who had a rare cancer. And that's because the ethics and governance, the red tape for studying cancers, particularly rare cancers, is just a profound wall. So this has revolutionised the types of questions we can ask and then answer. And now we're moving our targeted therapy experiments into the realm of immunotherapy. I can't say more strongly how personally this affects me. We have women with gynecological cancers who have not had sufficient research into their cancer. And perhaps that has also led me to this conviction that unless we engage the immune system at the same time, we will not succeed for enough patients. One of the features of being a clinician scientist means that we're always dabbling in someone else's pond. As a clinician, I work on a weekly basis with amazing clinicians who spend all their time being a clinician, and I can never be that good. Equally in the lab, as a scientist, I work with people who are pure scientists. So in a way, I feel like I don't make the grade in either of those areas. But what this recognition by the Australian Academy of Science means to me is that my scientific colleagues recognise the basic components of scientific research that I've managed to achieve. And that is extremely important to me personally, because at the end of the day, science will lead us more safely than anything else will through every course of our daily lives.
My team pioneers a polarization method that can efficiently increase the photocatalysis performance by up to 100 times. This technology can largely lower down the cost of producing green hydrogen. I also focus my work on carbon capture, utilization, and storage. My team integrates CO2 capture from industry processes with CO2 upgrading into value-added chemicals. This is one of the most important processes to help us to achieve net zero for Australia and also globally.
When I got into aerial robotics, it was 1998. I love working in a field where I feel like I'm contributing to society. My name is Rob Marnie. I'm a professor at the Australian National University.
And so what I'm doing is I'm actually acting standard symmetry on P.
I work with geometry and I work with symmetry, and I'm really trying to apply the mathematical theory of symmetry to physical systems, to robotic systems.
Act on pose.
What I love about these problems is that you can do quite sophisticated mathematics and interesting control problems, and you can actually do that for problems that matter, for systems that actually go out into society and make a difference. There were algorithms that worked, but they really required military-grade electronics, and there was nothing that worked reliably with consumer-grade electronics. And so we found this symmetry, this Lie group, the special orthogonal group, and reformulated the filter on that group and gained a huge amount of robustness. And eventually we actually got a filter that worked. It was picked up by all of the major drone companies of the era. So from 2005, 2008, all of those initial consumer drones were flying that filter. There's huge advances in society that have come out of the drone industry. I'm working at the moment on a project in bushfire detection, so where we're flying drones over the Brindabella Mountains at night after lightning storms and looking for ignitions.
You're 10 degrees to the right.
What you wouldn't want to do with a crewed aircraft, quite low, so a very dangerous flight for a crewed aircraft, but quite safe for a drone. But I'm also working on submersible vehicles. I'm interested in the same transformation in submersible vehicles. So you have submersible vehicles available at the moment, but they're very expensive. They're run by specialist firms. They require very expensive sensor units. And so I'm researching at the moment into consumer-grade localization, state estimation, communication systems to try and create the same capability in underwater vehicles as we've seen over the last 20 years in aerial vehicles. I work right from the very fundamental theory, but it's very important that I'm also trying to put algorithms and understanding how these things work on real systems. And seeing the real system, when it fails is when you learn the most. Really, it's been a life goal to become an Academy Fellow. I will certainly engage in the Academy, and there's a number of projects that I'd like to do to start supporting the next generation and looking at technology transfer in Australia. But at heart, I'm a researcher. That's what I do.
We need to always think the big question, how to feed 10 billion people by 2050. Will be great to see a world where we don't have anybody hungry. My name is Rajeev Varshney. I'm Director for Center for Crop and Food Innovation and State Agricultural Biotechnology Center at Murdoch University. I'm an agricultural scientist who specializes in genetics, genomics, and molecular breeding. We try to identify traits that are responsible for how well a crop can deal with drought, or be more resistant to diseases, or grow well with less fertilizer. Genomics takes this one step further. Instead of looking at just one gene at a time, genomics examines the entire instruction manual. It's like the difference between reading one page of a cookbook versus studying the whole cookbook to understand how all the recipes work together. And you see that some plants are short, some are tall. Traditionally, breeding was a case of trial and error. But in molecular breeding, we can look directly at the DNA and identify the genes we want that then breed for those specific traits. The recurrent parent genome recovery mode, we are working on horticultural crops and most importantly, cereal crops such as wheat and pulses crop like chickpea and lentil. Together with the partnerships with many countries, we decoded the genome of chickpea in 2013. Then in 2021, we published a paper where we presented the chickpea genetic variation map for 3,000 chickpeas. And now we have completed the genome sequencing of 10,000 chickpea line, which is a largest plant genome sequencing project. But the most important aspect from this genomics research is that we were able to identify the genes associated with the drought tolerance. And we delivered the improved chickpea varieties to the farmers in India, Ethiopia, Kenya, and Tanzania. And I'm so happy to mention that these farmers, they are growing those chickpeas, they have enhanced their income. This plant, now way back at home here in Australia, we are working with the GRDC, with Chickpea Breeding Australia, and we are trying to introgress the QTL hotspot in Australian chickpea varieties so that we are enhancing the farmers' profitability here in Australia, but we are contributing towards the food security around the world. It's an immense honor and privilege to be an Academy Fellow and to be listed alongside with the true titans of the scientific community. And I'm eager to play my role, delivering tangible research outcomes that benefit all Australians here, as well as to the other people around the world.
When I see these attacks to the science, I only feel that we need to be even stronger than ever before, more unshakable than ever before, because the evidence just keeps accumulating. There is no question about the anthropogenic origin of the current global warming. I'm Pep Canadell, and I'm a chief research scientist at CSIRO. So the carbon cycle is the whole flow of carbon anywhere from plants and the ocean into the atmosphere, and humans have come here and they have introduced a huge new flux, which is the combustion of fossil fuels, which were buried millions of years ago. Plants take some of the carbon dioxide, they grow, they store it in roots, in soils, in the trunks, and then the oceans also absorb CO2. These, what we call the natural CO2 sinks, they take about half of all emissions coming from human activities. Nature's giving us a 50% discount on climate change, but the rest of the 50% stays, and it stays for thousands of years. And so we have more energy for any major climatic event that might happen, either hurricanes, tornadoes, the heat extremes, the marine heat extremes, everything is kind of more pumped up because of the extra energy. We have multiple independent evidence that is helping us to understand that this is what we're doing, and it's not the natural world. We have this huge amount of carbon frozen in the northern latitudes. Most of it, of course, has been frozen for thousands of years. So what we've done is, for the first time, the complete greenhouse budget of the permafrost region. An amount of carbon, it's more than double the total amount of carbon that is already in the atmosphere. As temperature warms up, we are beginning to expose some of the surface layers to thawing. We want not to go unchecked into the future because there'll be no limit to the amount of carbon that can go into the atmosphere. Make the budget talk to the atmospheric inversions, because atmospheric inversions, they see everything. Over my life, I've been blessed by being able to actually work with a lot of great scientists from all over the world. We have been able to develop these global carbon budgets and budgets of other greenhouse gases, which are now the fundamental pieces to track and attribute climate change to the very sources of either human or in the carbon climate feedbacks of nature, which are contributing to the accumulation of CO2. Being fellow of the Australian Academy of Science means a lot to me because it is a recognition to the team work that we've done here in CSIRO and globally with these huge networks that we have been able to create through the Global Carbon Project. I'm grateful to the Academy that will be able to amplify some of these key climate science information to really accelerate the process of change.
We've noticed something slightly weird. Gravity, when you go onto the biggest scales, appears to be pushing, not pulling. Something out there has some sort of anti-gravity, and we're trying to discover what it is. My name's Professor Tamara Davis. I'm an astrophysicist at the University of Queensland.
So this is our remote control room for the Anglo-Australian telescope.
I study cosmology, which means I study the universe as a whole. I try and understand how the universe began, what the laws of physics are that govern it, and how the universe is going to end. My first memory of being intrigued by astrophysics was when Halley's Comet came by when I was about 10 or 11. Now, my parents took my siblings and I out on a houseboat and they showed us this comet that spread like halfway across the night sky. And while lots of people were going, oh, isn't that pretty? I was sitting there thinking, how do they know that this comet is going to come back 75 years from now? The fact that humans could understand something like that and know that it was going to come back, that caught my interest, and that's my first spark of being intrigued by astronomy. We now know that most of our universe is made up of dark components that we can't see with our eyes. The first is dark matter. We think it makes up about 25% of the universe and it binds galaxies together. The second is dark energy. It is the name that we give to whatever is accelerating the expansion of the universe. So it's the thing that seems to have a repulsive sort of gravity that causes the entire expansion of the universe to be speeding up. One of the things I think I'm most proud of is having made some of the biggest data sets that are the most precise that we've ever had to understand the expansion of the universe. Our dark energy survey just made a huge collection of distant supernovae, these supernovae that we use to trace the expansion history. If we can understand how the universe has expanded and how that expansion is changing with time now, we might be able to figure out what the fate of the universe will be. It's really a huge honour to have been elected as a Fellow of the Australian Academy of Science. You're joining a group where everybody is trying to improve the world in some way through their science. When I look at the vastness of space, it makes me realise just how precious our Earth is here. There's still sort of 100 kilometres or so of atmosphere that we have above us is all the air that we're ever going to have to breathe. It makes you think about all of these petty struggles that we're having here on Earth. What do they mean compared to the importance of looking after our planet?
This research summit has been fantastic because it's the inaugural one. It's the first of a series of 3. Next year it'll be hosted by New Zealand, and the following year it'll be hosted by Australia. Already we're seeing the enthusiasm by members participating, knowing that this is really the start of a process for some in understanding how Indigenous practices occur in other countries. I think there's some great opportunities coming out of forums like this between our 3 countries. I think at a number of different levels. One is just the information exchange, the knowledge exchange that takes place here at the summit, but also the contacts that we're able to make with each other. And we have got to establish a global network of like minds, and the academies are a great example of where we can get those like minds together to share the information and to learn from each other.
Okay, welcome back everyone. We'll start this next session. Yesterday and this morning we celebrated our newly elected fellows, and we now continue our celebrations with the awarding of our 2025 honorific awards. In our pursuit of advancing science in Australia and abroad, celebrating outstanding scientific contributions is at our core as an academy. The magnitude of achievements that we are celebrating today is truly staggering, and it is something that we would not be able to do without the support of our generous donors and sponsors. Before we hear from our premier awardees, please join me in acknowledging our event partners. You can see a list here. It's a long list, but please indulge me as I mention them by name. The Department of Defence, the Department of Industry, Science and Resources, the University of Sydney, University of Queensland, GSK, Digital Science, the University of Adelaide, Deakin University, the University of Melbourne, Monash University, Evans and Partners, QUT, Swinburne University of Technology, the Grant Ed Group, the Victor Chang Cardiac Research Institute and UniSuper. We also thank the institutions who are EMCR supporters. These institutions have enabled their early and mid-career researchers to participate in science at the Shine Dome and benefit from the network opportunity that presents amongst Australia's top scientists. We often hear how these conversations can spark long-lasting collaborations in the future, so please do take advantage of the opportunity. I'll acknowledge the institutions. They are UNSW Sydney, Macquarie University, University of Adelaide, University of Melbourne, University of Western Australia, Curtin University, Forest Research Foundation, and the NCI. We are also very fortunate to have had the generous support of our event partners and EMCR supporters to bring science at the Shine Dome to life this year. Now, I'd like to introduce our first Premier Award for 2025, the 2025 Matthew Flinders Medal and Lecture. This award is a Premier Career Award that recognises scientific research of the highest standing in the physical sciences and honours the contribution of Australia's early scientific researchers. Along with the Macfarlane Burnett and Ruby Payne-Scott medals, it is one of the most prestigious career awards of the Academy. The 2025 winner of the Matthew Flinders Medal and Lecture is Academy Fellow Professor Yuri Kivshar. Optics is an art of manipulating light through devices including lenses, gratings, and nonlinear crystals. Photonics allows us to use light for many applications. Professor Yuri Kivshar is a pioneer in optics who has described with his team whole new classes of materials and devices by combining the properties of nonlinear structures that are patterned on a sub-wavelength scale. He is globally recognised for introducing the field of metaphotonics using artificial materials with novel properties and multiple applications. Professor Kivshar is one of the founders of all dielectric resonant metaphotonics that derives unique functionalities from electric and magnetic Me resonances. His work has led to the development of innovative photonic devices such as chiral nanolasers, employed now in a new generation of optical communication systems, biomedical sensing, and defence and security applications. He has trained a remarkable number of students and early career researchers over his career, with many of them now holding leading positions in industry and academy worldwide. Yuri will receive his medal tonight at the gala dinner, but today we will hear his lecture. Please welcome Professor Yuri Kivshar.
Thank you, Ian. Great pleasure for me to be here and share my excitement about fantastic research which we are doing in photonics. And I name my talk metamaterials and metaphotonics, and I will try to explain you what is excitement is in these fields. First of all, I understand not everyone knows about light, so this is one of the images of light from a famous photo of Berenice Abbott. But you see light everywhere. Problem is that it spreads around, and it's no use unless you use something very simple like bulbs. But light is actually a very small fraction of what we call electromagnetic radiation. So it spans from radio waves to gamma rays, so you definitely came across of all these wavelengths in your life. So visible light is actually a very small part of that. And electromagnetic waves in physics are known to describe so-called Maxwell's equation. Maxwell is a great name in physics, and this is a statue of Maxwell in Edinburgh with a record of basic form of Maxwell equation. So amazingly, Maxwell equation actually describe all these types of wavelengths span around electromagnetic spectrum. So the question is how to control light, and of course we are most interested in how to control visible light or infrared light which can be used for different applications. There are several things which you already know very well, because currently the trend is to replace electrons in electronic devices by light, and this is happening step by step through different types of efforts and possibilities. So we use photons now for communications, and of course, I'm sure everyone knows about optical fibers. We use optics for storage, optics for laser machinery, for fabrication, imaging, a lot of applications which you may imagine if you remember this huge spectrum of electromagnetic waves. Of course, in terms of progress, we have a period now, some years of so-called photonic integration revolution to actually take larger devices which we use for light manipulation to scale them down to very small sizes. That was a goal of our Kudos Center of Excellence, which was for 15 years. Some of my colleagues are actually in audience. And the idea was to take what we know very well from yesterday expensive devices with low bandwidths, scale them down, and making them smaller, making so-called photonic chip, and then, of course, making smaller and more compatible with current technology, and then looking for future, future does not work very well. To achieve what? High functionality, okay, and low footprints. But still, there is a big question how to scale it down. Just to give you a very simple impression, imagine this is wavelength of light which is used for optical communication, 1.5 micrometers. But we want to scale devices much smaller sizes, a few hundred nanometers. And it's clear that wavelength of light, this is the size of devices, how you can squeeze light of that wavelength into a small device. And this is basically what nanophotonics is doing. This is basically our future to make everything much, much smaller and integrate with many useful, in particular, variable devices. Okay, so we come to concept of matter. Of course, the world matter is spoiled by all these social media technologies who stolen that name for their purposes. But originally, matter comes from Greek words, which mean beyond, and that was much earlier than social media took that name in terms of metamaterials. So very simple idea of metamaterials is just to mimic nature. Nature has all object composed of molecules and atoms, and basically you don't see what's happening inside. So the same idea with matter. So what we do, we take structures, we compose them of very small elements, which are much smaller than wavelength of light. This is example. And then if I try to fill it, of course I will not fill it because it's a microns scale of my roughness, and then nanometers, hundreds of nanometer size of object. So you don't feel it, it's like a smooth. But inside they consist of small elements which can be engineered at will. And that's our idea of metamaterials. So this is example actually from RMIT University from Kudos time, which shows you metamaterials which on a flexible substrate. Of course, you don't see that what is actually done inside, but it consists of small elements. This is an example from our lab, what we are using, different type of structures. Of course, where these elements are much, much smaller. We are engineered in a special way to achieve specific functionalities we want. The basic idea, if for those who knows Maxwell equation, is to control independently magnetic and electric component of light. So light or electromagnetic radiation, electric and magnetic. For that, we need resonances. We need structure to be sensitive to particular type of our components of waves. And this structure, which we do, composed of electric atoms and magnetic atoms. We call them atoms because a few hundred nanometers, still elementary units. Here is an example from very early days. So initially we use plasmonics, and then adding magnetic resonances, we actually create structure sensitive to both electric and magnetic field. This field started about 20 plus years ago. That was a special issue of Optics Photonics News, celebrating some achievements of our pioneers who first predicted and demonstrated such structures. But of course, it's always was the idea to move from bigger structures, from microwave, as I mentioned, driven by nanophotonics ideas, to optics. First time when I heard word meta-optics actually was when I collaborated with fellow of our academy, Ross McFadden, a retired professor from University of Sydney. When we write in a paper back to 2011, he actually suggested name meta-optics. And then later when I asked him what was the idea, and he actually explains that he tried to explain, to put the concept that optics has started to use ideas from metamaterials, metaphotonics. That time, it was no meta-optics at all, no single example, and it was not clear if it ever happened. So what was interesting in the initial days of meta-optics? With meta-optics, we can engineer responsive magnetic and electric responsive. And for that, we can actually change refractive index from positive to negative. It's a very amazing concept, making refractive index negative. And that was a lot of hype around that. In particular, if you look at lenses with swap, convex to concave, and that produce interesting ideas like breaking of diffraction limit, was many discussions, many interesting things. And this is a first experimental verification, of course, for microwaves, because initially microwave were used to verify all these ideas. And then, of course, a lot of hype. As always, when new field appears, journalists and all this media attracted by hype. So hype was creating invisibility clock. Why? Because rays now can be bent in a different way. So we can construct a device bending light, which is impossible for normal media. And then invisibility clock was a very big hype. We also were involved in that hype. Of course, our media also not different from international media. We call it waving Harry Potter magic. Collaborated with the Department of Jagadish. Was a lot of fun these days. But if you look at what was used for a long time in this field was metals. So metals were actually used to create effective magnetic moments. to control magnetic component of electromagnetic waves. All this yellow is metals. And of course, the main idea was to create resonances for magnetic component of light. For some time, it was a lot of fun, but it was not clear if you can still do optics. Because if you move metals to optics, they become very lossy. And metals can't actually perform well at visible or mid-infrared wavelengths. So idea came actually suddenly from several related investigation, and as always in science, you need to look in history and see what happened before you. And just in 1908, me theory actually was developed by me, who did not have a mathematical, fancy numerical simulations. He just solved Maxwell equation exactly by pen and piece of paper. You know, this is his solutions. And then he actually described how light interact with a sphere made of, it's a theory, hypothetical material. This is some flavor of me resonances, if it will work. Let's try. This is 2 grapes in microwave. So we are not big, but don't try to repeat it at home. And 2 grapes touching each other and we create magnetic plasma. This is a result of waves which are accumulated inside. This is a kind of flavor of me resonances. Again, don't try to repeat it. And that came to idea of metronic or me-resonant photonics. We need resonances to control both electric and magnetic fields, and that somehow evolved later on in the name of metronics, which is now used and had more than several hundred citations. Okay, so we go ahead, and this is all a bit of physics, but now comes to some applications. What's happening now, it's happening revolution in optics, because we can actually use these elementary units to cover surfaces by them and engineer properties of surfaces. Surface is absolutely flat, so we can create lenses, so-called flat lenses. So this is example that we replace polished pieces of glass and decouple shape, specific shape for lenses, just using only flat surface engineering elements. And decoupling shape and function gives you a lot of advantage. This is where people believe it will be a lot of achievement. This is some example of our fabricated structures from germanium, but I just give you more basic information. So what we are doing, we're replacing lenses with very flat, actually it's not 0 thickness, it's a half of wavelength thickness type of structures. What is important, we can replace in your Canon cameras, this type of lenses, we can replace by much thinner structures which will use several of these meta lenses. And that's where field of meta optics actually expect a lot of progress in the future. Of course, we discovered some resonances. That's probably, I just give you an example of structures which fabricated, but also among applications which we developed, I want to mention biosensing. That's a 10 by 10 metasurface created from pixels. Each pixels create a resonance, and then it's combo of, in this case, 10 by 10, 100 resonances, allows you to do sensing without any specific spectral measuring devices, just in a full transmission. So many different applications of this type of ideas, and this is the most recent one, just published a few weeks ago, so-called meta laser. So lasers generate usually Gaussian beam, and then people use in the lab different type of devices to actually structure this laser beam to create particular shape. So we can actually create a hybrid structure, combining gain and specific shape creating device from meta surface with a specific arrangement of these resonators, and actually create shape, any type of shape immediately from a single surface, active surface. I just selected few applications just to give you a flavor where it may go. It may go to different directions already happening. These applications from my former students and postdocs, which already realized. Believe it or not, we can reduce reflection of solar panel stacks. This will give you a saving of several percent, which is a huge number in terms of money associated with the solar cell technology. This is my former student working in Germany. Now we can, this is not very important now after COVID, but just before COVID, that was a very big splash of activities and security screening. So instead of all these frames and everything, you just go through and then system indicate when you can be checked in less than 1%. Believe it or not, stealth technologies are based on metamaterials. Initially, when people use that, we did not mention metamaterials, but metamaterials are very important because this is a structure based on antennas. MRI, you can do patches, you can apply it for specific areas, and dramatically reduce time in patients spent in a chamber. This is actually terrible things to stay there. And more recent one, for example, you can introduce holograms in your iPhone, a lot of sensing application, but still it's very hard to do in terms of application because it should be cheap. Anyway, I suggest you a few messages we should remember. So metamaterials, meta again, word beyond, is a new exciting field of research in physics. I didn't mention about acoustic or mechanical metamaterials, a very simple idea, also applied for completely different class of materials. For example, you may engineer negative Poisson nature. Then it's actually new and very exciting field in optics. We got a new life in the terms of meta-optics because we can use optics wavelength and metaphotonics. And then recent advantages I mentioned related to resonances. This is very important, and this is where our contribution was critical. And then I just mentioned about meta surfaces, meta lenses, and that's what we expect to see in a very, very near future. At the very end, I wanted to mention, I still have time, that I'm very grateful to Academy of Science because I was very happy to actually got all 3 awards from Academy associated with physics. I came to Australia in '93, and then in '98 I got Posey Medal, just to make sure that you understand that it's for different fields. I just put specific words from citation. This is for optical solitons. I was doing optical solitons before coming to Australia from Soviet Union, then France, Spain and Germany. And then I got Lyle medal. It's for periodic photonic structures, including photonic crystals. And I was very, very happy and grateful to get third award because I changed fields. So basically I moved from one field to another one. This is what I called my 4th life in science. Matthew Flinders medal for metaphotonics and for applications. And I believe this is very special medal in Academy because I knew name Flinders for many years. Of course, when I started to read about Australia, even before coming here. And just for people who do not know that Matthew Flinders was one who actually suggested the word Australia. And it happened exactly 222 years. Very remarkable. Finally, I just want to mention that it's extremely important in the life of any scientist to have good advisors. And I was very lucky to actually get a very good advisors. You remember, I started from the Maxwell, and I will show you how I connected to Maxwell. There is a site which Academic Genealogy, which you can actually apply for yourself. And my advisor was Professor Kosevich. Most of them, of course, all of them passed away. He was co-author of the famous 7-volume of book of Landau-Lifshitz. His advisor was Lev Landau. People who are working in physics, we know his name very well. Lev Landau was coming from Niels Bohr. Niels Bohr has Arthur Schuster and finally wrote Rayleigh and James Maxwell. Thank you very much for your attention.
Thank you, Yuri, for that wonderful talk, and what an amazing genealogy. I know what I'm doing this weekend. I'm going to go buy some grapes. We have time for questions, so please raise your hand away from a microphone because people online won't hear you without the mic. Those of you online, there's a QR code, so please submit your questions and we can have them. Those of you up top, there's a microphone. Any questions? Francis, just mic coming.
Oh, thank you and congratulations. I'm just going to ask a very specific question because you mentioned MRI. So are you using optically enhanced magnetic resonance? Is that how you're enhancing the signal?
Thank you for that question. Oh, okay. Actually, we tried to push that activities in Australia, but we applied 3 times for NHMRC grants and never succeeded. Now this moves to UK. And together with Russia, we created company and we have patents. So no, this is not optically. I mentioned electromagnetic spectrum is very broad. Of course, we're enhancing microwaves, so magnetic resonance associated waves. So we create structure which will be responsible to resonances from MRI. And why do we need that? Current machines are very expensive. To replace machine in a hospital, it's a big amount of money. It will happen in maybe once in 30 years. And so what the idea was to use patches. Imagine that we create patch which will be resonant, cover it by wires, because that set frequency range wires will be very useful in different structures. Then you apply it, so we have patent and actually products for breast cancer. You apply patch, you go to MRI machine, and believe it or not, it's much easier in terms of timing. Initially, of course, quality of picture will be very high, so much higher quality of picture because of enhancing of resonances. But most important, when we talk to people actually dealing with MRI machine, the most important advantage to reduce time spending there. I did MRI once, and it was terrible. 20 minutes, and that, many people actually claustrophobic, 20%. Yeah.
Thanks for your presentation. Yeah, I have a question about the matte material. So for them, how about the conductivity? Do they have some special material with good conductivity? So I'm thinking the application in the electrochemical sensor. So for the electrochemical sensor, we should have the material with good conductivity. Thank you.
Thanks for that question. Again, it depends on wavelength and compatibility with electric circuits. So electro-optics is now an agenda of application, but there are many different types of activities. I initially had a slide which show how this field diverges. Now there are groups which do not talk to each other. So they do what they come from the basic idea and do completely different things. Just very simple example is biosensing, mainly driven by biologists and chemists. Then there is people who work for security. They basically disappeared from literature, as you understand why, because it's all related to military applications and all this. This conductivity is actually very important, and people are looking for new materials, conducting materials, so combining that high resonances and properties of conductivity. So it's not just normal materials which we're using, but several groups are doing that or trying to do that. Thanks for your question. Yeah, it's very important, but it can develop in any different direction, because the main idea is that we have structure which actually much bigger, which you don't see actually elements, but you can engineer elements, and you can engineer specific properties imposed on that structure. That's a basic concept. It applies to almost everything. I mentioned that mechanical one, and that's a basic concept, and people started to use it for developing materials which do not exist in nature at all. Nature does not allow many of the properties here. Thank you.
Thank you very much for the interesting presentation. You mentioned resonances, and so in my mind I'm imagining very easily MRI, it's very narrow band. But you also talked about replacing a camera lens, for example, which is quite broadband. So do these materials already, you have solutions for these more broadband cases, or is it more narrow?
Very, very good question. For broadband, of course, you need to avoid sharp resonances, as you understand. So there are different developments of that. For example, meta lens. Of course, you can do it for specific frequency, and you can do it broadband. You can compensate achromatic properties. You know, it's a lot of possibilities. The question is how you will design it. Of course, we had about 15 years ago, when all this business started, we tried to combine broadband operation with their highest transmission. Of course, we can't achieve highest transmission, so roughly it was meta surfaces with a transmission about 80%. Close to 90 will be very difficult because we're usually on substrate, so substrate itself is lossy or absorbive. But we can span, for example, across free telecom bands and employ it, for example, in printing different type of structured light in the fiber, because now in fiber communication, people feel how to make information packaging more denser. So this means that we can use not only Gaussian beam, but high order beams like vortex beam, high order vortices. That's what we had experiment with Göteborg University when use fiber and actually imprinted from metasurfaces different type of such structures, and that's covered actually free telecom bands. So that's also a possibility. But of course, all of us trade off because you can't get very sharp resonances and broadband. That's a good question. Yeah, definitely. Many people are working on different aspects, and yeah, that's true. Thank you.
That's the last question. If not, I'd like to ask that you join me again thanking Yuri for an inspiring and wonderful lecture. And now I hand over to Professor Bob Graham.
Thanks very much, Ivan. So look, I'm delighted now to introduce our second Premier Award for 2025, the Ruby Payne-Scott Medal and Lecture. This award is a Premier Career Award that recognises women researchers of the highest standing in the physical or biological sciences. It's one of the most prestigious career awards of the Academy and honours Ruby Payne-Scott's pioneering contributions to radiophysics and radioastronomy. The 2025 recipient of the Ruby Payne-Scott Medal and Lecture is Academy Fellow Professor Jane Visvader. Professor Visvader is an esteemed scientific researcher in the field of breast stem cell biology, having made seminal discoveries that continue to define how the breast develops, how errors in the development lead to breast cancer, and how we might prevent or treat that cancer. Her outstanding contributions encompass fundamental research, translational discoveries, and an inspiring commitment to teaching and mentorship. Jane's research leadership has provided a series of important discoveries that underpin our current understanding of breast stem cell biology, including the isolation of mouse breast stem cells, which provide methodologies now used in research laboratories worldwide, defining the sensitivity of breast stem cells to ovarian hormones and their impact on the development of breast cancer, discoveries of the cellular origin of BRCA1-associated breast cancer, and validation of a specific cell signaling pathway as a therapeutic target to prevent breast cancer in people carrying a BRCA1 mutation. Jane will receive her medal tonight at the gala dinner, but now it's my pleasure to welcome her to deliver the 2025 Ruby Payne-Scott Lecture.
Thank you very much for those generous words.
I am deeply honoured to receive this recognition from the Academy and to help honour the legacy of Ruby Payne-Scott. Obviously, she was one of the world's first radio astronomers, and she did so at a time which was virtually impossible for female scientists. I'm going to start with the acknowledgements. I did, actually, Yuri, in your wonderful presentation, just reminded me to reflect on my career path. I haven't always been a breast cancer researcher. I actually started life as a plant virologist with Bob Simons in Adelaide, then moved to leukemia, and finally ended up with breast cancer, in breast cancer research. So this award reflects the hard work of many talented students and postdocs that have been through our lab over about 2 decades. And in particular, I would like to acknowledge Jeff Lindemann, my close collaborator from the inception of the Breast Cancer Laboratory, someone who has also paved the way for our work on human tissue through the creation of one of the first tissue banks in Australia. I'm also indebted to Gordon Smythe. I have worked very closely with this extraordinary bioinformatician over many, many years, and to my mentors, many mentors, but especially Jerry Adams, Suzanne Cory, and Doug Hilton. Right, well, breast cancer needs very little introduction. It remains a significant problem. In fact, 1 woman dies every minute globally, so there are more than 685,000 deaths worldwide each year. And it is not a single disease, it's a very heterogeneous disease. There are about 20 histopathological subtypes, and this impacts on clinical behaviour and response. And our laboratory, from the start, has tried to understand mechanisms that lead to breast cancer through the prism of normal breast development. And we see the little lobules in which these are the tips of the branches where breast cancers usually arise. So the mammary gland is a highly dynamic organ. During puberty, we start off with a fairly rudimentary tree which undergoes dramatic expansion to form a beautiful branched tree. And each duct contains two layers. There's the blue and the green. So the blue is the cuboidal and the green cells are the highly elongated and contractile myoepithelial cells. So this is all done by high resolution 3-dimensional imaging by Anne Rios in our lab. It's truly spectacular. Next, this is a short journey through the lactating roots. A short journey. No? Thank you. A short journey through the lactating mammary gland where we see milk-producing alveoli come into existence, and you can actually visualise these sacs of milk-producing cells very closely here, and they're encased by baskets of these myoepithelial cells. Right? Nope. This is very, sorry about my apologies. So the mammary gland is not simply a ductal tree, but it is embedded in a complex stroma composed of connective tissue and immune cells. And in the early 2000s, we asked the question, was it possible to identify an adult stem cell present in breast tissue? And indeed, Marc and Francois were able to show that only one subset, the DP subset, comprised cells that were capable of reconstituting an entire mammary ductal system. A single cell, in fact, could fully reconstitute a functional mammary gland, and this was truly a eureka moment. And when I say fully functional, it went all the way to milk production. So we worked for many years to decipher the hierarchy, and this was really my foray into stem cell biology. We subsequently identified common progenitors that lie downstream of the stem cell, and these progenitors go on to give rise to the mature cells that line the ducts of the mammary ductal tree. So the stem cell sits at the apex of the hierarchy, and then in a very regulated fashion gives rise to these daughter progenitor cells, so forth, onto mature cells. These cells can also be visualised in vivo. Here I'm showing a mouse mammary gland. And if we zoom in, for example, onto the red clone there, you can see that there are two cell types present in this particular clone. And that indicates that a bipotential stem cell gave rise to this particular clone. So we can track these cells in vivo. Not all cells can do this, only a rare population of cells have this bipotential capacity. Moreover, stem cells have special properties of longevity. They're incredibly long-lived, and they contribute to ductal maintenance in the longer term. And here with these unicoloured domains, they are actually being formed by one or very few stem cells. That is, a stem cell one stem cell has been responsible for maintaining the integrity of a specific region of duct. If we move to the earliest stages of tumorigenesis, we can also track stem cells and their progeny. If we look up in B and C, we see a plethora of colour indicating thousands of individual clones, and they're one of four colours. As tumour progression occurs, one can see that unicoloured domains become apparent, and finally in the tumour shown in G, we see an example of profound competition occurring between these clones, because there were thousands of clones in the pre-neoplastic period, hundreds in the hyperplastic period, but by the time we get to the tumour, we see here that a handful of clones had won out and contributed to tumorigenesis. So in vivo, moving away from in vivo and going ex vivo, again, these cells have very special properties. Only the basal or myopithelial cells have the capacity to form two types of cells. We see the red and the green on the left-hand side, whereas the luminal cells, because there are two branches, do not have this ability. And these are so-called mini organs because they're not perfect ducts, but we have the inner luminal cells encased by the outer red elongated cells, trying to form a mini organ. Most importantly, we've been able to extend these studies to human breast. Again, only one population, one flow cytometry population was able to give rise to human breast lobules and ducts upon transplantation. And it's very important to note that the cell constituents in human and mouse breast tissue appear to be identical despite quite large changes in architecture and anatomy. Remarkably, the stem cell in both human and mouse lacks the receptors for the key ovarian hormone receptors, estrogen and progesterone. However, hormone deprivation was shown to drastically reduce mammary or breast stem cell numbers and their function. Conversely, sustained hormone exposure led to stem cell expansion. So these findings really provided evidence that sustained hormone exposure can predispose to breast cancer by expanding the stem cell pool. And the data really provide an explanation for this link. It's a very well-established link between sustained exposure to female hormones and breast cancer risk. The team went on to map many regulators. Two of my favourite master regulators are shown here. FOXP1 is essential for activation of dormant stem cells. So these stem cells are normally in a dormant state. They need to be activated, and FOXP1 is the master regulator of this process. GATA3, on the other hand, it's one of the most highly mutated genes found in breast cancer. It is essential for the maturation of a progenitor to a mature cell. Hence, highlighting why this gene is so frequently mutated in breast cancer. It's critical for differentiation. So moving along to more recent molecular analyses, we have begun to deconstruct the complexity within breast tissue, cell by cell, that is, at the single cell level. And we've done this with a fantastic team of people. For the mouse, it's led to the discovery of novel intermediates. Perhaps more interestingly, in the case of dissecting human tissue at the single cell level, we've been able to visualise this little pink, this little pink area down here, which is the first time we could actually appreciate the unique molecular characteristics of stem cells. It needed to be achieved at the single cell level. So this is for human breast tissue. So now turning to breast cancer. I mentioned that there are many pathological subtypes. There are also 5 major molecular subtypes shown on the right-hand side. And a key question in the field has been, do these distinct molecular subtypes of breast cancer emanate from a distinct cell within normal breast tissue? That is, are there different cells of origin of cancer? So we, through the bioinformatic expertise of Gordon Smythe, we were able to have a look at the potential etiology of the different subtypes of breast cancer. And through comparisons of the gene signatures of normal breast ductal cells and the different subtypes, molecular subtypes of breast cancer, we saw remarkable correlations. For example, the stem cell has a remarkably concordant gene signature to the very aggressive Claudin-low subtype of breast cancer. We next asked, were there alterations in breast stem or progenitor cells in precancerous tissue from BRCA1 or BRCA2 mutation carriers? And these individuals are highly predisposed to both breast and ovarian cancer. Indeed, we were able to show that the luminal progenitor cell is the culprit. It is the main culprit that likely gives rise to triple-negative BRCA1 tumours. And this debunked a long-held theory that it was the stem cell that was the culprit. It is not. It is the daughter of a stem cell, this luminal progenitor cell. This cell is expanded. It expresses a molecule on its surface called Rank. Rank responds to its ligand called Rank ligand. And it led to the idea that switching off these errant cells in breast tissue might prevent the formation of breast tumours. So how do cells of origin in normal tissue lead to breast cancer? Well, in this case, we have this aberrant population, because it's very augmented, of daughter progenitor cells. We know that they accumulate DNA genetic errors at a very high rate, and these eventually culminate in breast tumours. So through blocking signalling along this rank axis, we hope to prevent or delay the development of tumours in patients. And this work has culminated in an international randomised breast cancer prevention trial, and it's been spearheaded by Jeff Lindemann and Christiane Singer. So that's very exciting. So we applied the same principles to normal tissue from BRCA2 mutation carriers. This was a little more difficult than for BRCA1. It required multiple single-cell technologies, both proteomic and transcriptomic. Nevertheless, we were able to pinpoint an aberrant daughter cell, again a daughter cell, not the stem cell, and these are called ERB3 low cells. They were expanded and they showed a uniquely perturbed signature that had upregulated protein translation, both the RNA and protein level. And to cut a long story short, and quite remarkably, we were able to visualise these aberrant regions in precancerous tissue from BRCA2 carriers. And this has led to, a proof of principle for a future prevention trial, because we showed that everolimus, currently in use for breast cancer patients, can actually switch off the activity of these cells in mouse models and prevent or delay the formation of tumours. So for the last few slides, I wanted to return to our single-cell work and more recent work where we are applying different single-cell technologies to really try and decipher the ecosystems both in the normal tissue and in tumours. So if we firstly, this is important if we perhaps go down to tumours. So while normal ducts exist in a complex stroma, tumours actually comprise a remarkably complex ecosystem with many types of immune cells and stromal cells. And we've profiled more than 1 million cells from 80 breast tumours to date, with the purpose of uncovering cell-cell interactions that promote the growth of tumour cells. And this is important if we are to find new biomarkers and ways to treat breast cancer, particularly for the many women that relapse. They may show an initial response, but there's relapse. So if we just go to I wanted to quickly talk about the normal tissue context. So using single-cell high-resolution imaging, Caleb actually discovered a novel population of macrophages that are crucial for maintaining normal tissue integrity. So these sense damaged cells and then engulf them. And I love this movie, I had to show it. Back to the breast cancers and the single-cell analyses. We have profiled many tumours, and we noted that ER-positive, the hormone-responsive tumours, lacked a cluster that was present in triple-negative breast cancers. And to show this more clearly, we see on the bottom here that there's a specialised type of cycling T cell present in these triple-negative tumours that is not seen in ER-positive, and it provides, I think, some explanation as to why ER-positive tumours are not responsive to immunotherapy. My final slide, this is very new and exciting work involving spatial imaging of the entire tumour ecosystem. And it's just to highlight that you can actually map the gene signatures of cells directly onto tumour specimens from patients. You can zoom in to their precise spatial location. You can define niches, so these are unique domains. For example, we have a red versus an orange niche on the right-hand side. They are both tumour niches. What is different about them? What are the cells that are interacting here? And does this impact on treatment response? So this is a new and exciting era made possible by high-resolution spatial imaging. So just to conclude, I would like to thank my many collaborators and colleagues. Once again, it's a photo of the recent lab. It would be remiss of me not to thank our many funders also, and all of these people over the last 2 decades have made this work possible, so thank you.
Thank you, Jane. What a wonderful talk. So we have time now for some questions. Maybe I could just start off and take the prerogative of the chair and just ask you, are there differences in epigenetic signatures between the various progenitor cells, and do they play a role in cancer formation?
Yes, that's a good question. There are profound differences in the epigenomes of the stem cells and progenitor cells and also the cancer cells, but we don't know how they're regulating gene expression.
And men get breast cancer, although rarely. Do they get the same subtypes as women, or the same mechanisms you think are going on there?
Yes, about 1% or slightly less than 1% of males have breast cancer, and it's usually ER positive, so it's more, it's usually responsible to endocrine therapy. Having said that, men that carry a BRCA2 mutation develop more aggressive luminal tumours, and these are not good news. So they're ER positive, but they're a more aggressive type of ER positive.
Thank you. Yeah, sorry. Go ahead.
Thank you for that wonderful presentation, and I apologise if this is a naive question. I'm a sociologist, but I want to maybe get to the treatment. Are the implications that you can respond to this by, like, getting to the nub before it takes place, or it allows for more, I suppose, boutique treatments because you know which cell is mutating? Again, I'm sorry if it's a very naive question.
No, it's a very relevant question. So if I just go back to the BRCA1 carriers, their only options are a double mastectomy and removal of both ovaries and fallopian tubes. So that is very radical and invasive. So the idea behind this breast cancer prevention trial is to give them denosumab, so it is actually already in existence for osteoporosis and bone mets, so it has a fairly safe profile. The idea is to give them 5, in 2 groups, randomised groups. Some women will receive 2 doses per year for 5 years, and then their tissue will be assessed. Some of those women will opt to go to delay their surgery. And others will probably take their chances. So we don't know. It is going to be 5 to 10 years before we know whether this strategy is going to be effective enough to prevent tumour development. But in the very least, we hope that it can delay decisions for these women because they're very invasive.
In that regard, Jane, I thought that if you had carried the BRCA1 mutation, you only had a 60% chance of getting breast cancer, so 40% will not get cancers. Yeah?
No, yes, it's 70%.
70%.
70%, yes. So it's very high.
Yes, we got a question there.
Hi, Jane. Thanks for a great talk, and I think also for everything you've done for our field over so many years. I'm really fascinated in this point you made about how repeated exposure to female sex steroid hormones expands, for example, progesterone expands the luminal progenitor pool, increasing the risk of breast cancer over time. And I'm wondering if you think it's truly just a numbers thing, or whether these repeated cycles of hormonal exposure change the stem cells in some way that then predisposes to tumour formation.
I think it's both. So firstly, we need a long-lived cell to accumulate mutations because breast cancer is still largely a disease of ageing. So stem cells really fit that criteria. And I think if we've got something I was alluding to, now that we may be able to pinpoint the cells at a molecular level with these single cell studies, we may be able to do this kind of tracking with humans because we clearly can't do the same as we have done for the mouse models. So stem cells, I think, will be a large part of the answer, but then luminal progenitors clearly are highly proliferative, and they will often manifest the errors that have been sort of engendered by the stem cell. So I think it's going to be both.
Thank you.
Yes. Can we get a mic down here? Thanks.
Thank you, Jane. The involvement of mTORC1 in the BRCA2 story, obviously mTORC1 is a key ageing canonical pathway. It's driven by overnutrition. It's linked to, obviously, inhibition by rapamycin. Is there any of that that's in any way useful in the BRCA2 story?
Yes, we found in animal models that those that carried a BRCA2 mutation were much more sensitive to mTORC1 inhibition than a wild type genotype. And it also prevented, delayed the formation of breast mammary tumours. So, of course, that just provides proof of principle because everolimus is associated with toxicities, and I think there is a need to develop more selective inhibitors that could be used in a prevention setting because, of course, nobody wants to make a well woman unwell on a prevention strategy. So there's a lot more to do there, but I think we've got a very solid proof of principle because these cells were completely aberrant at both the cellular and molecular level, which is something we did not actually see with the BRCA1. Their signature looked relatively normal, even though the cells were expanded.
I think we've got time for one more question. Well, is there one back there?
Thanks, Jane. If we do have time for one more, I'm very interested what you showed with the macrophage and that it's probably a reparative process, eating the debris, etc. And to what extent do these tumours create a niche that tricks the body into it, trying to constantly repair it, but it can never overcome that repair? Do you think that's part of the process that's going on? And typically that can be quite anti-inflammatory in some settings, and is the difference between those with the T cells coming in to attack the cells versus the ones that aren't?
Yes, that's a very interesting question. So you mentioned those ductal macrophages. They almost certainly are the bad tumour-associated macrophages because we profiled a lot of models, and the tumour-associated macrophages that expand in the later stages of tumour progression have a signature that is so close to this normal ductal macrophage. So I think that it's going both ways. The tumour cells are probably reprogramming the resident macrophages, but reprogramming them in an aberrant sense so that they then turn into pro-tumorigenic macrophages. Yeah. But they're bad. I think they're not going to be good.
So look, thank you very much, Jane, and I think we owe you a debt of gratitude for your wonderful work, and I think it's a model of how great science allows us to really progress in a major way to address such an important problem. Thank you very much. And thank you all for your great questions. So now we'll move to hearing from some of our career awardees. And to officially present this medal, I invite Jagadish to the stage. So the first of our medals is the Susan Cory Medal, which honours the contributions made to science by molecular biologists and Academy Fellow and past President Professor Susan Cory. It recognises outstanding research in all of the biological sciences, being awarded in alternate years in the biomedical sciences and in all of the biological sciences, excluding biomedical sciences. I hope I haven't confused you there. And this year it'll be awarded to Academy Fellow, Professor Stephen Chown. Professor Stephen Chown's research concerns biodiversity, variation through space and time, and the conservation requirements for mitigating the impacts of environmental change. He co-developed the field of macrophysiology, the investigation of large-scale patterns in and processes underlying physiological variation and their ecological implications. He's worked in Australia, Africa, the Asia-Pacific, the UK, and in the Antarctic, where he has over 30 years of field experience. For many years, Professor Chown represented the International Scientific Committee on Antarctic Research, or SCAR, maybe a bit of an unfortunate acronym, of which he is also president. He was president from 2016 to 21 at the Antarctic Treaty Consultative Meetings, providing scientific advice on a broad range of environmental and science policy matters. He has been a national delegate to SCAR for both Australia and South Africa. He provides a range of advice to international Antarctic programs through advisory committees and boards. Congratulations, Professor Stephen Chown.
Congratulations, Stephen.
Thank you, Bob. Jagadish, it's indeed truly an honour to be awarded the Susan Cory Medal, and in particular because of Susan Cory's incredible contributions. The work I do is about species and populations. The species and populations that aren't part of our everyday lives sometimes, but frequently are. When I travelled to Canberra, many people said, "Oh, it'll be amazing. You'll come to the Academy, you'll see the Shine Dome, you'll visit the extraordinary Ian Potter House, and have a great many other wonderful experiences." And instead of only having those, I also saw superb parrot in the trees yesterday, noisy friarbird in another set of trees, several roach species, I won't tell you where they were in case the hotel gets a bad reputation. And that's essentially part of the work that I do. It's about understanding the variation of life that we see and its importance to the way in which ecosystems provide services to us, and you'll all have experienced those: clean water, fresh air, the drawdown of carbon dioxide in the Southern Ocean that prevents our planet from being even hotter than it currently is. The questions that have engaged me for some time are about the fact that diversity is not distributed uniformly across the planet. And this is something we genuinely recognize routinely in our daily lives now. If you go to the Arctic, you'll see saxifrage and you'll see reindeer. If you move further south and you're lucky enough to go to the northeast of Poland, you will see European bison in their original forests. And of course, you can see koala and kums here. And if you travel further south, you will see no terrestrial mammals whatsoever on Antarctica. The question of why there may be more species in the tropics, or why we may have this difference of identity of species across the planet, is quite an old one. And unfortunately, Alexander von Humboldt was somewhat eclipsed by Darwin's findings. He was a tremendous author of studies about the distribution of life across the planet. Darwin came along and much of the focus turned to evolution, and for some reason, the Anglophone literature temporarily forgot von Humboldt's work. But it is extraordinary and was brought to life recently by his biographer, Andrea Wolff. It's truly worth a read. Look up her book on von Humboldt. And he pointed out that life is essentially richer in the tropics. It's poorer at high altitude. There are different forms in different places. And while that was an early significant question, the significance of that question has continued. In soils, there are small animals known as springtails. They're about 3 millimeters to a centimeter if you're lucky. And it turns out that their highest abundances are in fact in the tundra and taiga. And if you look at metabolism across the Earth's soils by springtails, you will find it's invariant because of this difference in abundance from north through the equator to the south. So even though this is a long-standing question, we keep revealing new aspects to it, while at the same time strangely compromising our capability to investigate it by destroying the habitats of the organisms that make up our planet. Studying the diversity of life also has implications for how we look after it. We tend to see Antarctica as a wilderness, as not really very much visited. After all, people only started visiting the continent as we know it about 200 years ago. But if we look across its unvisited places, they don't do a really good job of capturing biodiversity. It turns out that we visit places that are ice-free, and that's where most of life is. So understanding that diversity gives us an indication of how well we're doing in looking after it despite our presence there, which has grown over the last 200 years, both in scientific footprint. There are more stations now than there were in the International Geophysical Year of 1957-1958, and there are more tourists now than there were from the early days of the 1990s, which started at less than 1,000, and we're now sitting at about 120,000 visitors per annum. Of course, the distribution of life is not only about natural systems. We share all of our habitats with other species. And of course, those of you in the medical field will know that they're essential for us. They do also cause us some trouble. So understanding what the causes of diversity's distribution is also helpful for understanding how we may look after those that are much less fortunate than ourselves in some communities. So I would draw attention to, we have done some work on mosquito vectors in informal settlements funded by the Wellcome Trust and others, trying to figure out, if you look after someone's water provision, what do you do to vector-borne disease at the same time if you're using green solutions? The diversity and distribution of life has enormous implications for ourselves. I work on two groups of organisms. Springtails, this is a species from the Dandenong Ranges, which explains why I live close to them. You can see them. They're really quite big. They're a centimetre long. And of course, the other side of the world, I spend a lot of time thinking about Antarctic organisms. And what's really been amazing over the past decade or two is that computational approaches have changed how we view biodiversity. We can now aggregate data in ways we never did before. If you look at the image labeled alpha diversity, this is a typical image of birds showing that there are more bird species in the tropics than elsewhere. If you look at uniqueness, though, there's a monotonic increase to the southern hemisphere. And the reason for that is that in the northern hemisphere, birds have really big ranges. They can go all the way across Eurasia. And in the southern hemisphere, we're completely broken up by the Southern Ocean. And computational approaches that enable one to study this on a fine scale are starting to reveal that our previously held notions may need some adjustment. Macrophysiology is about putting together single lab studies or doing enormous phenotyping studies in ways that weren't previously possible. And doing so, we can understand that soil organisms that are introduced by humans accidentally are actually tougher than those that live here when you expose them to high temperature, providing significant insight into what we may be doing by combining non-indigenous species and climate change at the same time. The insights you get from combining single species studies or doing phenotyping also allow you to go to the heart of the fundamentals of biology. For those of you who have spent any time wondering about metabolism, you will likely have heard of Kleiber's equation of the bigger you get, the higher your metabolism is, with a slope of about 0.75. And in fact, many have said it's just size that matters. Working with colleagues just recently on ants, and Alan, I thought I'd put this in, hoping you would be in the audience. Turns out who you're related to is really important. It also turns out that what you do in your environment is just as important. So from the foundations all the way through to the applications. I've worked a great deal in the Antarctic, and the physical scientists working there make it clear that we're in real trouble. There are abrupt changes to the cryosphere. The West Antarctic ice sheet is, in the opinion of many, basically living out its last days. Sea ice has declined enormously in the Antarctic when previously it was stable. The implications of life require us to understand how diversity is distributed and what its physiological responses are, so that we can understand what the implications of those impacts might be. And to give you a final idea, if we were to stop having the Southern Ocean absorb as much heat and carbon dioxide as it currently does and turn into a source rather than a sink, our trouble would magnify several-fold. One of the things I've spent a lot of time doing is simply working out how to translate this into understandable policy. And this is a set of banners that went to the Antarctic Treaty Consultative meetings, and in a very difficult meeting in 2022, convinced a consensus-based organization that had two parties at war, Ukraine and the Russian Federation, still to come to consensus that climate change needed their continued and more urgent attention. We never travel alone in science, although we often write alone. I would like to thank those I've been traveling with, the Academy for its recognition, and all of those that have supported my work over the years and have contributed to an extraordinary time of biodiversity study. Thank you.
Thank you, Stephen. So I'll now open this lecture up for questions. Yes, we've got one at the back.
Thank you for an absolutely inspiring presentation. I was so interested in your comment about birds, and I wondered if you could talk a little bit more about the migration of birds, because some of them spend time in the tropics and then migrate. For example, the swallow that people in Britain see moving from South Africa through and back every year. And I guess they're in trouble in some parts of the world. But how do you build these into your models, please, for the migration of many other species too?
Yes, it's worth, in the biodiversity models we look at, we tend to treat the migrants slightly differently because they have their homes in two different places. And understanding what their breeding locality is and what their residential locality is out of the breeding season is really important and considered in the models. With respect to the introduction of your question, are migratory species in trouble? I would say that one of the groups of organisms that's facing most difficulty at the moment are wading migrants. The shorebirds you see, such as whimbrels or plovers, they're in real trouble because we're hardening our shores. So many, many nations are turning what was previously mud flats and maybe a bit icky for some into hard seawalled areas. And that's been devastating for migratory birds and certainly is here too.
Yes, we've got a question here. Francis.
Thank you, Stephen. I'm going to ask about the water. So 70% of the Earth is water, and when you showed biodiversity, maybe it's intensely blue means there's a lot there, or was it just black because you were only talking about the rest of the Earth?
I tend to work in terrestrial systems. I have worked in marine ones. But in marine systems, you have very similar biodiversity gradients where you have very high richness in coral reefs and so on in the tropical areas, and somewhat of a decline to the polar regions. One exception, though, is the Antarctic in some organisms, such as sea spiders, which sort of just look like sea spiders but are not, bryozoans, and others have some of the highest species richness values in the Antarctic, and we also find that they're reef builders that are polychaetes, or worms. So there really is a similar kind of variation across the oceans, but I thought for the purposes of a 10-minute lecture and my favorite organisms which live on land, I'd just stick with them.
So if we don't have any more questions, it just remains for me to thank Stephen and congratulations again. I'd like to now hand back to Ivan to introduce our next awardee.
Thanks, Bob. Our next awardee is our 2025 Hannan medalist. This is a career award that recognises outstanding research in any of the fields of statistical science, pure mathematics, applied mathematics, and computational mathematics. It honours the contribution of time series analysis of the late Professor Edward Hannan, Professor of Statistics at the Research School of Social Sciences at the ANU. And this year, we congratulate Academy Fellow Professor Noel Cressie. Professor Noel Cressie is a world leader in statistical science for the analysis of spatial and space-time data, especially in environmental science. He has made pioneering, fundamental, paradigm-shifting and highly influential contributions to theory, methodology and applications. Professor Cressie is a leading exponent of statistical methods in environmental science, especially for large-scale phenomena such as oceanic and atmospheric circulation and climate. By combining physical principles with stochastic models to capture uncertainties, he has developed a powerful methodology to investigate causal links in these complex global processes. He has overcome huge challenges to make the methodology computational feasible for large complex data sets. Professor Cressie has played a key role in applications to global CO2 flux, regional climate, sea surface temperature, air pollution, disease mapping, biogeochemical cycles, soil carbon dynamics, movement of glaciers, and river pollution. So you can see he has very broad interests. His recent work on climate model uncertainty may ultimately have a substantial impact on science and policy. Congratulations, Professor Noel Cressie.
Thank you, Ivan. Congratulations. Well, thank you to the Academy. What an amazing honour to be named the Hannan medalist. I'm really privileged. I look upon it as my first Academy Award, and I want to call it the TED. So I'm very pleased to get a TED. I would like to go through my journey just a little bit. I started out at the University of Western Australia, and Don McNeill actually was there for a short time and suggested I apply to Princeton. On my way to Princeton, I stopped at the ANU for 6 months, and there I got to know Ted. I was at Princeton for a while. I was a Fulbright scholar, actually, and there I had advisors of Jeff Watson, John Tukey, and had some postdocs. Came back to Australia for a few years to Flinders University, and then was offered jobs in the Midwest of the United States, and was probably thinking of staying there for about 5 years, and 30 years later, came back to Wollongong with the urging of David Steele, and have been director of the Centre for Environmental Informatics since then. All right, so as Monty Python says, now for something completely different. I thought we'd go to a different planet, that we go to Mars, and this represents some work that I've done in the last year and a half with NASA and the European Space Agency, ESA. My story starts 4.5 billion years ago when the solar system was very young and there was a lot of exoplanets and asteroids smashing into Mars and Earth for that matter. And with a small stop about 1 billion years later in the Jezero crater, we go to the present. So NASA launched a mission to Mars, and it was the Perseverance rover, and it landed on Mars on the 18th of February 2021. The Jezero crater was active about that 1 billion years later, about 3.6 billion years ago, and there was water and river valleys. And the thought was that the rover would land in the Jezero crater where there was a possibility of life because of all that water. And there is still water underneath the surface of Mars. The goal, one of the major goals, was to search for ancient or possibly extant life. And currently the rover is drilling into the rock. It's also collecting rubble. And in these tubes, these sample tubes, which are being stored in the rover, it's also collecting atmosphere. So there's an excellent sort of choice, if you like, of where that life might exist or might have existed. This is a very ambitious project. It's called Mars Sample Return. And the idea is to send a rocket from Earth to Mars, and in the rocket there would be another rocket which would collect the sample tubes, take off from Earth and end up sending the samples back in a package that would land in the Nevada desert in about 15 years. As you can imagine, it's very ambitious and actually quite expensive. But we have that mission, and I was honoured to be asked to be part of the Sample Safety Assessment Protocol. This is a planetary protection group that is an attempt to keep the Earth safe, or Earth safe, from possible potential biohazards. And my presence on there was as a discussion leader in statistical science, and there I am in the group. One of the key deliverables of this group was to put together a protocol to keep Earth safe, and we developed what was called a Tiger team. Now, NASA develops Tiger teams to get onto problems really quickly and to get out of them again. So the Tiger team disbanded late last year, and we're very pleased to learn that we actually won a NASA award, the Group Award for Research at NASA in June of this year. It was really a wonderful experience to be part of a very interdisciplinary team, astrobiologists, geologists, virologists, and one statistical scientist. And in there we have about, in each of the tubes, and there's 30-odd tubes, 10 to 27 grams of material. Now, those grams of material actually have to be used for something, for science. However, it also has to be used to keep the planet safe, and you can see there's a tension between testing, often destructive testing, to keep the planet safe and leaving enough of that material behind for science. And that's where I got involved. So this whole idea that we developed is centred around a 3-step protocol. First of all, you get heterogeneity, you work out where you might sample in the tubes to take the subsamples. Then there's the notion of an abiotic baseline, and if you exceed the abiotic baseline, then by the way, all of this is in a sample receiving facility, which is built specifically in the Nevada desert to do this sort of planetary protection work. If the molecules look biologically oriented, then you would then do further testing to see if they're hazardous or not. The challenge would be to determine the number of subsamples needed to test this hypothesis, the absence hypothesis, no biohazards, versus the presence hypothesis, biohazards. And the optimization that I was involved with was to use as little of the returned material as possible, but not so little that we would not keep the planet safe. So you can see there's some sort of existential questions going on here. And so what I developed was a criterion which I call the Bayes error, or BE, which is a function of the number of subsamples in each of the tubes. So we did this tube by tube, but we have to know what it means to keep the planet safe. And so NASA ESA specify a risk tolerance threshold, which I've called P star, and typically that's 1 in a million. But I could have developed it, and I have developed it for smaller and higher thresholds. First of all, as a statistician, and I'm sure you know that nothing is certain, and Oliver Cromwell tried to relay that to the Scots before he defeated them at the Battle of Dunbar, and also the famous Benjamin Franklin quote that there's nothing certain except death and taxes. So what happens is we do that heterogeneity. We look at the types of rocks that are in the samples. We look at the atmosphere, the regolith, and various rubble and things. We have these two models, the absence model and the presence model, and we worry about false positive errors and false negative errors because nothing is as certain. So the false negative error is the one I think that should get more attention, and I certainly concentrated on that in the work that I did. It says that we've found no biohazard, but in fact there is a biohazard present. The false positive error would be, if you like, less consequential, that you've declared a biohazard to be present, but it's not present, and perhaps further testing would resolve that. But if, in fact, you've declared no biohazard and there is a biohazard present, perhaps you've then released these biohazards onto our planet. And as I mentioned, there's this tension between using enough so that we keep everything below the safety sample threshold, but leaving enough for science at the same time. So as a Bayesian, I thought we need to introduce whether these models are more realistic or not, the absence model versus the presence model. And so I used pi naught to represent the probability that there is no biohazard present, and pi 1, the probability that there is a biohazard present, and pi 1 is equal to 1 minus pi naught. We also have measurements. These are measurements that haven't been taken yet, and this is partly the problem of this. This is a design problem. We're pre-measurement. We have to design, it sounds impossible, but it's possible. We have to design something where we have no measurements. And so the distribution of these measurements, which we know are uncertain as well, there's uncertainty in those, are called likelihoods. And so under each model, M0, we have a likelihood F0, and under the model M1, the presence model, we have a likelihood F1. These are the components, if you like, as I'm building up this notion that there is uncertainty everywhere, and I'm trying to then characterize it as a statistical scientist. I mentioned that this is a design problem, and what I developed was a Bayesian design criterion, which I called the Bayes error. And it is, in a sense, the expected error, the false positive error and the false negative error weighted by the priors pi naught and pi 1. This is the sort of thing that you get as a cartoon, obviously, but it's showing the Bayes error on the vertical axis. It shows P star, the risk tolerance threshold that NASA tells us, 1 in a million, for example. And then as I take more and more samples, that error decreases, as you would expect. So the Bayes error keeps going down. I can keep driving it down as far as I can, more and more samples until I run out of samples. But clearly what I want to do is drive it until it's just below that threshold, that I can then say 1 in a million, we've achieved that. And I use that number, in this case, it's n star equal to 5. So what happens is that this optimal n star actually depends on a number of key quantities, and it just takes building the model to work out what those key quantities are. Pi naught I've already mentioned, it's the prior. Something called kappa, which I've only indirectly referred to, and that is the sort of loss you would make if you had a false negative error versus the loss you would make if you had a false positive error. And the ratio of those two, and we keep kappa small in this case, is really important, and it's an important dial, if you like, which determines how large n should be. The last one is the distance, so-called distance or divergence, between those two likelihoods under the two different models. And how would you get a hold of this divergence? Well, there are actually experiments around that are attempting to come up with Mars-like materials that would be biotic and abiotic, and there is such a paper which we use to then build the model in a principal component space. This is what it looks like after you do the sort of fitting that statisticians do, and that's a normal distribution in 2 dimensions, the principal component dimensions, with both the abiotic and biotic on the same graph. So the important part of this particular slide is the second dot point, which is how do you calculate this Bayes error? Well, it's possible to do it analytically, but remember these 3 stages of the protocol that we developed. Each one leads to the other. And so there's actually a sequence of tests of models. And it became clear to me that the way to do this was to pretend to be in the sample receiving facility and construct a digital twin of what was going on, and then using all of this information to come up with the appropriate dials, the kappas, the deltas, and the pi naughts. Okay, the chairman who managed to herd a whole bunch of cats, including a statistical scientist, called this the money plot. And it's very American, you know, to think about the money plot. That's where the money is. That's what's going to get NASA really excited, and it did. This is the money plot where I get to plot pi naught and delta, and I hold kappa fixed, because I've only got 2 dimensions on a money plot. And I have my P star, which are the red dotted lines, 1 in a million. I then work out under these different circumstances, different pi's and different deltas, what n star would be. And it can be 1 if your deltas are very far apart. And as the deltas get closer together, the number that you need gets bigger. And I gave him some numbers which had, I don't know, 15 or 20, and they were a bit unrealistic. But on this particular money plot, the largest n star that I came up with per sample tube was 5. And then one works out how much of that sample that you would need. This is a plot to just show you what happens as you artificially move those 2 likelihoods closer together. And as you would imagine, as they get harder to determine which model is correct, the number that you need goes up. So n star keeps going up and up. But basically, we're sitting at around 5. So this is my hitchhiker's guide to Mars. And of course, it's not to the galaxy, it's only a smaller place. It isn't 42, it's 5, but with a plus or minus. So thank you very much. I'll take questions. Thank you.
Thank you, Noel. So you just have to wait till 2040 for you. Australia Post can't help you out, you know. Okay, any questions?
So great talk. What sort of reliance is there on a homogeneous distribution in the samples? Are they going to crush them and take fractions of them and then assess the likelihood of the analysis from that?
Yes, so we have actually a geologist. She was part of the technical specialisation. There's a whole chapter on that in the report, 200-page protocol. And I helped her with that. So basically, these are transparent tubes. You can actually see into them. And through her geological knowledge, we were going to look for, well, in statistics, we call this blocking, which basically means you try to get a lot of variability as to what you're looking at. You try to get your blocks as far apart and you get as much heterogeneity as possible. So you're sampling across the spectrum of what's in the tube. And so that's one quite large chapter in the report. And I would rely on the geologist and the transparency of the tube to do that. So there's a lot of initial testing simply by looking, by visualising the tube, and then the material would be taken out very carefully, I can assure you.
There's a question from David Crowley.
Thank you very much for your talk. And I have done a bunch of Bayesian statistics in environmental aspects of the atmospheric science. But I have a question, because often Bayesian statistics depends upon the assumptions that you've made in developing the models. And I'm surprised that you didn't consider the case, as a rocket scientist might, the chances of the rocket not landing safely, but crashing and distributing the material in the Earth system environment prior to it being tested for whether it's actually got astrobiologically active, dangerous material.
Look, they have thought of that, and in fact, I didn't...
Measure the frequency of that from crashes.
Right. So they're not actually going to land a rocket on the Nevada desert. They're going to send a packet, and this is the Australia Post idea. They're going to send a packet, and they're not even going to parachute the packet down. They're going to smash it onto the Nevada desert, and it's supposed to be totally self-contained. Well, we all know there might be a 0.0000 or something like that happening, but that was not taken into account in my calculations.
Well, one last question at the back there.
Thank you for the wonderful presentation. As a geologist, I'm extremely curious to find out what your specific characteristics are when you're factoring in what's biotech and what's not.
What's what? Sorry?
What's biotech and what's not? What specific characteristics are you looking in your materials?
Yeah, so we fully expect that in 15 years, some of the mass spectrometry that is done currently may actually be superseded by better tests. So what's happening is I've built a model, and the likelihoods that are there would change according to the different tests that you make. And I think that is behind your question, which is what test would you make? And the geologists and astrobiologists have produced a lot of data, and Buckner et al., which I used, there's a long paper published in Astrobiology where she produced, I think there were probably 200 different measurements that were taken, and they were reduced using principal components, which is a statistical method. So in a sense, if you add another 10 tests because of your expertise, it would mean that that principal component analysis would be done on 210 measurements, not on 200.
So we are assuming that what we're going to find is going to be Earth-like?
No. But in order to build the likelihoods, what we do is the Buckner et al. Data was biotic and abiotic. So the abiotic were actually Mars meteorites, but they had gone through the atmosphere and considered to be abiotic because they burned up. The biotic part has come from very extreme environments, which on Earth, right? Because we haven't gone to Mars yet. So they are representative, if you like, of likelihoods, what likelihoods would look like. Now, what happens in 15 years is there would be tests of various forms that I cannot predict because it's in 15 years' time to try to detect that biohazard. And I'm not the expert to sort of tell you what those tests are. And in fact, all our experts had really an idea of what it might look like currently, but would not look like in 15 years. But the structure, the mathematical structure and the probabilistic structure remain strong, because whatever the likelihoods are that are being built would then be used to calculate delta, and there would be a kappa and there would be a pi naught, from which the money plot could be made under those circumstances.
Okay, unfortunately we've run out of time, so please join me in thanking Noel again.
Thank
you. Noel will be available at lunch, so please, continue the conversations. That now concludes this session. Before we move on to our next set of awards, we'll take a break for lunch. Our awardees, donors and special guests are invited to move to the large marquee at Ian Potter House for their lunch. Everyone else is invited to move to the Jaeger room, which is just outside, the Dorothy Hill room, or outside on the lawns. We'll reconvene at 1:15 p.m. for our next award presentation. Thank you.
The average Australian spends at least 90% of their time indoors. But how often do you think about the air you're breathing in? Indoor air pollution is considered a serious risk to public health in societies around the world. Despite that, most countries don't have any indoor air quality standards.
Every drop of water we drink from the tap, its quality is highly regulated. Every piece of food we take to our mouth is highly regulated in quality. Indoor air, which we take into our lungs 12 times a minute, is not regulated at all.
Professor Lydia Moravska from Queensland University of Technology was named one of Time magazine's 100 most influential people in 2021 for pressuring the World Health Organization into acknowledging that COVID-19 could be spread through the air, particularly indoors. Now she's leading a new push for international action. Professor Moravska and a group of global experts have used the prestigious journal Science to publish a blueprint for mandating indoor air quality in public buildings.
Our paper, which is a consensus of over 40 scientists and experts from many disciplines from around the world, is the best practice, better than anything else which is happening anywhere in the world.
The group proposes standards for 3 key indoor pollutants: carbon dioxide, carbon monoxide, and PM2.5, which are particles so small they can lodge deep in the lungs and enter the bloodstream.
This is my favorite little monitor. It measures carbon dioxide. Together with carbon dioxide, we emit other things, in particular pathogens, bacteria or viruses if we are infected. So if concentration of carbon dioxide increases, potentially concentration of these pathogens increases as well. And it fits into my handbag, so it goes with me everywhere.
The scientists also want to ensure mechanical ventilation systems remove and dilute pollutants at a higher rate than their production. She points to schools as an obvious place for action, where many classrooms are now being fitted with air conditioning.
One issue with this is that while it provides a good thermal comfort, it does not provide fresh air. It just recirculates the air, so it doesn't do anything to improve air quality.
Outdoor air quality is already regulated in many places in line with the World Health Organization's global air guidelines. Professor Moravska says countries such as Australia can now lead by example for indoor air as well.
We know how to do it. We know how important it is. Why won't we start now?
I grew up in the rice growing region in China. In the 1970s, when China started to use chemical nitrogen fertilizer, urea, I actually was fascinated when rice plant become green, farmers get a high yield. But in the afternoon, I walk from school walking home, I saw the fish die. I think it contributed me. I want to study agriculture, soil science, chemistry. My name is Deli Chen, Professor of Soil Science at the University of Melbourne. My research very much is agriculture and environment, focused on the nitrogen fertilizer use efficiency and its impact on the environment. The cost of farmers globally more than $200 billion on nitrogen fertilizer, but unfortunately only half is used by plants. The other half is lost to the environment. And then the nitrate leaching to the waterways, contributing to water pollution. That's the main cause for green algae bloom. If you cannot measure, you cannot mitigate. So we develop a method to quantify how this nitrogen is lost from agriculture, from plant soil system. We need to use a very sophisticated instrument. We use a stable isotope N15 to trace the movement of the fertilizer nitrogen. We can know exactly when the fertilizer ended up in plant or in soil or losses. But beyond that, we're now working with organic chemists to develop a next generation inhibitors. We hope in the future we can develop a smarter fertilizer. In 1987, I received the full scholarship to the PhD from Melbourne University. And then, in 2009, I was the first mainland Chinese become full professor at Melbourne University. And now I'm a fellow. Certainly, I never expected that from the young farmer boy growing a rice paddy, and to receive this amazing recognition. If we work together with Australia and China, I think it's not only good for Australia and the China economy, in my area, the food security environment, but I think it's a benefit globally.
You're looking at the largest single dish radio telescope in the world. It's called FAST and located within the mountains of southwestern China.
And that is by far the most sensitive radio telescope in the world. I first went there while they were putting the first few things on the ground, and it was amazing to see how quickly in China you'd come back a year later and the whole dish would be built.
At the heart of FAST is a primary multi-beam receiver designed and built by Australian scientists. China chose to collaborate with Australia because of our experience with the Parkes radio telescope in regional New South Wales.
The Parkes radio telescope is by far one of the greatest telescopes in the world. It's been upgraded many, many, many times, so it keeps getting upgraded. It's not a 60-year-old telescope. The most recent upgrade was just a couple of years ago with a very wideband receiver system that allows us to observe the radio waves over a much wider bandwidth than any other telescope on the Earth.
Parkes has special significance to Dr. Shi Dai. His first visit was as an international student.
I mean, the second you see the 64-meter big telescope there with wonderful weather, blue sky, I was really shocked. I saw this is the way to do astronomy.
He later returned to Australia to complete his PhD under the supervision of George Hobbs, before accepting a teaching position at Western Sydney University, where his research focuses on rapidly spinning neutron stars known as pulsars. The Parkes radio telescope has detected more pulsars than any other facility in the world.
Radio astronomy in Australia is really strong. The experience and expertise here are really the most attractive for a student in China.
That's thanks in part to the work of people such as Professor Elaine Sadler. For more than 30 years, Professor Sadler has been at the forefront of research into astrophysics. Her team discovered a 5 billion-year-old galaxy using the ASCAP radio telescope in Western Australia.
Now, 5 billion years ago, the Earth didn't exist. So in the time that signal's been travelling to us from this galaxy, the sun has formed, it's made planets, The Earth has settled down, it's got oceans, life has evolved, we've got a technology, we've built telescopes, and there we are just in time to catch this signal.
Professor Sadler started working regularly with Chinese scientists 7 years ago. She says international collaboration is critical to understanding our universe.
I think one of the great things about working collaboratively with people is it allows you to build up the trust and the common interests that allow you to make advances in science.
Astronomy is a very collaborative science. You can't just do your own research simply on your own.
It's about actually exchanging your idea with different people, different background, different expertise. If we don't have that, we are not doing the best science.
I love finding out new things or discovering new things. We actually start with what is the problem. So you have to learn the problem. My name is Rick Middleton and I'm an emeritus professor at the University of Newcastle. I'm a researcher in the area of systems and control. This is about the many technological and natural systems where we have things where there's an action of sensing, there's a decision process or a thinking process, and there's an action process. This control was part of what got people to the moon in the late 60s.
We have a liftoff.
And my work has been looking at the theory of how these things work, what can go wrong, how to make the best use of them, primarily from a mathematical viewpoint. That could include things like satellite tracking systems. I've worked more recently on telecommunications, some systems biology work. For example, what's the best way to treat a pathogen like HIV? If you have a few different treatment options, how should you best use those? So we build differential equations where the time derivative of some things depends on past values of those variables. And so we use that as a way of trying to model what we would call the system, the thing we're trying to affect is going to do and how it's going to behave. Another example is telecommunications. Ericsson produces base stations, and they said, we want everyone's phone to be able to work. We don't just want one or two to be able to work. So you've got this really complicated control problem about regulating everything to do with that so that everybody gets a fair share, things operate smoothly. And our work led to several joint patents we had with them. You know, so we've impacted the technology that goes into some of those base stations. I was born in the Newcastle area. It's one of the environments that I think has many of the advantages of a big city, but without being too big. And at Newcastle University, there was this fantastic intellectual environment. Fine to ask questions or to query something. This sort of open-mindedness was something that was really encouraged. Becoming a Fellow of the Academy is a great honour. It also means joining this group of, really esteemed scientists. It's something I guess I also deeply believe in, as well as what I've worked on, which is that, a diversity of views is really important because there is so much in the universe we don't know. There's so much on the Earth we don't know. And I just find it really exciting to understand all of those different things just as much as we can.
ADSTAR is our national showcase and summit that celebrates all things defence, science, technology and innovation. It explores the highest priorities for defence. ADSTAR allows our innovators, our researchers, our technologists in industry and in universities and in government to come together to really build the relationships required to make sure that the knowledge we create in this country is used by our ADF.
Over the last 20 years, there's been a huge increase in fatalities due to opioid addiction, and it's compounds such as fentanyl. The sooner we can find an alternative for opioids, the better. And my name is David Adams. I'm at the University of Wollongong. I'm a physiologist. I study nerve cells, and in particular, those nerve cells are involved in transmitting pain from the site of where it rises to the brain. Cone snails obviously found primarily on the Great Barrier Reef, but it's been known that cone snails could be deadly. People have picked up a cone snail and have been injected with the toxin. These toxins are peptides, small proteins, very small proteins. These peptides had been shown to be very effective in animal pain models. In these models, the distinguishing feature of these peptides is how long-lasting they are. So it's not just for a couple of minutes. It can last for a significant period of time. For me, the challenge has been to get a full understanding of how these peptides produce analgesia. And is this in the absence or presence of peptide? We understand now that when the peptide binds to the GABA B receptor, it inhibits calcium channels and it potentiates potassium channels. And it's that combination that really shuts down nerve transmission of the pain signal. So what we've been trying to do is target where the pain arises, either at the nociceptive nerve ending or where the damaged site is on the nerve cells. The dream is to get this peptide that we've been working on now for at least 20 years to get it into a clinical trial. To me, the potential application is to have an orally available tablet that you can take for treatment of chronic visceral pain, irritable bowel syndrome, endometriosis, for any chronic pain condition. By going to a different type of analgesic, such as these peptides, you avoid those problems that have afflicted, generations through the rapid rise in the increase in opioid addiction. For a scientist, it's most probably one of the highest honours you can get in Australia. As a physiologist and a neuroscientist, to be able to give something back is important to me. I'd love to finish with this peptide going through a clinical trial and getting onto the market. I'd be prepared to take it myself.
You're watching time-lapse footage of dung beetles at work. Their remarkable ability to remove dung makes them the perfect biocontrol agents. Research has shown their presence significantly reduces the number of flies and other pests, while also improving pasture growth and soil retention. So, scientists like Professor Leslie Weston have been searching the world for the best species to bring home. But wait, doesn't Australia already have dung beetles?
We have 500 native dung beetles, but those dung beetles are geared to sourcing marsupial dung or drier, small, hard pelleted dung, whereas when cattle, sheep, horses and other livestock were introduced to Australia, they generate a larger, wet, moist paddy.
CSIRO began importing dung beetles to Australia in the 1960s. The program has been overwhelmingly successful. However, the introduced species haven't adapted to all regions of Australia, and some will simply stop working during certain periods of the year. Researchers are working to address these gaps with a joint venture called the Dung Beetles Ecosystems Engineers Project, supported by Meat and Livestock Australia, with federal funding from the Department of Agriculture, Fisheries and Forestry.
So we'd like to have a full complement of dung beetles that are active all year round, because obviously cattle or sheep are producing dung all year round that needs to be processed.
3 new species have been introduced from the Mediterranean region, but they don't all deal with dung in identical ways.
We have rollers that create these rolled balls and bring them below. We have the tunnelers that bring the dung down and create the balls. And then we have those that remain on the surface and dwell above. The dung beetles actually subsist by sucking out the moisture on the dung. It's like a liquid milkshake. That's their nutritional source.
Researchers at Charles Sturt University in Wagga Wagga are helping to rear the new arrivals after their strict quarantine regime is over. The offspring will then be distributed to field sites across southern Australia.
I tell people that I am a mathematician
And I get two different reactions from that. Sometimes the deer in the headlights, oh, I hated mathematics when I was in high school, and often the, oh yeah, I love mathematics. My name is Jessica Purcell, and I am a mathematician at Monash University. What I do as a mathematician is I work in a branch of geometry, which is something that people wouldn't have seen in high school. One thing that I work on, for example, is an area of mathematics called knot theory. This is studying the mathematics of knots. You take your shoelace, you tie it up, you fuse the ends together, and then the mathematical questions around that would be along the lines of, can you undo the knot without cutting it? Is this knot the same as a different knot? How do you tell two knots apart? You see knots in DNA and in protein folding. Some of the spaces that I study also appear in crystallography. There are connections to quantum physics and string theory. So these are some of the reasons that knot theory is still popular and becoming more popular. Okay, now we were going to try and triangulate this. When I was going through high school, university, there often weren't very many women in mathematics, and that kind of was a little bit painful.
But then we cut along all the 6 planes.
When mathematics it's for all humans, it's for everyone.
So we should be able to collapse the bygons, right?
So I always felt that it was quite important to be able to bring everyone into mathematics. So men and women, young people, old people. I want to involve them. I want them to be able to feel like they can be a part of this as well. When I'm doing mathematical research, it just feels good in the brain to sit and think and to puzzle out difficult, complicated problems. And then it's just extremely satisfying to be able to fit pieces together, to make generalizations, to prove general theorems and statements about geometry, about mathematics. Becoming a fellow of the Australian Academy of Science is a great honor. It's amazing to be part of this group of people, to be able to do some of the good work that they are doing. It's very important to help people to find their strengths in mathematics and to be able to help people to see that there's actually this incredible creative process to discovering mathematics. I think that's really, really important.
I think it's the idea that you can't be what you can't see. So if we don't have women, people of colour, all sorts of diverse representation in science, the next generation is going to look at that and say, well, it's not for me.
My first position as a bioinformatician was in the computer science department, and you could really count the number of females that were in the building, let alone in leadership roles. When you have people who you can relate to, it certainly allows you to at least imagine that that could be possible for you. So I think that representation, that diversity does matter when you're trying to inspire the next generation.
There's been studies that show people with diverse experience bring new ideas, a fresh perspective, and just think of all the scientific breakthroughs that we will miss out on if people don't see this as a career where they are valued and their experiences are valued.
Michael, good to see you after all this time.
Good to see you, Robert.
About one of my favourite people in Australian history, Mark Oliphant. Would you remind us, what sort of scientific research was he famous for?
So when I talk to people who don't know who Mark Oliphant is, I might have casually dropped in conversation, because I don't have the surname, that I'm related to him, and they ask what he did. I say, well, have you got a microwave oven? Because the cavity magnetron, which makes every microwave oven work, was an invention of Mark's team at Birmingham University when he was Professor of Physics there, but more importantly, was invented as a tool for helping the Allies to win the war against the Axis powers, because it's at the centre of microwave radar, which allowed you to take something that used to be housed in a building and put it in the nose of an aircraft.
He came back to Australia, which was very interesting, because he could have stayed there indefinitely, Why did he come back?
He came back, and lots of people have commented that he really put a torpedo through a very promising and well-remunerated research career by coming back to Australia.
The fundamental particles that we use, called protons, are fired by means of this little cyclotron.
But he felt that studying science and doing scientific research at a world-class level was an important capability that Australia needed to develop.
Let me just quote to illustrate his idealism. He said, "I have a deep confidence in the part which science can play in making us strong and prosperous." He's talking about Australia, of course. "And an idea that the proper use of science within its diverse territories may point the way to a secure and good life for all," for the community. The idealism is so strong.
Indeed, the community and the population of the world. And when you think about some of the scientific discoveries he'd been involved with in particle physics, in terms of the team at the Cavendish laboratories at Cambridge that split the atom, his work later on microwave radar at the University of Birmingham, and his work on putting atomic physics into play to, as they felt at the time, win victory in the Second World War. His view of science and learning was fundamentally connected to his view of the well-being of humanity and the advancement of humanity in all fields.
There's one paradox, though. He, very peaceful man, as you implied, and I read that quotation, idealistic. And yet he was the one who recognised the problem of the bomb and that the Germans might get it. And he famously, although not in the movie, recently,
not in the movie,
took the letter to America that made the Manhattan Project happen. You know, was he always happy about having done that, or was he conflicted?
So, yes, you make reference to Oppenheimer, the movie, and if you watch the movie, the story, really starts with Ernest Lawrence at Berkeley University in California. And it is true, my grandfather was the one who saw Lawrence as the one who would have the vision but also the political nous to sell the idea of developing the atomic bomb to the Americans, which was the only Allied power that had the finances and scientific capability to carry it off. It was quite clear to everyone. To answer your question, yes, he was conflicted about it, but what he would always say was, what you need to understand is we were in a real war of survival against an ideology manifest in the Nazi regime and Adolf Hitler in Germany.
Of course, we're here to celebrate the formation of the Academy of Science, and he was playing such a big part in that move, which they'd tried to establish in 1901, a long time ago. What did Mark manage to do that no one else could seem to bring off?
I think there are several parts to that, but perhaps the most important one, the Royal Society played a fundamental role in connecting people and bringing the importance of scientific research to light and to the public consciousness.
So he saw an example that he wanted to follow.
He saw an example that he wanted to follow, and he didn't think the answer was to nominate Australians to be Fellows of the Royal Society in London. He wanted an Australian institution which would herald our excellence and which would provide a home, as this place does, for all of those high-level, world-level discussions and debates. That's why he felt it was important.
So when the Academy was set up, of course, the Shine Dome was being built, this extraordinary structure. What did Mark say about the design?
That was absolutely something that Mark was proud of. As most people know, it was quite a controversial design in its time, notwithstanding that Canberra was still developing architecturally, and many of the national institutions we know in the parliamentary triangle today weren't there when the Shine Dome was originally built. But he was tremendously proud of it, and he saw, I think, the controversy associated with its design as a bit of a wake-up call, which helped him with his narrative about why things were important.
Yes, Australia's got its own flying saucer.
Australia's got its own flying saucer.
Middle of town.
And we've dared to do something different. We've dared to be experimental. And we're happy to be proud of it.
And the Academy has this wonderful library, but he was strongly involved. In what way?
He was very insistent that part of the Academy's offering and part of the Academy's importance as an institution would rely on the information that it held, including about the development of the Academy itself as it went through the decades. So he personally insisted that the design of the building and the budget for running the Academy included strong allowances for a proper library.
They conducted most of their business by letter, so that's what this stack of paper over here is. So this is Mark Oliphant's Academy-related correspondence for 1954.
I'd be keen to dig into that correspondence a bit.
Yes. What do you think he would make of the work of the Academy were he able to comment today?
He'd certainly have views. I think he would hope that the Academy would continue to develop its ability to broker important discussions and provide a platform for a reasoned debate on where the country should go and where the globe should go on some difficult and controversial issues.
Thank you.
Thanks, Robin. Good to talk.
Most experiments don't work. They don't work the first time. Often, they don't work the second, third, fourth, fifth times. But then the eureka moments, occasionally, it's really awe-inspiring. My name is Professor Jeff Errington. I'm an Australian Laureate Fellow at the University of Sydney. My main interest for virtually the whole of my career has been trying to understand how cells grow and divide. We work on a bacterium called Bacillus subtilis. Like many bacteria, it can divide and replicate itself in about 20 minutes. It has 4 million base pairs of double-stranded DNA, and that means that it has to be able to, every 20 minutes, completely replicate those 4 million bases absolutely perfectly without any changes in the DNA structure. Working with bacterial cells enables us to make very rapid progress, and get deep understanding of very important biological problems. When I look back at my career, I'm really proud of the fact that we were really among the pioneers of understanding how those very fundamental processes worked in bacterial cells. The discovery of antibiotics for decades was done by just taking random chemicals and look to see if they kill the bacterium. And it turned out that some of the functions we were interested in, these fundamental processes like cell division, were potential new targets for antibiotics. And I've actually had 2 spin-out companies now, both of which have been looking really for novel antibiotics.
Number 3 looks really good.
Wow.
The other line of research is on some very strange bacteria that are called L-forms.
So it's like they're not regenerating their cell wall, and you can see there's like a ghost.
Yeah, these are really strange. So most bacteria have this thick cell wall on the outside, so that when penicillin acts on that and stops them from making the cell wall, the cells are killed. But it turns out that for most bacteria, if you get the conditions right, you can actually grow them without a cell wall, and they call these things L-forms. And that's turned out to be very interesting. It's got us into all kinds of other interesting areas of science, not least the origins of life. As soon as we published our first paper on this, we started to get emails from people who were working on the origins of life, because they're trying to think about how very primitive cells would have divided. I was at Oxford for 25 years, and in the second half of that period, I was actually at a wonderful place called the Sir William Dunn School of Pathology, and it has incredibly strong connections with Australia. It's also the place where penicillin was purified by Florian chain, and so being elected to the Australian Academy of Science means a great deal to me. And although I've only been in Australia for 3 years. I see this as a wonderful opportunity to engage with other Australian scientists, potentially to make a difference in bringing my expertise and experience from the UK to Australia.
We're trying to focus in on what happened in our very earliest solar system. And so that's important because we look out and see all these exoplanets now. How did they form? Well, how do we answer that question unless we understand how our own solar system forms? My name is Trevor Ireland and I'm a geochemist at the University of Queensland. There's nothing like having a rock to actually take into the lab and be able to analyse in great detail and tell you about what's actually going on in terms of processes. Having that sample in hand is paramount to understanding what's going on. A lot of my analyses are actually very serendipitous as to somebody's found something interesting in one of these rocks and wants to understand it a bit better. So this is a sensitive high-resolution ion microprobe. It uses a focused ion beam to ablate a very small area of a sample which is held in the source chamber here, and the ions are then extracted out through the mass spectrometer and passed through the end of the collector. So we can look at the chemistry of the rock, we can understand the elemental distribution, but we can also understand the isotope distribution between different mineral phases, what the composition of, say, oxygen is in the early solar system, which is quite different to what we have on Earth. And so all of these give us a signal as to what processes have actually occurred. This is basically a piece of the moon that was found in northwest Africa after something hit the moon and material splashed down onto Earth. Quite often, it's not so much the people walking into the office saying, I've found a meteorite, which generally is a meteor wrong. So we have all these issues about, where we get samples from, and mostly it's from museum curators. So you've got to be on friendly terms with museum curators to give you samples that they've got. Getting an understanding of how all that material came to be is an important component of how you deal with looking at basically just spectra of faraway, distant exoplanets. Being elected as a Fellow of the Academy of Science is truly a privilege and an honour. It's almost for a culmination of any scientist's career to be acknowledged by your peers as being worthy of recognition. So I'm just absolutely amazed that it's come about.
I work on elastin. I work on the stretchy stuff that's found in human tissue, but it breaks down. Human tissue gets damaged and disease. Human tissue happens to undergo a whole range of changes simply as a result of aging. And so it's important for us to find ways to keep those working. In the very early days, people said it just could not be done. My name is Professor Tony Weiss. I'm the McCarthy Chair in Biochemistry and also the Professor of Biochemistry and Molecular Biotechnology at the University of Sydney. So regardless of whether you go, for example, to the skin that bends and flexes, that needs elastin. If you happen to go across to blood vessels that expand and contract with every heartbeat, that needs elastin as well over countless heartbeats in a lifetime. Even as we breathe in and out, we need elasticity in the lung to make sure that works well too. And yet at the same time, it's really impossible to be able to repair that elastic tissue because the key building block called tropoelastin drops down within the first few years of life. And my research is involved in repairing and replacing those tissues. Take these two different solutions and modify it slightly so that it can be used to treat really compromised wounds.
Yes, this can be used for different type of healing, diabetes wound healing, burn healing as well.
So what better way to do things than to use little bacteria to make precise replicas, precise copies of the natural building blocks. We've been able to build those building blocks in the lab and use those building blocks to be able to assemble large amounts of human tissue, fibres and sheets and tubes.
So these were freshly printed out.
So now we can make very even, very carefully made tubes here, which can be basically used as blood vessel replacements. One of my dreams is to be able to talk to surgeons who can one day go to a shelf somewhere in the operating theatre and say, aha, you are a size 4B, and they go 1, 2, 3, 4, here's 4B, pluck it off the shelf, and there is a pre-assembled tube made with our material, and that's, by the way, exactly what we're doing now in the lab. So one of the things I like about this is that we're using a protein-based material. When we had this building block from nature, we had to learn the rules of assembly. And learning those rules of assembly has not only given us an insight in terms of how to repair human tissue, it's also taught us a lot about the exquisite beauty of nature. As far back as I can remember, I've always wanted to be a scientist. For example, when I was a little kid, I was asked to draw a picture of what I wanted to be one day. And I knew then I wanted to draw a scientist. And so to be a fellow is really one of the highlights of my life. I am so thrilled and very, very humbled at being a fellow of the Australian Academy because it's able to decide and direct where science might go. It's a structure of influence. I pinch myself quite often, and I think it's quite an extraordinary journey we've been down. But I don't think the journey has stopped, of course. There are so many things we still need to do and I want to do.
The impacts of climate change are being felt around the world, from extreme weather events to biodiversity loss. We've been put on notice to take urgent action. Countries, including Australia, have committed to limiting global warming to well below 2 degrees above pre-industrial temperatures. But right now, we're on track for 2.1 to 3.5 degrees of warming by 2100. That's beyond what scientists consider manageable and could have potentially catastrophic impacts for the environment, people, and the economy.
So what we have is a world which is increasingly stressed by human activity.
The solution is to address the tens of billions of tons of greenhouse gases being pumped into the atmosphere every year. That requires not just dramatically reducing future emissions, but removing greenhouse gases already released.
This means drawing down or removing these historical emissions from the atmosphere for, if you like, geological time.
Natural systems such as our oceans and forests are highly effective carbon sinks, but they need help, especially given the further pressures from events such as bushfires and ocean warming. We don't yet have the right toolkit to effectively remove greenhouse gases at the scale required. So, the Australian Academy of Science brought together the country's leading experts to identify opportunities to expand research efforts and start the conversation in Australia. Beyond strengthening existing carbon sinks, new technologies will need to be developed.
I think the scale of the problem that we're facing means that we can't discount anything, and in fact, everything needs to be on the table.
Including things such as direct air capture, a technology that mimics trees.
We can develop these nanoporous materials that are really highly selective for capturing carbon dioxide from the atmosphere and other greenhouse gases. So in the same way that a tree uses photosynthesis, it takes in carbon dioxide and processes that carbon dioxide and spits out oxygen and energy at the other end, we've got a way to do that, if you like, using technology.
The first air capture units in Australia designed by Southern Green Gas are now in the prototype stage. But there are many other emerging technologies. This Hawaii-based startup, for example, uses solar power to make seawater less acidic, enabling the ocean to absorb more CO2. Scientists are now working to better understand the implications of the various emerging technologies.
There's a big gap between doing things at a lab base and doing them at a real-world scale significant enough to actually make a difference to climate change. That jump brings a whole series of uncertainties, risks and costs.
Once we have a better understanding of those consequences, the technologies could well be used alongside other strategies to help fight climate change.
I don't think there's any such thing as a silver bullet. So this is going to take a mammoth effort across the world, including a portfolio of approaches in order to get us to this point where we can indeed stabilise our climate and avoid a climate catastrophe.
Science knows no borders. Right now, Australia is opening its doors to the brightest minds to shape discoveries that matter to all of us.
We didn't know much about the research environment in Australia. As it turned out, Monash had very little in my area of research in quantum condensed matter physics, but the facilities in Australia were absolutely excellent. Better lab space than I had at University of Maryland, really world class. It was an opportunity to start something new.
We're partnering with government, business, philanthropy and the research sector to create pathways where extraordinary talent can thrive.
On my first trip to Australia, we went out to the Parkes telescope. You're in control of this most powerful radio telescope in the Southern Hemisphere, and you're pointing it at the one thing that you want to look at. You feel the telescope moving. It's a full body experience.
Australia's an exciting place to do science.
Australia, where science lives.
The vision was, why can't we do cognitive behaviour therapy through the internet so that we can reach young people who are at risk of depression or have depression who are simply not getting any care. My name is Helen Christensen and I'm a Scientia professor at the University of New South Wales, Sydney.
I'd like to draw your attention to the client referrals by mums.
I'm a researcher and I'm interested in trying to look at how we can do better suicide prevention, depression, and how we can prevent mental health disorders before they actually arise, particularly in youth. Think back to the 1990s. We didn't have Medicare, we didn't have anything covering psychological services, so you actually had to pay. Around the same time, people started using the internet, and so that started the whole process of using cognitive behaviour therapy skills that I already had, putting it into internet-based format, so you actually got interactive therapy. One of our first websites, called Mood Gym, was used in nearly 200 countries around the world, 1 million users. And what we did was measure the progress of the people using standardised tests across the website to say, wow, they seem to be getting better. They seem to be getting less anxious and depressed. I think my whole career has been predicated on the idea that we can use technology to transform mental health. So you've got the smartphone. It provided the opportunity to develop apps on the go, to take measurements on the go. There's a lot of research now on trying to say, if somebody's at risk of suicide, is it possible to deliver a message to them or some sort of intervention on the go at that time, in real time? At the moment, one of my concerns is that young people go to ChatGPT and use it as if it's a therapist. And of course, there's a rush now to develop different large language models that would be sophisticated enough to be able to respond appropriately and to have those tested. So yes, we do have to be careful, and it is a balance. I'm really delighted to be a member of the Academy of Science, and I think there is a perception that psychology is a soft science. All my work that I've done, I think, is about how strong psychology is a science, both from the point of view of measuring things properly, but also to using scientific methods in every possible way to show that psychological changes can make a real difference to people's health.
Good afternoon, everyone. Welcome back. We hope you all enjoyed good food and good company during the lunch break. Certainly, I had to rush. Before we hear from our awardees, I would like to again mention our event partners. They help us make this event possible and such a remarkable celebration of science. This morning, in particular, I would like to this morning, it would have been this morning one, but Qantas had got different plans than we have planned for. And this afternoon, in particular, I would like to thank one of our major event partners, the University of Queensland. UQ has been partnering with the Academy of Science at the Shine Dome for many years, and once again this year, they are our diversity and inclusion partner. This partnership allows us the Academy to offer support of all kinds to attendees who may not otherwise be able to attend. We offer support such as childminding here in Canberra, support for carers for their caring responsibilities at home, and covering costs for attendees to travel with carers. The Academy is a national leader in diversity and inclusion in Australia's science sector, and we are proud to be able to offer this support for attendees at our flagship event. Now, to speak on behalf of UQ, please welcome Academy Fellow, Professor Alan Rowan. Alan, please.
Thank you, Professor Jagadish. Thank you, academicians. And on a personal note, thank you. It's such a pleasure to be here and listen to all the intellectual science today. It's been an absolute joy. It's a pleasure to be here as a science in the Shine Dome, representing the University of Queensland. But I'd first like to begin by acknowledging the traditional owners on the land of which we meet, the Noel and Ngambri people, and pay my respects to the elders past, present, and emerging. And I think especially at the Academy, we should really embrace the wealth of knowledge and connection to the country the First Nation people bring to our community. So important. I'd also like to acknowledge the fellows, distinguished guests, colleagues from across the government, industry, and the research community. And I pass on my apologies for the UQ's Vice-Chancellor, Professor Deborah Terry, who's unable to attend today. When I think about the roles of universities in society, one idea comes to mind. Society isn't always set up to give everybody a voice, but universities are. Those voices can make contributions that reach far beyond the campus. Those voices can be nurtured in a similar way we might train a soprano, an opera singer, nurtured to be the best and to bring the best, a great gift to society. Universities must remain places where different opinions and backgrounds come together. All creeds and colours must be tolerated, all nationalities, a place which supports all people, many who may not fit elsewhere. And I think as an academician, we all know that we are relatively unique people, and a university is a place where we could go and our abilities could shine. A place which not only fits all people, but has this unique talent, a broad open church for all. It isn't always easy, but it's essential, because diversity drives innovation and discovery. It's inclusion that makes collaboration work. We have serious problems across the globe, from global warming to aging, to the role of AI and the sustainability, to name but a few. Global problems which will require the efforts of many from all walks of life in a cooperative way. And it's the combination of inclusion and collaboration that will allow us to solve problems that no single person or single discipline can solve alone. We need that variety of persons if we're going to meet the challenges ahead. And we are only going to achieve real progress if we involve all. At UQ, we have been working to make such an inclusivity as part of everyday practice. We recently set up the National Indigenous Science and Technology Centre, specifically creating pathways for the First Nations students to enter and thrive in STEM. The first national centre dedicated to helping develop Indigenous entrepreneurship, to turn the vast wealth of knowledge of the First Nations people into real outcomes and viable businesses in the community. And I'm delighted to report that this year we will have the first Indigenous-owned companies starting from wound dressings through to seed coatings through to dermal fillers. Through the Athena Swan process, we're making structural change, genuine structural change, to improve equity and diversity across our research culture, making it a level playing field for all. And through the Queensland commitment, we're ensuring that the financial background of a student is never a barrier to a higher education. These things matter because science is ultimately about people and teams. People whose curiosity drives discovery. People with different perspectives and strengths. People who are prepared to share ideas, test them, and build something better together. I also want to really recognise the steps that the Academy has taken this year to make this event even more inclusive. This includes support with childminding at sessions and evening functions, flights for family members, family-appropriate accommodation and accessibility transfer. And I noticed one of our fellows this morning, Professor Webster, told us about that difficult role of trying to juggle family and academia. As a fellow academician, I know there is still much to be done. As director of the AIBN, one of the largest biotech institutes in Australia, I can actually see how small changes can have an amplifying effect in empowering all to achieve. I see the joy of these up-and-coming ECNRs to do their passion, while at the same time we support them being able to share time with their family. So as we gather here to celebrate Australian science, I hope we can also celebrate the role of our universities as incubators of talent, as places of tolerance and inclusivity. They are places of discovery and debate. They are where the next generation of researchers are trained, and they are one of the few institutions in society that are designed to look beyond the short-term gain to create knowledge that lasts for all of society. They are going to be the critical component in accessing this talent and helping raise Australia's productivity to benefit all. If we keep them inclusive, collaborative, and strongly connected to the world around us, they will continue to deliver for society, not just for today, but for generations to come. Thank you.
Thank you, Alan, for those thoughtful words. And we completely agree with you the importance of the role of the universities in Australian system and in the global system as well, to be able to create opportunities and train people to be able to solve the global problems, and also the importance of inclusion of everyone so that collectively we can address these complex problems society is facing. And once again, thank you to the University of Queensland for your ongoing support. But Alan, we would like you to stay with us on stage, even though you were elected in 2020 as a Fellow of the Academy, you are yet to sign the charter book. So we'd like to invite you to do that today. I will now hand over to Francis to read your official citation.
So thank you, Jagadish, and thank you, Alan, for that lovely speech. So Professor Alan Rowan is well known for his seminal work in diverse fields ranging from single enzyme studies, single molecule magnets, organic solar cells, biomimetic catalysis, through to hybrid catalysis, biomimetic gels, and polymer therapeutics. So quite a range there. Rowan has a passion for enhancing communication between the science community and the general public, and for training the next generation of scientists, which is a particular strength. Many of his postgraduate students hold permanent academic positions worldwide, while others are employed in the chemical industry. So congratulations, Alan. So now I think you move over there to, I probably should congratulate you.
Thank you very much.
Congratulations. Let's move over to
Congratulations, Alan. I welcome you to the Australian Academy of Science Fellowship.
So we'll now hear more about Alan's work, noting his video was produced in 2020, and we recognise he's probably done a lot more since then. So thank you. Please play the video.
Understanding nature and trying to mimic it is at the heart of biomanufacturing, and by redesigning cells, scientists can make new products, new materials and new catalysts.
The Egyptians were taking yeast, giving it sugar and making beer, one of the world's greatest products. You can now reprogram a cell to feed to a yeast cell sugar and get it to make vitamin A or vitamin E or a variety of different products. So this redesigning of nature to make new products is one area we're extremely successful in.
Professor Alan Rowan's research in bioengineering and nanomedicine has created a variety of new products, and while developing polymers for banknotes to make them unforgeable, his team developed a whole new family of materials.
One bag of sugar of this material would gelate your whole swimming pool. So you suddenly go, well, that doesn't work. It forms a gel when you warm it up, and a tiny amount gelates all of this water. So it's 99.99% water, but it's solid. And that was that eureka moment. We don't understand how this works.
It then took a team of 20 people 5 years to understand the unique properties of this new substance, which stiffens when a force is applied and mimics materials in our body. And interest was widespread.
Could we use it in banknotes? Could we use it in medicine? Could we inject it into aneurysms? Could I use it for people who have amputees, where there would be a material which would stiffen up when they felt this force?
He also works in nanomedicine, tailoring medicine to people's specific needs.
We try and understand how human cells work, particularly in areas of aging, bone growth, cell differentiation, and we work with some people now trying to grow organs so that we can test drugs on real organs rather than going and testing them on mice or humans.
Professor Rowan says he always wanted to be a scientist, and his teachers predicted it. But he decided to have a short career as an Air Force pilot beforehand.
When I was 13 years old, my teachers at school said, you're going to be a professor. In spite for them, I joined the Air Force at 17 to become a pilot, and then was flying planes at the same time as actually doing chemistry. And chemistry won. They were right.
Now his passion is training the next generation of scientists.
And so that becomes more and more the joy, is to see these people grow. You still get the pleasure from the final result, but to see it done through other people is more pleasurable. First it was about me, then it's about the team, and now it's about a bigger team, and I think that's what really is pleasurable.
So congratulations, Alan. We're also joined this afternoon by one other fellow who will be signing the book. So he was elected in 2022, so we're going to welcome Professor Peter Hoy. So Peter Hoy has made distinguished contributions to Australian society, particularly through more than 2 decades of leadership as Vice-Chancellor of the University of Adelaide, the University of Queensland and the University of South Australia, and also as CEO of the Australian Research Council and the Australian Wine Research Institute. Hoy has also been a member of numerous high-level policy committees following his early career as one of Australia's leading young biochemists. And you still are. Yeah. So now I invite Peter to sign the book. So congratulations, Peter.
Thank you so much.
Congratulations, Peter. I welcome you into the Australian Academy of Science Fellowship.
Thank you for having me.
So we'll now hear more about Peter's work. So we've got a short video, and note again, this was made 3 years ago, and since then he's been involved in a lot of other things, including the merger of the University of Adelaide and the University of South Australia. So please play the video.
For Professor Peter Hoy, scientific curiosity began while cleaning the floors of his father's fish shop in Copenhagen.
I remember this very clearly. It was 1967, so I was 10 years old.
I observed some blood on the floor, and under those blood clots, the floor was beautifully white. And I said to my dad, "Why is that?" And he said, "I think it's something called enzymes." And I thought, I got to figure out what an enzyme is.
He would go on to study biochemistry. While working in fundamental research at Denmark's Carlsberg laboratories, he met an Aussie girl.
We both got our PhDs and got married, had children, and decided to move to Australia.
Professor Hoj was named one of Australia's top biochemists under 40 and was soon tapped to join the University of Adelaide as Australia's first Professor of Viticultural Science.
And one of the reasons why you appreciate Australian wine is it's incredibly rare that you buy a bad product. We understand so much about the underlying science in Australia that we virtually get it right every time.
His love for the application of science saw Professor Hoj lead the Australian Wine Research Institute and then the Australian Research Council before being named President and Vice-Chancellor of the University of South Australia.
When you're fortunate enough to go into large universities, you realise how much talent there is. And whilst I might be missing doing my own science, I know that if I can improve the working conditions for the 1,500 other people who are equally good at it, then my impact is much greater than it is as a singular scientist.
Boosting research talent was also his priority while leading the University of Queensland to a top 50 global university. And now, upon his return to South Australia, as Vice-Chancellor of the University of Adelaide.
We have a very, very low gross expenditure on research in Australia, and mostly because industry and business is not investing enough in research.
Professor Hoj says that's one of the key reasons international students are so important.
It would be fair to say that international students are so keen to get an Australian education that they are prepared to pay fees that actually greater than what you get from an Australian student. And that fee differential is something that actually underpins much of the research that we're doing in Australia.
He describes it as a major win when some of those students decide to make Australia their permanent home, just as he once did. Professor Hoj has been named a Fellow of the Australian Academy of Science in recognition of his distinguished contribution to society.
So congratulations, Peter. So now we're going to hear from another of our career awardees. So firstly, the 2025 Jaeger Medal. This is a career award made in honour of the contribution of the late Professor John Conrad Jaeger to Australian earth science. The award is made to a scientist for investigations of a high order into the solid earth or its oceans carried out in Australia or having some connection with Australian earth science. The 2025 recipient of the Jaeger Medal is Academy Fellow Professor Hugh O'Neill. Professor Hugh O'Neill is a widely recognised international leader in the field of experimental petrology and geochemistry with applications to the understanding of planetary processes. His research addresses many aspects of the chemical behaviour of minerals and rocks through detailed laboratory studies complemented by thermodynamic modelling. The resulting insights elucidate the diverse processes by which Earth and other planetary bodies were formed and their subsequent chemical evolution. His influential research has provided new insights into the formation of the Moon through the impact with Earth of a Mars-sized body, the segregation and composition of Earth's iron-rich metallic core, the redox conditions prevailing within Earth's interior, and the generation of basaltic magmas by partial melting within Earth's upper mantle. So Hugh, congratulations.
Francis, thank you.
So please.
Congratulations, Hugh.
Anyone who stands here has got many, many people to thank for the journey, and I can see some of them in the audience. It's actually 50 years last week since I became what I am, which is an experimental petrologist. And I thought that I would just take some more recent excerpts from this 50-year journey to tell you a little bit about what people like myself do. Experimental petrologists are not strangers to the Jaeger medal. The two first winners, the late David Green and also the late Ted Ringwood, were the first two winners, and both of them have been strong mentors in the early stages of my career. So I'd like to thank them, if you like, in retrospect, for setting me on this journey. In fact, 50 years ago, when I turned up at the University of Manchester to be supervised in my PhD by Professor Bernie Wood, who's still going strong, he wanted me early because he was going off on sabbatical to the United States. So we spent a couple of days looking around the lab. He showed me how to do things. We made up a starting mix. We put part of it into a high temperature furnace in order to sinter it into something that looked like a rock. And off we went to the pub, came back the next morning, and there was this unholy mess all over the laboratory bench. The thermocouple had failed in the night, and the temperature had reached in excess of 1800 degrees centigrade, which I know because the platinum had melted. And some people might have been put off by this. I was actually thrilled. I'd made my own little volcano. Right, so I've always looked at the way that we do experimental petrology as being entirely complementary to a very large part of the earth sciences. Some people would say that we do experiments at high temperatures and high pressures in order to simulate what happens in the earth. I do not believe that, and this is very important. I believe that we are here to provide the hard physical chemical data that we can put into models for how the earth works. This is something I learned from Bernie Wood and has been right throughout my own career. This has enabled me to collaborate with a wondrous collection of colleagues on all sorts of different problems. And you talk to people. When it comes to science, I'm a social animal. And, you know, we need to know this, we need to, oh, well, look, perhaps I can go back home and do this. And sometimes it works, sometimes it doesn't, but you're continuously learning. So it's been 50 years of further education on my part. Right, what do we do in experimental petrology? Well, basically, as I said, to me it's physical chemistry at high temperatures and pressures because I'm interested in the interior of planets like the Earth and also Earth-like planets. Sometimes we use simplified compositions chemically, sometimes we use natural materials. The two are complementary to each other, so it pays to look at both. And I thought that I'd just sort of talk about two of the fairly standard apparatuses that have been so important in my own career. One is the piston cylinder apparatus, high pressure, high temperature. The other is something I started after I'd been going for a decade or so, which is to look at rock-like samples at high temperature but without the pressure, but under very carefully controlled conditions, usually involving gas mixing. And I'm going to just run through some fairly recent, which means last decade, results using sulfur, which was a fairly new development that I got onto about 20 years ago. It took some time to get it actually going. And we put these into furnaces that these days can go up to in excess of 1700 degrees, very precisely controlled with temperature. If we're down to below 1500 degrees, we can actually control it to 1 degree centigrade, and we need this for getting the good thermodynamic data out of these experiments. And these are roughly what the samples look like. You'll see half a dozen or so different silicate melt compositions hanging off the end of a tube there that then goes into a furnace that you can see above me. And at the end of a certain amount of time, with gases that you have flowing past to provide the atmosphere, that gets dropped into a bucket of water, mounted up, and then taken off to all sorts of different analytical facilities. I've got to say that a major, if you like, change of direction in my career was when synchrotron science became available, which once again was an outcome of talking to colleagues, Andrew Berry, and off we went to do some of the first quantitative measurements using X-ray absorption near-edge spectroscopy, the first quantitative because having access to the laboratory, we were able to make careful matrix-matched standards for the first time. Anyway, back to sulfur. Why is looking at the solubility of sulfur in silicate melts important? I thought I'd put this up to show you that there is more than one reason. In fact, the applications encompass a large area of the solid earth sciences, and even beyond the solid earth sciences, there is overlap with metallurgy. And by collecting these fundamental thermodynamic data, we can feed in to all these other exciting areas. One of which I'll actually go back and then talk about the high pressure work. So this is the piston cylinder apparatus. We can take this up to 60 kilobars, 6 GPa, which corresponds to about 200 kilometers depth in the earth. It is perfectly possible to go to far higher pressures, but not with the sufficiently large sample sizes that you need for really precise control of pressure, temperature, and importantly, the environment, the chemical environment. So we can control temperatures in the piston cylinder apparatus to plus or minus 5 degrees. I've just recently been working on refining the measurement of the pressure in these apparatus, even though they've been going for 80 years. This is not sort of absolutely solidified science yet, and I reckon that we can do it to 0.02 of a GPa now. And I'll skip over that. This is how we put an experiment together. The heating is just by electric current through a graphite heater. You've got to insulate that from the pressure vessel, which is on the left-hand side. This is back to front here. And at the end of the experiment, you turn off the power, you recover the sample, and the analysis chain is pretty much like the 1 atmosphere experiments. And this is a before and after. So the after is a real-life experiment done at about 3 GPa and 1400 degrees centigrade. And you can see the structure of the experiment after looks reasonably like what we wanted it to be from the before diagram. Something else that I thought I could not leave out, considering that this is the John Jaeger medal, is we started looking at diffusion in silicates because constraining the important variable of time in geology is largely done by radiometric methods, but these have their limitations. and alternative ways are always of interest. And an obvious one is to look at diffusion profiles, and it was Castlaw and Jaeger, 1947, that wrote the book on solving the second-order differential equations for actually heat flow, but it's exactly the same equation as for diffusion. But what I do is not the mathematics. I actually look at obtaining the diffusion coefficients. And we developed a novel way in which we can measure diffusion of a good portion of the periodic table in one experiment. And the advantage of this is that we can compare the different elements and how they diffuse. And this gives us a really good insight into the point defect mechanisms of diffusion. Now, this is an experiment with olivine. I also did some on magnetite fairly recently, and you can see the results, a very interesting dependence on the oxygen activity, oxygen fugacity of the environment. That's why this environment is so important. And this mechanism corresponds to the development of point defects and so on and so forth. What we've discovered is that a lot of the existing experiments in the literature are severely under-constrained as regards variables, and that's being optimistic. Often, what these people were measuring was probably not actually diffusion at all. So this is a wonderful new direction for someone like me that likes to get up, let's say, people's nose and say that, no, you haven't been doing it right. Have a look at this, guys. Last thing is that just doing rocks in the lab is not necessarily as fun as traditional geology of going out there and looking at rocks in the field. This was highly necessary for my interests because of what the Earth composition was like. And when I started this in the late 1970s, you only got sort of glimpses of the Earth's composition from nodules in basalts, but now we know there are wide swathes of upper mantle that have been tectonically emplaced on the Earth, including holiday islands like New Caledonia, good place to do field work. Also the Italian Alps. I'm one of these people there, sort of hammering away at the rocks. And I'll just finish with a view about artificial intelligence. And I don't know if you guys have thought about this, but this is an abstract, perfectly normal Q O'Neill abstract that was submitted to a conference earlier in the year. And you'll see that, you know, it's the usual technical stuff, perhaps a little bit boring. I don't expect you to read more than the first line or two. I won't give you time, because I'm going to show you what ChatGPT can do. Thank you very much.
So thank you. And don't forget. So we've got time for a question. We just had some hot chemistry, so any questions for Hugh? Lynn?
Thank you so much. It's a fascinating area. I know you could talk for many hours about your work on Mars and the Moon, but can you just give us little tiny more glimpses into that, please?
Well, it's expanded to exoplanets. And basically, a big question is, what is the chemistry of a planet that we need to support the development of life like we have on Earth? And since one of my long career interests is in the oxygen fugacity of the Earth's mantle and rocks and everything else, I'm very interested in oxygen in exoplanets. And if I can just sum up a sort of 3-hour talk, I believe that we're here by accident. I believe that there was an extraordinary coincidence that we had events in the building of the Earth, everything happened to be in the right order, that we ended up with a sufficiently oxygenated solid Earth that we could evolve the CO2, H2O, rather than methane hydrogen gases, at the right distance from the sun, because also Venus did that, but it's just too hot on the surface of Venus to maintain an atmosphere. So it's, this has been said 50 years, it's Goldilocks.
Just right.
Yeah.
So Hugh mentioned he's very sociable and he likes to talk about his work, so grab him at afternoon tea and at the dinner tonight. So thank you very much, Hugh. Congratulations. I'll just hand over to Stephen now.
Thanks very much, Francis. Our next awardee is the 2025 David Craig Medalist. This medal is awarded in honour of the outstanding contributions to chemical research of the late Professor David Craig. It recognises contributions of a high order to any branch of chemistry by active researchers, and the recipient of this medal delivers several public lectures across Australia. The 2025 winner of the David Craig Medal is Academy Fellow Professor Alison Rodgers. Scientific advances invariably depend on the quality and diversity of available techniques and instrumentation, and on the ability of researchers to understand the data that are produced. Professor Alison Rodger has spent her career inventing new spectroscopic techniques to advance understanding of the molecular world. She's used polarised light to give data about the helical structures and molecular assemblies. She complements the experimental work by developing the theoretical frameworks required to enable use of the data, and applications such as characterising the structure of biopharmaceutical products and understanding the basic biology of cell division. Professor Rodger's most recent invention is that of linearly polarised luminescence, where the intensity of polarised light emitted is used to characterise biomolecular assemblages such as DNA drug complexes. She's passionate about working to ensure equal opportunity and has benefited from working with a wide range of people from all over the world. So Professor Alison Rodger, congratulations, and please join us on the stage to accept your award.
Congratulations, Alison.
Everybody else made the technology work. Now, right. That is just typical. I am really pleased to be here, and I'm honoured and delighted and completely intimidated, so it's not surprising that I made the technology go wrong. I've had a career that's benefited from being in a number of different places, funded by a wide range of different organisations, but most of all, I'm really, really grateful to my co-authors, because usually they're the people who forced me to think about something differently, do something new, because they sort of assumed I could help them solve a problem, and that's been a brilliant inspiration. So we've heard quite a lot about science and its social and political context, and I would say it's also limited by the available technology. This caused me to go back and say, what was I doing in my first decade or so? Well, I enjoyed mathematics and experiments with light. There was a memorable fortnight when I was 10 or 11 where the girls were made to go into the woodwork shop, and that was brilliant. I made a pencil case, and about 7 years later, I actually sold it to someone who wanted one. And I think that was probably the origin of founding a company to sell equipment I'd made. Quite what we did in the other 40 or so weeks of the year, I don't know. I vaguely remember my failure with a sponge cake, and I couldn't understand how the treadle sewing machine worked. So that's the very early stages. My first research project was really when I started honours with Eddie Skipper at Sydney University. Eddie was one of David Craig's PhD students, so I claimed David Craig as my scientific grandfather. Eddie started me off on my research career by being gone when I started and leaving me a paper with a note saying, when you understand the error in this paper, you'll be on the right track. I then, after my PhD, which was examined by David, went as an independent research fellow to Cambridge. And I realised that I couldn't do experimental work because I didn't have laboratory facilities, so I'd have to be a theoretician. And I needed some guidance. And so I actually used my first research grant of £38.50 to buy David Craig's book. And it really did guide me and still guides me. My second research grant was for a spectrometer, which meant that I then could do experiment and theory. David Craig's book is still with me, a bit worse for wear. The spectrometer has disappeared. So what's my career been about? I started off with biomolecules and focusing on their handed nature. Their mirror images are usually not the same. But to measure the chirality, the handedness of those molecules, we have to have a chiral tool. So the first spectroscopic technique I worked with was circular dichroism, which is the difference between absorption of left and right circularly polarized light. We get traces that we can then translate with, if we've got the theory sorted, into understanding of molecular structures. Bengt Nordin introduced me at about this stage to the wonders of linear dichroism. Linear dichroism uses linearly polarized light and gives you relative orientations of molecules in systems, such as molecules binding to DNA. Alternatively, if you're working on a budget, what you can do with a circular dichroism machine and a quarter wave plate or other adjustments. Perhaps the experiment that led to the most variety of things I've studied was a real failure. At the time, polymerase chain reaction was really exciting new technology, which some of you won't remember, but we wanted to measure real-time PCR. So I was working with Tim DeFawn and Dave Halsall, and we did a proof of concept experiment that did seem to be just about right. And we thought, great. And then Dave said, 20 PCR reactions for the first experiment's fine. You've got to reduce the sample requirements by at least a factor of 20 for the second experiment. So I went away for about 3 years. Meantime, other real-time PCR instrumentation was produced, but I did reduce the sample volume required for flow linear dichroism, and that was really good because biologists started talking to me because I was in their sample domain rather than the chemist's sample domain where you've got grams of stuff. Biologists have so little that you have to work on their dimensions. So what have I been doing more recently? I've been looking at combining fluorescence and polarized light spectroscopy. Fluorescence is where somehow you get the molecule to an excited state and you measure the light that is emitted. It's more selective than absorbance, which is good and bad, and it also measures properties of the excited state, which can be useful. But there was no instrument to do what I wanted to do. So I spent a long time talking to Jasco to get them to design one instrument that was a combination of two of their other instruments with an extra detector and some extra capabilities to do linearly polarized luminescence. So successfully measured, that was great, and measured the first wavelength scanning linearly polarized luminescence experiment. So I was really excited about that, and I'm now looking for good applications if any of you've got any of them. And we can do things like compare different binding modes, again on DNA. This was the last time that I've actually used David Craig's book seriously. It's now available in Google Books, and so you can get it there and also follow my journey. At about this stage, I realised that electronic spectroscopy circular dichroism wasn't going to be good enough for protein biopharmaceuticals because the companies were making them more and more concentrated. And I went on a journey to understand how to do infrared spectroscopy accurately, reproducibly, and as with so many other things, it required do some experiments, test things out, understand the theory, so I could know what we were in the end measuring. Again, collaboration's been really important because coming from a different perspective, I realised that circular dichroism methodologies might work for infrared. I could do it manually with a spreadsheet, but I needed colleagues to code it up for me, which is exactly what Søren and Nick did. So infrared is not quite as good as circular dichroism for protein secondary structure, but it's nearly as good now. So that was great. I then wanted to do polarised light, and this is really a work in progress. We did transmission fine. We can measure stuff with an attenuated total reflectance unit. But if any of you are on the ball, if you look at the bottom, you'll realise that I've got linear dichroism spectra for completely isotropic solvent molecules. Beautiful spectra, but it should be dead flat. So something's really going wrong. And I think this is about science. You try something, it works or it doesn't work, and you then have to just keep going. So it looks promising, but, and I hope maybe the next time I see most of you, I will have sorted it out, but it's a bit challenging. Where I'm at the moment is saying, how can we use spectroscopic techniques for really complex systems? And I've got involved with an ARC-funded industrial transformational training centre in bioactives, and we're trying to look at what bioactive products contain, how the molecules arrange themselves, what are they? How can you make the same product twice? All a bit complicated. Good old infrared spectroscopy is really helpful for molecular identification and quantification. We complement it with a lot of other techniques to try to understand how the molecules are assembled in products. And finally, I very recently moved to ANU, April last year. I found a great school of chemistry, wonderful colleagues, but about a month later, the budget crisis hit ANU. And I said, where does our budget go? And I discovered to my horror that we spent a quarter of our non-salary budget on waste solvent disposal. If your university pays for your waste solvent disposal, you may not have worried about this, but I pay out of the school budget. So after looking around and trying to use my scientific expertise to solve this problem, we bought 2 solvent recycling units. We use NMR and infrared spectroscopy to check what's happened. Molly and Wally are named after local rivers, and my colleagues have really been great. They've used them, pure solvents, and we've reduced our waste solvent by 50%. My challenge is look for how to do your science more sustainably. And just lastly, none of this would have been possible without the support of my family. I don't burst into tears in front of you. I need to find a cave to hide in.
Every time I give a talk and I mention my family, I burst into tears as well. So that was just tremendous, Alison. And I'm not sure what our waste solvent bill here is at the Academy, but perhaps a way to reduce the budget deficit. Mel? Questions from the audience?
Or suggestions for sustainable science.
Or suggestions for things that you might do with the wonderful things that you're inventing spectroscopically. There's one.
You're wonderful, Liz.
Thank you. It's just wonderful to hear your story from the beginning and look back on that young lady who made her first sale. What advice would you have to young scientists coming through? Because you have such an inspirational, important role in science, yet there can't be many who follow your trail. It must be a very unusual one. How do you inspire the next ones?
I think my advice, which you must have all worked out, is when you get an opportunity, take it. It may sound slightly mad or it may not be quite what you wanted. Remember your direction of travel and take every opportunity that comes your way.
Thank you.
And then you need a very tolerant family.
And spend your entire grant on a textbook. I think that's wonderful. So I think on that note, many, many congratulations. Back to Francis.
So next is our 2025 Thomas Ranken Lyle Medalist. This is a career award that commemorates the contribution of Sir Thomas Ranken Lyle to Australian science and industry, in particular to his own fields of physics and mathematics. The purpose of the medal is to recognise outstanding achievement by a scientist in Australia for research in mathematics or physics. And this year it is awarded to Professor George Willis. Beyond solving problems, Professor George Willis is a true creator of new mathematics. Through his invention of the scale and its function, Professor Willis gave an entirely new insight into the unexpected structures and classification of totally disconnected, locally compact groups, a previously intractable area. This novel approach, now known as Willis theory, has broad implications for diverse fields of mathematics. Professor Willis's research extends beyond theoretical elegance, leading to unexpected and pivotal real-world applications related to symmetric infinite networks. By describing how finite patterns might continue indefinitely, his work informs the growth and optimization of real-world networks like computer systems and global social networks. In a lifetime of achievement, Professor Willis has ensured Australia is at the frontier of knowledge in pure mathematics. His ongoing contributions add to his already remarkable legacy of innovative academic research and dedicated education leadership in pure mathematics at the Regional University of Newcastle in Australia. So congratulations, Professor Willis. Please join us on the stage to accept your medal.
Congratulations, George. Thank you.
That's 2 cents. Okay, I think I'll just have to start. Okay, yes, it's a great honour to receive this award. It's not something that I imagined when I started out and when I was doing this work, and so I'm very grateful to the Academy for the award. I also am very grateful to the various people I've collaborated with and who've inspired me over the years. There are a couple of photos at the end. I'm afraid I don't have a gallery of photos as many other people have. But also, as was mentioned, I was able to do this work at the Regional University of Newcastle, and it was great to be able to share the fundamental research and international research with students there, with local students. And it's notable that 2 of the new fellows this year are Newcastle products. And so I think it's very important to be able to continue the fundamental research and research at regional universities. So let me begin, and I'm going to be speaking about some concepts, and so that's a bit of a challenge. How do I Here we go. Yeah, so what I'm going to do is I think I'll be confronting you with a few words and concepts on this slide, and then I hope on the subsequent slides telling you what they might mean by relating them to other things you might be familiar with. So the let me say first of all what a group is. It's an algebraic structure, and groups encode symmetry or reversible transformations, and that being an algebraic structure just means you can multiply things in the group and find their inverses. And the scale is this is the core idea, and it's just a positive integer that is the function on the group, and it's the related concept is that of the tidy subgroup. So if I can, yeah, okay. So the tidy subgroups have a standard form which really pictures them as a rectangle, as shown here. And they, in Robert Marnie's talk yesterday, he talked about the importance of having the right coordinates. Well, the tidy subgroups are the right coordinates to see the scale. And the standard form pictures them as a rectangle, and then the group element sort of expands this rectangle in one direction and shrinks it in another. So here we see it expanding by a factor of 3, and so the scale here we're seeing is 3. Now this picture also lets me say something about what totally disconnected might mean. The rectangle here, I can do this, yeah. This rectangle is divided into 3. It's not a connected rectangle, it's disconnected. It's divided into 3 parts in the horizontal direction and 2 vertically. And so it actually is divided into 6 smaller rectangles, and each of those is then divided in the same way. So if you imagine this sort of iterating, and we continue dividing each of these smaller rectangles according to the same pattern, then what you end up is with totally disconnected. So that's the idea of a totally disconnected set. And then if we reverse the element here, we expand by a factor of 2 in the vertical direction and shrink by a factor of 3 in the horizontal direction. There's one other concept that I wish to mention, and that's the idea of a flat group, and these are related to having multiple elements. Now, next I want to say something about locally compact groups, and so they are groups, but they also have a notion of a topology, which gives you a notion of continuity on the group, and it was something that was shown in the 1930s that on locally compact groups you have an integral. Now, a typical example of a locally compact group is just the real numbers, where the algebraic structure is addition, and that's modelling or encoding translation of a line. And then there's a topology comes from the usual distance measure on the line. So that's a typical example, but the locally compact groups are some abstractly defined structure, but they have an integral. This is something that was shown in the 1930s. We can integrate functions on locally compact groups, and this is just as we integrate when you learn calculus, learn about integrating functions on the line. And those two features, being able to translate functions with the group elements and being able to integrate, is really all that's needed to do Fourier analysis. So locally compact groups are the natural domain in which we can do Fourier analysis. And a general locally compact group factors into 2 parts. There's a connected group and a totally disconnected group. Now, the connected groups are, this is something also that Robert Marnie talked about yesterday. He gave some examples, but they are essentially groups of matrices, as he was talking about yesterday. and functions on those can be differentiated. So we have the other aspect we learned in first year calculus of being able to differentiate the functions. And the Lie groups, I said they encode symmetry. Lie groups, or these matrix groups, encode symmetries of smooth structures. And the totally disconnected groups, on the other hand, they encode symmetries of digital structures, such as networks. But they also have some importance in number theory. Now, so here we have on the left, we have a sphere. So this is an example of the rotations, or example of a Lie group, and the isometries or symmetries of this tree structure. This is, you should imagine this continuing, the branching continuing infinitely here. Now, I want to say what the scale and so on are by making a comparison. So the Lie groups, or the matrix groups, some examples are here. There's a group of rotations of a sphere. There's the Euclidean group and the Lorentz group. I think the Euclidean group is probably what Robert was referring to as the Galilean group yesterday, and then there's the Lorentz group, which is a group of symmetries of the equations of space-time and special relativity. These are all matrix groups, but the difference is that TDLC groups need not be matrix groups. For example, the isometries of a tree are not a matrix group. Now, what that means is that for matrices, they have eigenvalues, and they are a very powerful tool for studying matrices, but also for studying Lie groups. We don't have that that tool available for the totally disconnected locally compact groups, and the scale is the analog of an eigenvalue, or a set of eigenvalues. And similarly, we have a basis of eigenvectors for a matrix. That's also a very important fact in studying matrices and studying Lie groups. We don't have those for groups that are not matrix groups, but the tidy subgroups are the analogues of the basis of eigenvectors. And then commuting matrices have common eigenvectors. This is also a very important fact. And the analogue here is flatness. And when we have a flat group, the tidy subgroup and the scale actually factor into a spectrum of subgroups or values, just as we see for the spectrum of a matrix. The scale is just a single integer, but once we have a flat group, we actually get a spectrum of scale values. Okay, so that's the ideas. Now, let me just say something briefly about, there are many consequences of this, but let me say briefly one of them, and that relates to something called superrigidity. So if we have matrices with integer entries and a determinant 1, they form a group, and it was shown by Fields medalist Grigory Margulis in the 1980s that there's only one embedding of that into a connected group, and that is into the obvious one. There's a conjecture was made for a similar statement about totally disconnected groups, and I was able to prove that with the Hugh echelon. Now, to sort of finish, I would like to say something about relating the trees here to this picture of the tidy subgroup. So here we have a tree where every vertex has 3 in edges and 2 out edges. Now, the in edges here, each of those has another 3 coming in, and so on. That corresponds to the continued division of in the horizontal direction here into a third, and then a third, and a third, and so on. And similarly for the out edges, in halving and then halving again. The symmetry here is just a translation along this axis, and the tidy subgroup is associated with this vertex. When we translate, the tidy subgroup comes up here, and you now see this expansion here corresponds to picking up more in edges and losing some out edges. And yeah, so That's how these two things relate. In a general group, we don't have this nice tree picture, but we do always have this rectangular picture. That's sort of the point. Now, I hope I've been able to get something across of what this is about. It's a bit hard to take in. Let me just finish with this sort of Tim Tam version of the same picture. It might be easier to take in. So this is something I did at there was a graduate student event where students showed photographs of their research, and I helped out with the catering by doing this. And this also is another occasion at a conference in Canada. It shows a couple of my collaborators here, Helga Glockner and Pierre-Emmanuel Capraus. There's another one present, Jackie Ramage here, and but yeah, unfortunately I don't have photos of the others. Thank you.
So thank you, George. So do we have any questions? I know we mentioned Euclid yesterday and Lee Group, so they should be in the audience. Yeah. No questions? Yes, just up the back there. Thank you.
George, could you say a little bit about, in the introduction it said it has applications to things like large social networks. Could you say something about that?
Well, I think that was a bit of a stretch. I think that was something that the person writing the nomination put in there. But what was also mentioned was networks, and there is a connection there with binary search trees. Now, this actually comes back to the mention of Euclid yesterday. I think Jessica mentioned Euclid. The second axiom talked about if a line can be extended infinitely in any direction. Now, in practice, people don't draw infinite lines. They only work with finite line segments. And in binary search trees, these are a way of storing information, and they are, in practice, you only ever use a finite tree. But in principle, they can be extended, and that's actually what you want, because as you add more data, you have to extend the tree. And so, in principle, they can be extended just as a line can be extended to be infinite, right? And so that structure is an example, gives an example of a totally disconnected locally compact group, and the symmetries there correspond to the sort of operations that people want to do to, there's a notion of balancing trees in order to keep them efficient. And they correspond to certain symmetries which give rise to a totally disconnected locally compact group.
All right, thank you very much.
Oh, thank you.
So I wonder how long the Tim Tams lasted. But anyway. So we're just going to move on to our next prize winner. Our next award is the Nancy Millis Medal, which honours the contributions made to science by the late Professor Nancy Millis and recognises her importance as a role model for women aspiring to be research leaders. The award is open to women mid-career researchers, 8 to 15 years post-PhD in the calendar year of nomination in any branch of the physical and biological sciences. Recipients have established an independent research program and demonstrated exceptional leadership. And this year, it is awarded to Associate Professor Natasha Hurley-Walker. Associate Professor Natasha Hurley-Walker has transformed our view of the radio sky. She uses powerful supercomputers to analyse petabytes of data from Western Australian telescopes to explore the universe. Her discoveries include the remains of stellar explosions, insights into the lives of supermassive black holes, and a new kind of repeating radio source unlike anything astronomers have seen before. Associate Professor Hurley-Walker has mapped the southern sky in radio colour, bringing a new view of the cosmos to the world. Giving public talks and media interviews and filming for documentaries, she has reached tens of millions across the world, inspiring interests in STEM careers. Her work will help Australian astronomers find the unexpected within the Square Kilometre Array, which will be the world's largest radio telescope. So congratulations, and please join Jaeger to the stage.
Congratulations, Natasha.
Thank you all, and thank you so much to the Academy. It's truly a privilege and an honour to receive this award and to carry on the spirit and legacy of Nancy Millis, a pioneer female in science. So today I'm going to talk to you about the radio sky and how I've helped explore it using Australian radio telescopes. So, to kick us off, a quick primer on radio astronomy. Now, the only other wavelength that can get through the Earth's atmosphere, besides the visible light, is radio. So this allows us to see a different view of the universe than we would with visible light, without the expense of building space telescopes. So many of the high-energy processes in our universe involve the interaction of electrons and powerful magnetic fields. This is very different from the processes that produce visible light, like thermal emission that we see from stars. So to the top right and the bottom right, you can see two different radio maps of the universe, one made with a single dish telescope. Part of this map was made with the Parkes-Murray telescope in New South Wales. And you can see this sort of low resolution, black and white image, particularly with this stripe across the middle, which I'll talk about more in a minute. And at the bottom right is an image made with the VLA telescope in New Mexico. And this is uses a concept of interferometry, where we tie together multiple radio telescopes to build up a higher resolution view of the sky. Now, this is a bit of a challenge. You can see that there are some artifacts in this image, and basically all radio interferometric surveys are missing important parts of information about the sky. So we use the two techniques in tandem. Now, we just had a mention of the Square Kilometre Array. So here in Australia, we're building part of the SKA. It will be the world's largest radio telescope. But you don't start such a huge mega project completely ab initio. So 14 years ago, almost to the day, I moved here to Australia to work on a prototype instrument, the Murchison Wide Field Array. So this precursor is in the same location that the SKA is currently being constructed in Yarimana, Ilgaribundara, CSIRO's Murchison Radio Astronomy Observatory. And I'd like to pay my respects now to the Wadjuri Yamaji and the Southern Yamaji, who are the traditional custodians of the lands in which this telescope was built. And it's only with their enduring custodianship and cooperation that we can explore the radio universe together. So this is a very unusual radio telescope. You might have thought of radio telescopes as being like big dishes. You've all seen the movie The Dish, I hope. But this is a very unusual instrument. It consists of these small antennas laid out across the ground, and they're spread out across about 5.5 kilometres of the Western Australian desert. So it's by pulling together all of the signals from this telescope that we can build exciting maps of the sky. Now, the MWA was constructed to undertake several different science cases. So we have solar, heliospheric, and ionospheric physics, galactic and extragalactic, which is essentially everything that's outside the solar system, transient astrophysics, and a very, very difficult experiment, the detection of the epoch of reionization. So early in our universe, everything just after the Big Bang was completely ionized. It then cooled, became pure hydrogen, and then the first stars switched on and reionized this hydrogen, hence the epoch of reionization. Now, as you all know, hydrogen produces a spin-flip transition. This is when the electron and the proton go from aligned to anti-aligned. In that transition, it produces a radio wave at 21 centimeters. So if we are looking for this signal in the early universe, then it is redshifted to very long wavelengths, about a few meters. And this is why the MWA was built at very low frequencies, and so will the part of the SKA that's built at the same observatory, to detect this signal from the first stars. Now, there's a big challenge with this. Between us and the first stars is the entire universe, and it really gets in the way, I have to say. The signal that we're looking for is millions of times fainter than everything that's in between us. So, one of the early things that we needed to do with this telescope was make a new map of everything that's in the radio sky so that we could work towards this very, very difficult detection. And I'm not going to go into the millions of hours of supercomputing and the large group of people that we worked with to create this, but I do want to show you the results of this work. So back to our black and white image from the Haslam map earlier. I led the processing of a radio survey called the Galactic and Extragalactic All-Sky MWA survey, and this is the view of that survey. For the first time, we're seeing the radio sky in color. And these colors are real. This isn't a false color effect. What I've done here is I've colored the low radio frequencies red and the higher radio frequencies blue, and everything in between. And so all of the different colors in this image correspond to real astrophysical processes. So things that are red tend to have an older age. The cosmic electrons that produce the synchrotron radiation have started to age, and so we're seeing less high-frequency emission. Things that are blue tend to be thermal emission. They're brighter at higher frequencies. So while this survey is at relatively low resolution, all of the different colors in this survey allow us to do astrophysics. And of course, it's really beautiful. So this is, of course, just a little zoom of the survey. It's an all southern sky survey. So if we zoom out, I can say that we did successfully cover the entire southern sky and detect over 300,000 radio galaxies. So all of those are galaxies millions to billions of light years away. Even 13 billion light years away is so far our record. And again, getting that radio color information gives us astrophysical information across the whole sky. So I've put some of the data release papers at the bottom right there by myself and colleagues, and these have received hundreds of citations. We're nearing up to 1,000 now because of the work that this has enabled. And a brief interlude is that we've recently had the first images from the SKA. They've compared their data with the data that we took in this survey, and everything matches precisely. So this is really good news for the SKA and really good news for us. So GLEAM has been a tremendous success, and I was really delighted to lead that about 7 or 8 years ago. But there are some limitations to this image. It's at about the same resolution as the human eye, a couple of arc minutes, and that leads to a problem that we have called confusion. So in any given resolution element, there are millions of galaxies, very faint, that we can't see. The wider the resolution element, the more your noise is limited, your sensitivity is limited. That's why that black and white image that I showed at the beginning has a very low resolution. It also has a noise floor that we can't get past. So this survey is limited in its resolution. But in 2018, the MWA underwent an upgrade, and I saw an opportunity. We could use this upgraded telescope to double the resolution of a survey, and we could get past that confusion limit and observe more deeply. Now, I won an ARC Future Fellowship at the beginning of 2020 to progress this. It was a very tough time to be starting a new large survey that would require a lot of supercomputing. So it's only just in the last week that we have just had approval from the referees that the final data release from the galactic plane is now approved. So I wanted to show you the incredible results from this survey that I've led over the last few years. So if we zoom in back into the galaxy, I can show you that we have now reached the sensitivity that we wanted to. This is 10 times deeper than GLEAM. Everything is exquisitely calibrated. This is a supernova remnant, the Vela supernova remnant, the remains of an exploded star, which I've written several papers on these supernova remnants. Here we're seeing stellar nurseries, and again, all of the different colors are telling us about the astrophysics of all of these different regions. As we go along, we get to the center of our galaxy, Sagittarius A star, where there is a supermassive black hole, and I've written a paper on that interesting blue feature around that region. And to make this image took tens of millions of CPU hours, incredible work from a big team, and solving a lot of challenges like how to deal with the Earth's ionosphere, how to deconvolve the instrumental response from all of the different images, and how to make sure that the flux density scale, i.e. The brightness of all these colors, is correct. And it basically passes every single test. And even one of the third papers there demonstrates that we can use this data for cosmology. So imprinted in the image is the structure of the universe. So it's a big success story, and I'm really pleased to have done that. But that's not even the most amazing thing that we've done with this survey. So I mentioned early on we started in 2020 and we didn't have access to all of the supercomputing resources that we needed in order to create such a gorgeous image. So I came up with a fast technique. I used different observations that were taken at different times, subtracted the data from itself, and looked for things that could change. And I gave this to a very talented undergraduate, Tyrone O'Doherty. He was just doing his honours project with me. And I'm delighted to say that we found a new type of transient radio source, so something that nobody expected in the sky. Hopefully some of you are familiar with the concept of pulsars. These were discovered in 1967 by Jocelyn Bell. And a pulsar consists of a neutron star, which is the remains of a very massive exploded star. It has a very strong magnetic field that produces radio emission. The magnetic axis is offset from the rotational axis. So from our point of view, it appears to produce bright flashes of radio light. And the thing that drives that radio emission is the rapid rotation of the neutron star. We hadn't previously expected to find pulsars that could rotate more slowly than a few tens of seconds. But in our data, we found an object that was repeating every 18 minutes. And this was completely unprecedented. It also happened to be one of the brightest sources in the sky. So I was not expecting to find this, and we sort of rapidly scrambled to come up with theories as to what it could be. One of the theories that we have is that it could be a neutron star with an unusually strong magnetic field, i.e. a magnetar. And another theory that we're working on is that these could be types of white dwarfs, and perhaps they're interacting with a binary companion, and that's driving unusual radio emission. Either way, this was a discovery that no one expected, and so I was really pleased to do this in a sort of serendipitous way, just by looking at the data from a new angle. Yes. So I wanted to say we are now finding lots more of these, which is exciting. So brief technical graph. On the x-axis here is the period of all the objects that we know about. So on the left is that big group of pulsars. The y-axis is how quickly they spin down or reduce their period. And we expect objects to evolve sort of diagonally from the top left to the bottom, top right to the bottom left of this, top left to the bottom right, there we go. And that whole space on the right-hand side was completely unexplored before we started this work. And it was our discovery of the first source, and then very, very, very careful precision timing of a second source that we discovered, which has basically opened up this whole new parameter space to radio surveys. And we've also come out of our group, have been the first multi-wavelength associations, i.e. being able to see these things in X-ray and optical, which gives us information that we can then use to try and work out what they are. So the space now is very occupied. Telescopes across the world are changing their search methods and updating the way that they work in order to find these objects. And I was delighted this year to be invited to a big conference for the SKA to talk about how we might optimize searches for these objects with the SKA. So I wanted to close with a thank you to all of my colleagues, students, everyone who supported me along the way. It's been a real journey, and a special shout out to Professor James Miller-Jones, who encouraged me and nominated me for this award. Of course, all of the people, the institutions that made it possible, the ARC for funding me, MWA for taking the data, PAWSI for making the supercomputing possible. And just to close on a note that I was last year able to take my family up to the observatory, which is quite unusual. And you can see behind us there the very first constructed station of the SKA. And as some of the speakers have said, this work often takes a toll on our families and our family life. And so it was so beautiful to be able to take them up there and get them to see the telescope. And I hope one day, maybe they'll be the ones who use it. So thank you all very much for this award.
Well, thank you, Natasha. I think you'll all agree that was a work of art. Weren't those pictures beautiful? While you're thinking of a question, I would just ask, do you sometimes show them in 3D? Because it's a 2D picture, but it's really...
Yeah. So it is a bit tricky to show it in 2D, in 3D, because we don't actually know often how far away those sources are. I have done a lot of outreach, and I did make a quasi-parallax image where if you put on a 3D viewer, the Milky Way would appear to pop out and be in front of you, and all the distant radio galaxies would appear to be further away. But I didn't actually know their distances, I just faked it. I'm a big fan of art and science, so some of you may have spotted me wearing this fabulous scarf of the image.
So how much is the scarf now?
Oh, about $100, actually. Since the paper's just been accepted, we are actually going to open sales on the scarf, yeah. But the money we just cover costs.
And send it to the Academy.
Yeah.
Okay, a question or two, come on. I can see one just there, and I'll probably have to cut them off, don't I? So just one, you're the only one. Yeah.
So congratulations, Natasha, on your award, and thanks for a lovely talk. I'm just wondering if you can say very briefly how much what will the increase and improvement of resolution, in particular the new SKA low, will be relative to MWA, and how do you see that opening up the discovery space further in those science areas you've just been talking about?
Yeah, absolutely. So the difference between the longest baseline, which is the furthest separation of the two antennas, and that's what dictates our resolution. For the MWA currently, it is about 6 kilometres. For the SKA, it will be 60 kilometres. The resolution, therefore, will be an order of magnitude better, but the confusion noise will be 100 times lower because it goes down as the square. The other thing is, for the work that I'm doing now with these radio transients, is that the instantaneous sensitivity of the SKA will just be so many orders of magnitude greater. So we'll be able to see many faint things on a short time scale rather than having to do long integrations like we did to produce these radio surveys. And that's very exciting for probing the unknown, even the search for extraterrestrial intelligence. So I'm very excited to use the SKA.
Thank you very much.
Thank you
Congratulations.
so much.
Yeah. I'll just hand back to Stephen.
Thank you, Francis. Our next awardee is our 2025 Jacques Miller medalist. This medal is for experimental biomedicine and was established to honour the contributions made to science by Professor Jacques Miller, including the discovery of the function of the thymus and the identification in mammalian species of the two major subsets of lymphocytes and their functions. The award recognises research of the highest standing in the field of experimental biomedicine and is open to researchers 8 to 15 years post-PhD in the calendar year of nomination. This year, it's awarded to Professor James Hudson. Professor James Hudson is a rising star of Australian health and medical science who has already made a significant impact in translational medical science and looks set to continue to do so into the future. He's engaged in stem cell and organoid research, particularly in the heart, and it has a clear translational focus. Professor Hudson sits at the very top of the highest rank of the next generation of Australian medical researchers. Evidence for this assessment is manifold and includes such things as the quality and impact of his publications, recognition through awards and prizes and distinguished fellowships, and invitation to present at leading international meetings. Professor James Hudson, congratulations. Please join us on the stage to present, to receive the medal and present your talk. So congratulations. Thank you. Thank you very much. Congratulations, James.
Thanks everyone, and also to the Academy. It's an absolutely incredible honour to receive this award here today. And it's also very hard to go after all these last talks. They're incredible science, and I hope this can continue through the years. Today I'm going to talk mostly about heart failure. That's what our research program is focused on. There's around 64 million patients around the world with heart failure, and it's a very large healthcare and socio-economic burden, particularly in countries like Australia, and particularly for disadvantaged areas. It has a very poor prognosis, and more recently, a distinct form of heart failure has become more and more prevalent. So traditionally, the form of heart failure you might be well aware of is you have a heart attack, part of your heart muscle dies, and that never comes back. The cardiomyocytes you're born with, you die with, and your body can't replace those. And so you get a reduce in the contractile function of your heart, and that leads to heart failure. The other form is stiffening of the heart muscle wall, and what happens there is your heart doesn't relax properly, can't pump enough blood around, and this typically occurs in patients that are aged, but also with obesity and diabetes. And this form of heart failure, as you can see there on the graph on the right-hand side for you, is actually much more prevalent in especially older women. In terms of heart failure management, we don't have very many levers to pull. A lot of the current medications are all to do with lowering your blood pressure, changing your heart rate, changing your kidney function to get rid of the salts. And these things together decrease the load on the heart, but they don't necessarily address the underlying problem of the heart muscle and what has gone wrong in the heart muscle. And this is a big issue because these other drugs can affect other aspects of your life. Once you progress past these first stages of heart failure, there's really limited options, and the only real cure is a heart transplant. And so the goal of my lab is to really investigate these areas to provide new treatments. And we've done that in our lab through bioengineering approaches, and today I'm going to give you two examples. The first example will be for HFrEF. That's where part of your heart muscle has died and your heart's no longer contracting properly. When I was lucky enough to do my postdoc at the University of Medicine in Göttingen, that's in Germany, I worked with Professor Wolfram Zimmermann and Malte Tiburci, and together we developed protocols to turn human pluripotent stem cells into heart muscle cells. Now, this builds on about 20-25 years of developmental biology that people had done in order to instruct those cells to become heart muscle cells. We developed these protocols. We also developed protocols to incorporate fibroblasts. Your heart is actually only 30% heart muscle cells. The rest is fibroblasts, vascular cells, other support cells like macrophages. And we really need the fibroblasts to make a tissue. Without them, you don't get a tissue at all. We came up with protocols to make patches, about 50 million cells per patch. And these, over a 10-year period, got translated to the clinic. So phase 1-2 trials have been completed in Germany with 17 patients dosed. The highest dose is 800 million cells per heart, that's 16 patches, and those patients have now lived over 3 years with their heart patches without requiring a transplant. They've now applied to the German healthcare system for a patient reimbursement system so that anyone in Germany with heart failure can get one of these. In my lab here in Australia, together with colleagues at MCRI, Enzo Perello, funded by Renew, we've developed the next generation of these heart patches. And in this system, what we've done is we've optimised that surgical handling so we don't need 16 patches. We've optimised it to have vascular cells and hypoxia resistance, so what we put in survives and integrates a lot better. And we think this is going to be a transformational approach to further improve this. You can see this here in a transplantation study we did in a mouse heart after a myocardial infarction, where you get these large vessels forming through the tissue. This has never been shown in a heart patch before. And then also 3 months after transplant in a pig heart. You can see in this immunosection of a heart muscle, where the heart muscle cells are white, you see the implanted graft on the outside of the heart, and we have elongated and striated cardiomyocytes in there as well. And then the real tell is doing a large animal study. These are very hard, long, expensive studies. Together with Dan Donner at the Baker, he gave pigs heart attacks and then waited 7 days, and so you already get a big decrease in heart function. Implanted the patches, and then we took them out to 90 days. Now, all the previous studies done by Wolfram and other colleagues are done in monkeys, where they're around 11 kilos. These are in 90 kilo pigs. And what you can see here is the MI only. We're at the top region of the heart there. This is a cross-section. You have a lot of damage to the heart dilates and it doesn't contract and push out enough blood. And you see with the ones with the patch, fully recover the heart function. We never expected this, and this is really a transformational finding. We're about to start a company and transfer that to the clinic in the next 3 years. The next story I want to tell you about is some of the work we've been doing more recently on HFpEF. For this, we use a different system. In my lab over the last 10 years, we've developed protocols to make human cardiac organoids. We make the various cell populations in the heart in a single mix, and these have progenitor cells. We form these 96-well plates that have these inserts, and you can see that with the 2 elastic pillars. And my lab's really well known for maturation protocols to make these really similar to the human heart for drug discovery. And so we can measure the force curves, as you can see in the video. And not only can we get the force of contraction, but we can get the kinetics. Now, remember when I said at the start, stiffening is a key issue with heart failure, with the HFpEF. Well, here, if you get an increase in that TR50, that's when you get this stiffening and diastolic dysfunction, and it mimics that same phenotype. And so one of the things we've been doing is these large-scale perturbation library screens to create a heart in a computer. To do that, we're doing functional phenotyping with those contraction curves, and also RNA sequencing from every single perturbation to try and create a full map of how the heart works. I don't have time to go through all our different approaches here. Today I'm going to focus on the ligand screen that we've been working on. In this pipeline, we mature the cardiomyocyte and the whole organoid system for the previous protocol I showed you. We then take a baseline video, we stimulate them with the ligand, and then we do an acute and a longer term recording, and then an RNA-seq. And we can correlate each individual organoid with its RNA sequencing profile, which gives us a lot of information about not only the function, but also the biology that's happening in that organoid at that time. And I'll show you how that's very transformational. In terms of what we're going to hit these tissues with, we downloaded the databases, and there's around 1,300 receptors expressed in the human body. About half of those are expressed in the heart, and our tissues, our organoid tissues, have a very good coverage of what's actually expressed in the human heart itself. When we narrow it down, we mostly focus here on soluble ligands, so these are ones that signal throughout the body, as we can just directly put a ligand on and test their function. This is a large, complicated functional map. The important thing is just to note what's in the 2 boxes here. So the top is force and rate, and these parameters increase when we give things that activate the adrenaline system. And all of these cluster together. Not only do they activate the force and rate, but they quicken the contraction curve. What we found in this study is in sort of the middle patch there for the ones I've labelled stiffening, these are all the kinetic parameters, and they actually stiffen quite a bit with a lot of the inflammatory factors and a lot of the factors that come from endothelial cells. When we look at the RNA-seq profile, and this is, each dot is an individual organoid treated with a different ligand. This is a representation on similar to a principal component analysis, where how distant the different dots are represent how different they are between each other. And you can see right around the edge are all the inflammatory stimuli, and the endothelins cluster as well, which is the endothelial stimuli. All the inotropes as a positive control there also cluster together. So we wanted to know what was really distinct about these. We got human heart failure biopsies, which were deposited in the literature, and we could access those and look at their signature. And we used our data to fingerprint these and determine which signalling pathways were perturbed. And this was not really known for HFpEF. And what I can show you here is that interferon gamma can almost completely predict the segregation of healthy controls versus human HFpEF. We did a lot of profiling studies to determine how interferon gamma and some of the other cytokines perturb the signaling, and then did a drug screen and found that bromodomain inhibitors can correct this dysfunction. We've now screened various drugs, we've shown the transcriptional response, and we've defined a really unique bromodomain subset that can prevent this response, and we have a MedChem Australia funded project to turn these into drugs for people now. So I hope that provides a snapshot of our work. I'm really privileged to receive this award, and I really have a lot of our lab members to thank, particularly Janice for those ligand studies I just showed you at the end, Andrew for his work on the patch, previously Richard, Simon and Holly, who are now their own group leaders, for a lot of setting up the model, and also extensive collaborative networks, including a close partnership with Enzo over many years. So thank you.
Thank you, James. A remarkable story. And testament to the support of philanthropy. I think that's a theme that's run throughout the meeting in Canberra this week, the SATS meeting, the significance of substantial philanthropy to support Australian science, and certainly Snow have been a supporter of James's work, so that's really wonderful. Jane, question? Yeah, have we got a...
Beautiful talk, James. You mentioned the importance of fibroblasts, but these are one of the most plastic cell populations. So I was wondering, how do you get around that? Have you seen any evidence of these fibroblasts changing in the heart patches or the organoids?
Yeah, that's an excellent question. I think equally the endothelial cells are very plastic as well. That's what we find. And in the organoids, we've co-mapped them with human heart, single nuclear sequencing data we have, and they overlap quite a bit, but there are some differences and there are some distinct populations. One of the most interesting things we found in that ligand screen is a lot of pro-fibrotic factors co-clustered together, like PDGF, TGF-beta, thrombin, things like that. But they actually activate very different responses if you look at the individual genes. And so I think a lot of this fibroblast heterogeneity can probably be explained by the ligands that they're exposed to in vivo, and whether they're sitting next to an endothelial cell that secretes PDGF or a macrophage that expresses IL-1B, you're going to get very different signatures.
Any other questions? Yes, of course. We can't have our leading cardiologist not ask a question. Over to you, Bob.
Thanks, James. Great work. Recently, there's been a theory that the problems with HFpEF is that you've got an inflammatory adipocyte problem with altered adipokine secretion. Do you see adipokine altered in your screens?
We've done another screen that's focused more on the adipokines and also the nuclear hormone receptors, and we haven't analysed all that data yet, but that'll be very interesting to answer that question, because I agree. But I think there's also distinct subsets of HFpEF, and there's perhaps a obese adipokine-driven one, there's an ageing, inflammatory cytokine-driven one. We see in some of the human data sets we're working with collaborators in Germany, we see T cell infiltration, and in our mouse model, if we take out the T cells, we can help restore the function. So I think it's going to be dependent on the exact features in that patient that cause the disease.
Right, well, I think a question at the back, last one.
This is way outside my area, but I was just thinking, when you make the patches, do you actually take the starting cells from the patient? Because otherwise, I guess you're going to get a kind of transplantation problem.
Yeah, that's a great question. So the economic expense to do the GMP QC is too high to do it from an individual patient, and it would take too long. and so they have to be done from an allogeneic line. We can match those lines to the population by really carefully choosing the lines. So, for example, the line we're using for GMP transfer will cover about 40% of the Australian Caucasian population. Obviously, you know, there's a diminishing return as you build up the cell lines, so you have to be careful in that. The other option is, and what some people are doing overseas, is what they call cloaking the cells. So they'll either knock out the NHC proteins or they'll overexpress anti-inflammatory proteins to make an immune barrier to either decrease or remove the need for immunosuppression.
Fantastic. Well, on that note, I think we'll say thank you and congratulations again. And that now concludes this session. And before we move on to our awards ceremony for 2025, we're going to take a break for afternoon tea. That will be, we'll stay until 3:30. So we come back at 3:30, and all our 2025 awardees are asked to move directly to the main foyer for your group photo. Everyone else, please enjoy afternoon tea in the Jaeger room, Dorothy Hill room and outside, and see you back here at 3:30.
I went to medical school like many Asians at the behest of my parents, but when I was at high school, I had very inspiring conversation with a very famous scientist called Max Perutz. He was one of the people who solved the first ever structure of a protein called hemoglobin. That conversation when I was 16, 17 really made me think I could go to medical school but have other kinds of careers beyond just being a doctor. My name is David Wang. I'm a scientist here at the Water and Lyle Institute of Medical Research, or WELI. I'm really used to study differentiation of T cells. What I do every day is to understand why a normal cell in our body becomes a cancer cell, and essentially to try to identify characteristics or features of a cancer cell that we could target for therapy. All of us in our bodies, there are cells past their use-by date. In all of us, billions of cells are removed by a mechanism that is amazing, works silently. None of us are aware of it happening. But if it goes wrong, it is catastrophic because those cancer cells that no longer die when they should die hang around. That can be really nasty. Our real interest is in this molecule called BCL2. It makes cells survive when they shouldn't be surviving, when it's overactive. And we realized early on that there was a groove on it that was critical for how BCL2 functioned. And that then led on to us thinking about how we could target that groove. The most advanced of those is the drug called venetoclax, and that drug works beautifully in blood cancers by targeting BCL2. Within a few hours of the first handful of patients treated, we could see that the leukemic cells were responding, and that was amazing to see that what you could see in the test tube or in the laboratory dish, what you predicted was actually happening in the patient. It is an amazing honor to be elected by your peers, by your colleagues, as a fellow of the Australian Academy of Science. I think it carries a certain degree of responsibility to speak up for science, for speak up for basic research. By nature, I like gossiping, so I love talking to colleagues, and more with scientific research, I think to me is just about sharing ideas. I went to medical school like many Asians at the behest of my parents, but when I was at high school, I had very inspiring conversation with a very famous scientist called Max Perutz. He was one of the people who solved the first ever structure of a protein called hemoglobin. That conversation when I was 16, 17 really made me think I could go to medical school but have other kinds of careers beyond just being a doctor. My name is David Wang. I'm a scientist here at the Water and Lysol Institute of Medical Research, or WELI. I'm really used to study differentiation of T cells. What I do every day is to understand why a normal cell in our body becomes a cancer cell, and essentially to try to identify characteristics or features of a cancer cell that we could target for therapy. All of us in our bodies, there are cells past their use-by date. In all of us, billions of cells are removed by a mechanism that is amazing, works silently. None of us are aware of it happening. But if it goes wrong, it is catastrophic, because those cancer cells that no longer die when they should die hang around. That can be really nasty. Our real interest is in this molecule called BCL2. It makes cells survive when they shouldn't be surviving, when it's overactive. And we realized early on that there was a groove on it that was critical for how BCL2 functioned. And that then led on to us thinking about how we could target that groove. The most advanced of those is the drug called venetoclax, and that drug works beautifully in blood cancers by targeting BCL2. Within a few hours of the first handful of patients treated, we could see that the leukemic cells were responding, and that was amazing to see that what you could see in the test tube or in the laboratory dish, what you predicted was actually happening in the patient. It is an amazing honor to be elected by your peers, by your colleagues, as a fellow of the Australian Academy of Science. I think it carries a certain degree of responsibility to speak up for science, for speak up for basic research. By nature, I like gossiping, so I love talking to colleagues, and more with scientific research. I think to me is just about sharing ideas. I went to medical school like many Asians at the behest of my parents, but when I was at high school, I had very inspiring conversation with a very famous scientist called Max Perutz. He was one of the people who solved the first ever structure of a protein called hemoglobin. That conversation when I was 16, 17 really made me think I could go to medical school but have other kinds of careers beyond just being a doctor. My name is David Wang. I'm a scientist here at the Water and Lysol Institute of Medical Research, or WELI. I'm really used to study differentiation of T cells. What I do every day is to understand why a normal cell in our body becomes a cancer cell, and essentially to try to identify characteristics or features of a cancer cell that we could target for therapy. All of us in our bodies, there are cells past their use-by date. In all of us, billions of cells are removed by a mechanism that is amazing, works silently. None of us are aware of it happening. But if it goes wrong, it is catastrophic because those cancer cells that no longer die when they should die hang around. That can be really nasty. Our real interest is in this molecule called BCL2. It makes cells survive when they shouldn't be surviving, when it's overactive. And we realized early on that there was a groove on it that was critical for how BCL2 functioned. And that then led on to us thinking about how we could target that groove. The most advanced of those is the drug called venetoclax, and that drug works beautifully in blood cancers by targeting BCL2. Within a few hours of the first handful of patients treated, we could see that the leukemic cells were responding, and that was amazing to see that what you could see in the test tube or in the laboratory dish, what you predicted was actually happening in the patient. It is an amazing honor to be elected by your peers, by your colleagues, as a fellow of the Australian Academy of Science. I think it carries a certain degree of responsibility to speak up for science, for speak up for basic research. By nature, I like gossiping, so I love talking to colleagues, and more with scientific research, I think to me is just about sharing ideas. I went to medical school like many Asians at the behest of my parents, but when I was at high school, I had very inspiring conversation with a very famous scientist called Max Perutz. He was one of the people who solved the first ever structure of a protein called hemoglobin. That conversation when I was 16, 17 really made me think I could go to medical school but have other kinds of careers beyond just being a doctor. My name is David Wang. I'm a scientist here at the Water and Lysol Institute of Medical Research, or WELI. I'm really used to study differentiation of T cells. What I do every day is to understand why a normal cell in our body becomes a cancer cell, and essentially to try to identify characteristics or features of a cancer cell that we could target for therapy. All of us in our bodies, there are cells past their use-by date. In all of us, billions of cells are removed by a mechanism that is amazing, works silently. None of us are aware of it happening. But if it goes wrong, it is catastrophic because those cancer cells that no longer die when they should die hang around. That can be really nasty. Our real interest is in this molecule called BCL2. It makes cells survive when they shouldn't be surviving, when it's overactive. And we realized early on that there was a groove on it that was critical for how BCL2 functioned. And that then led on to us thinking about how we could target that groove. The most advanced of those is the drug called venetoclax, and that drug works beautifully in blood cancers by targeting BCL2. Within a few hours of the first handful of patients treated, we could see that the leukemic cells were responding, and that was amazing to see that what you could see in the test tube or in the laboratory dish, what you predicted was actually happening in the patient. It is an amazing honor to be elected by your peers, by your colleagues, as a fellow of the Australian Academy of Science. I think it carries a certain degree of responsibility to speak up for science, for speak up for basic research. By nature, I like gossiping, so I love talking to colleagues, and more with scientific research, I think to me is just about sharing ideas. I went to medical school like many Asians at the behest of my parents, but when I was at high school, I had very inspiring conversation with a very famous scientist called Max Perutz. He was one of the people who solved the first ever structure of a protein called hemoglobin. That conversation when I was 16, 17 really made me think I could go to medical school but have other kinds of careers beyond just being a doctor. My name is David Wang. I'm a scientist here at the Water and Lysol Institute of Medical Research, or WELI. I'm really used to study differentiation of T cells. What I do every day is to understand why a normal cell in our body becomes a cancer cell, and essentially to try to identify characteristics or features of a cancer cell that we could target for therapy. All of us in our bodies, there are cells past their use-by date. In all of us, billions of cells are removed by a mechanism that is amazing, works silently. None of us are aware of it happening. But if it goes wrong, it is catastrophic because those cancer cells that no longer die when they should die hang around. That can be really nasty. Our real interest is in this molecule called BCL2. It makes cells survive when they shouldn't be surviving, when it's overactive. And we realized early on that there was a groove on it that was critical for how BCL2 functioned. And that then led on to us thinking about how we could target that groove. The most advanced of those is the drug called venetoclax, and that drug works beautifully in blood cancers by targeting BCL2. Within a few hours of the first handful of patients treated, we could see that the leukemic cells were responding, and that was amazing to see that what you could see in the test tube or in the laboratory dish, what you predicted was actually happening in the patient. It is an amazing honor to be elected by your peers, by your colleagues, as a fellow of the Australian Academy of Science. I think it carries a certain degree of responsibility to speak up for science, for speak up for basic research. By nature, I like gossiping, so I love talking to colleagues, and more with scientific research. I think to me it's just about sharing ideas.
The fact that we're increasing our greenhouse gas emissions, even though we're seeing clear evidence of climate change and very, very significant impacts from those climate changes, does result in a degree of frustration in me and other scientists, because we've actually had enough science to make clear decisions and good decisions for at least 20 years now. I'm Emeritus Professor Mark Howden. I'm affiliated with ANU and also a Vice Chair of the IPCC. So I've had a long engagement with the IPCC going back to 1990, and so I've seen it evolve. It's staffed by a small secretariat, but then most of the work is actually done by the science community across the globe. They're the people who scope the assessments, they write the chapters, they review the chapters, and in fact, I often characterise it as the biggest single science policy activity in the history of humankind. So the science can provide information into the consequences of different, for example, global temperatures, but it's governments who actually have to take in that information and then come up with some agreed global goals.
The eyes of the world have been on Paris for weeks.
The Paris Agreement, in setting its goals, was fundamentally informed by the science and particularly by the IPCC, and so that's, in a sense, a big tick for the science community. However, since then, what we've seen is instead of greenhouse gas emissions starting to flatten out and then go down, what we've actually seen is those emissions continue to go up. If we continue on our pathway in terms of emissions, we're likely to suffer much more from climate extremes. That's going to cause significant consequences in terms of pretty much every sector that we have. So disruption to food supplies and food securities, to water supply, to energy supply, etc. And economic analyses show that that could actually be a huge hit to GDP. So something in the region of 14% reduction in GDP per degree Celsius. I think it is important for scientists to respond to those people who are ignoring the evidence or have a denialist approach to this. That group is actually very small. It's probably about 7% across the nation, even though most people think it's much bigger. It tends to be quite a noisy group who tend to be overrepresented in public debates. The other important part is actually being faithful to the science itself and being able to say very clearly what the science can and can't say about this particular complex topic and what we might need to do to actually have more clear answers. ANU has been a really great place to put in action some of the ideas that come from the science community. One of my really high moments in ANU was where I formed the Institute for Climate, Energy and Disaster Solutions. That is a unique institution right across the globe, bringing those different and interconnected aspects of climate change together. Being elected to the Australian Academy of Science is a highlight of my career, particularly in terms of recognition of the different types of science that I've engaged in and how we've actually changed the world through that science.
Hey, can you see it?
See what?
The wonderful possible. Like overcoming language, distance and economic barriers to become the first nursing graduate in your community ever.
I love this.
Quite a story, huh?
What about a PhD at 80?
Still got it.
Pop-up law clinics anyone can access?
Nice.
Or lifting others up through education.
Yeah, I see it now.
Yeah, you do. Deakin University. See the wonderful possible.
We have some great therapies for very serious disease, but there's still a lot of areas where patients are really poorly treated, and the opportunity to do something about that, is a really rewarding career path. My name's Andrew Nash. Up until recently, for the last 18 to 20 years, in fact, I've been the global head of research for CSL. For the last 5 years, I've been CSL's chief scientific officer. Universities and medical research institutes are really set up for the most part to do basic science and to do some of that translational work. They are not really set up for that drug development process, which can take a very long time and is very expensive. So a large part of what I've done over the last 20 or 30 years is to collaborate very closely with scientists in medical research institutes to work with them on translating their work and the work that we've been doing in CSL into something that's going to benefit patients.
Any problems, any challenges? Everything's good.
The model that's most often pursued is small startup ventures, which can really focus on a single unique problem, get the funding and move that along to a point where they've taken some of the risk out of it and where big companies can either then come in and partner or participate in the further development of that drug. One of the things that we realised that was missing here in the Parkville precinct was a really high quality wet lab incubator. So a couple of years ago now, we partnered with WEHI and the University of Melbourne to establish Jumar Bioincubator. That's for small companies that are just starting on their journey. It's been very successful over the last 2 years. I think there's 30 to 40 small companies in that incubator at the moment, and I think that's filled a real gap. It's really about positioning yourself as a company to access innovation.
And did we need to come up with some new antibodies specifically for the models?
A lot of us who work in this area for a long time, you're full of doubts. Am I working on the right thing? Is my research ultimately really going to matter? Am I really helping do the things that I want to do? And to be acknowledged through a fellowship of the Academy, it goes at least some way towards, making you feel that the work you've been doing has made a difference. We have some great therapies for very serious disease, but there's still a lot of areas where patients are really poorly treated, and the opportunity
should make a point to start on time, right? Because we get 3 minutes. Yeah.
I'll just tell Stein where he's going to walk since he's first on.
Yeah. Shall we make a start?
Yeah, I'll do it.
Okay, welcome back everyone. We are now here to celebrate our awards ceremony for 2025. We will hear about each awardee's work and they will join Professor Jagadish on stage to be presented with their medals. Before we do, I would like to let you know that in 2026, we are pleased to be announcing the first recipient of a new award, that is the Anderson Medal for ICT. I would like to sincerely thank Brian and Diane Anderson for their commitment and support to creating this new award, and we look forward to the Anderson Medal awardee joining this exceptional cohort next year. We will start with the 2025 Anton Hales Medal, which recognises distinguished research in the earth sciences and honours the contributions to the earth sciences by the late Professor Anton Hales. Professor Hales was the founding director of the Research School of Earth Sciences at the ANU. I particularly want to acknowledge and thank Denise Hales, Anton's wife, who joins us today, for our ongoing support of this award. Thank you, Denise. And congratulations to this year's recipient, Associate Professor Stein Glory.
Congratulations, Stein.
Maybe think of it like this. Rocks are books, and what I'm trying to do is try to build a language so we can read those books, basically. And by continually accessing more of those books, we can work out how Earth has behaved over deep time. My name is Stein Glory, and I'm an earth scientist at the University of Adelaide. So my research is largely focused on understanding time, all the way from the initial formation of the solar system all the way until, let's say, the last couple of million years. So particularly understanding the rate and timing of plate reconstructions, when the plates move from one way to the other, how and when did mountain ranges form, and particularly understanding how timing can be used to help find the mineral resources that we use in our daily lives. Have you already checked how much lutetium or rubidium is inside those minerals? We need to use new technology, including thermochronology and new in situ dating methods. So lutetium stays behind. By doing that, we've opened up a whole range of new opportunities, because all the time that we could previously access was based on a very finite number of mineral systems. But now with these newer systems, we can access a whole range of minerals, which I would say are higher abundant in most rock types as well. So we get a better non-biased signal, if you like. towards how time progressed and how certain processes evolved in geological history.
Now, a new analysis shows that the 6-ton iconic altar stone at the heart of Stonehenge originated from northeastern Scotland rather than southwest Wales.
People have looked at the history of Stonehenge for decades, I'd say. And it's not until now, with those new techniques, that we can actually bring some new information to the puzzle. So I really like the idea that you can sometimes, by developing new methods, new ideas, really make breakthrough discoveries in science. But they're very tiny. And I moved from Belgium to Australia about 12 years ago. It gave me lots of new perspectives, particularly because in Belgium, the whole country is made up of largely soft rock. While here in Australia, at least if you go to the west from Adelaide, everything is almost hard rock. And the methods that I'm using here are catered towards accessing that deep time history. And also, I guess, the relevance towards mineral exploration is much more accurate here as well, because there's many mineral commodities here that are used to derive the energy sector, while back in Belgium, that's less so. So the move has certainly helped my career, and I feel privileged to now receive the Anton Hales Medal.
Next up is the Christopher Heide Medal. This award honours contributions to mathematics by the late Professor Christopher Heide. In recognition of Professor Heide's broad interests in mathematical sciences, the award is offered in one of 3 fields of mathematics on a rotating basis. We are joined today by Beth Heide, Christopher's wife, and I would like to thank you for your ongoing support of this award. So congratulations to the 2025 recipient, Associate Professor Anita Liebinau.
Congratulations, Anita.
Hi. What I really like about mathematics is that it's pure. You have an argument, and that argument is either correct or it's not correct. It doesn't matter whether you are, where you're from and what's your background. It just matters if you have a correct proof of something. I am Anita Liebinau, and I work at UNSW Sydney. I work in mathematics, and more specifically in combinatorics and graph theory. Combinatorics is dealing with counting discrete structures. So one example, imagine a network, like you might think of a social network. So you have lots of different people and they're linked, they may know each other, and then we can model those networks as graphs. So these are just a selection of nodes, and some of these nodes are linked. With a social network like Facebook, you could have a lot of people all over the world. In some networks we study, there could be tens of thousands or even billions of points. So what you get is a random network, and what I do is think about what are the properties of these random networks on a purely theoretical level.
The complete graph of n vertices has either a red copy of KS or a blue copy of KT. And so there definitely exists a red-blue coloring without any of this.
At the board, you're just standing there and it's just you and pure thinking.
Right.
But you need to sort of explain this to each other, and you want to explore various pathways and approaches. You just don't give up until you find some solutions.
An N here and vertices.
In order to prove something, it just takes many days, months, years sometimes. Some of my current work deals with problems in Ramsey theory. Ramsey's theorem says that you will always find 3 people that either mutually know each other or that mutually do not know each other. So go to any party, there will be either 3 people who are friends with each other or 3 who are strangers with each other. And it's often paraphrased by there cannot be complete chaos. Our work has improved comprehension of the relationship between graph structures and their Ramsey properties. And in particular, how complex structures can be guaranteed to appear under various conditions. I come from a non-academic family, so even just studying math and having the thought of staying in university was out of reach for me for a long time. So really receiving this medal from the Academy, this is amazing.
Next up is the Dorothy Hill Medal, which honours the contributions of the late Professor Dorothy Hill to Australian earth science and her work in opening tertiary science education to women. Please welcome the 2025 recipient of the Dorothy Hill Medal, Dr. Linda Armbrecht.
Congratulations, Linda.
Being in Antarctica is a very unique experience. I can't believe that I'm here, to be honest. Nothing I would have ever thought I would do when I was younger, and only a limited number of people see it, and I'm really happy that I'm one of those.
My name is Linda Armbrecht and I work at the Institute for Marine and Antarctic Studies at the University of Tasmania in Hobart. I'm a marine biologist, specifically paleogeneticist, which sounds really complicated, but I basically investigate ancient DNA in the seafloor and look at what organisms have been living in the ocean in the past. In Antarctica, we've been able to get DNA up to 1 million years old. It's really important to look at marine sediment records to get a good picture of how the Earth has changed through time. We were drilling here at the ice stream, and we can make predictions using that information about how the ecosystem will change into the future with ongoing climate change.
First part of the process of what we'll be doing in the field here is we drill a really long hole through the ice shelf to be able to get to the sediment, and then we drill a sediment core. We have a container that you can see set up in the background here, and we establish this sterile environment where we can do a clean sampling. Yep, that's pretty good. So I'll just take the picture and then show you on the computer.
The aim of this expedition is to look at how sensitive the West Antarctic ice sheet specifically was to warming in the past, and we want to now, when we cross 2 degrees of global warming, how does the West Antarctic ice sheet respond? When we're going to Antarctica and we're seeing the continent and how everything changes there, it is quite concerning. I mean, the habitat is changing and also the environment is changing. It just really makes you want to preserve and protect the continent. I was surprised that my colleagues actually considered nominating me for the medal, and now that it's been awarded, I feel a little proud, and I think it's an amazing recognition of the work and the research that I've done. And lucky that I get to follow in the footsteps of Dorothy Hill.
So next is the FAHMS Medal, which is awarded in honour of the outstanding contributions to science by the late Professor Frank Fenner. It recognises distinguished research in biology. The joint recipients, the committee couldn't separate these two, they were so good together, for the 2025 FAHMS Medal are Professors Catherine Mosby and Associate Professor Daniel Noble. Please first welcome Catherine to the stage.
Congratulations, Catherine.
I just love desert ecosystems. Love the open skies, the big starry nights, the lack of people. I just love walking around out there and seeing new things and trying to understand what's going on. My name is Catherine Mosby. I usually tell people that I'm a conservation biologist, so I'm trying to work out ways to stop threatened species from going extinct. We have a lot of things that we're monitoring, the species themselves, but also the environment, the soils, the invertebrates, the plants, the birds, the reptiles, all those sort of things where we're just looking at how they all interact and trying to understand that ecosystem so that we can then take those learnings and apply them on a much broader scale. I co-founded Arid Recovery, that's a safe haven, and then we've also got Mallee Refuge at Secret Rocks, and then we've got Wild Deserts as well in Sturt National Park in New South Wales. So they're basically fences that we designed to exclude cats and foxes effectively. They've got electric wires and floppy overhangs. It's quite a neat little fence design. We started Arid Recovery almost 30 years ago now, so it was one of the first fence reserves to go up. And I think everyone was just so shocked that if you could get rid of cats and foxes, how quickly you could reintroduce animals that used to be there and how they would survive and increase and how much the ecosystem changed when you did that. We reintroduced species like bilbies. They dig for a lot of their food, and those foraging pits trap seeds, increases nitrogen and carbon in the soil, leads to increased germination. So it has lots of flow-on effects to other parts of the ecosystem. I'm honoured to receive the Fenner Medal. I look at the other people that received that medal and I feel quite humble. Although I'm the one receiving this medal, all the work that I do is definitely a team, and you can't do field ecology or Arizona ecology on your own. So I just wanted to say thank you to all those staff and volunteers and interns and colleagues that I've worked with, because, you know, I think we've achieved a lot of stuff together.
And now please welcome Professor Daniel Noble.
Congratulations, Daniel.
Science is wonderful and amazing. You get to learn so much. You get to explore the natural world, build your curiosity, and I think it's the most amazing thing on the planet. My name is Daniel Noble. I'm an associate professor here at the Australian National University. I'm interested in understanding how environmental change affects how an organism functions and what that means for how they survive and reproduce.
All right, let's head in and see what we can find.
Our main model systems are lizards.
Go for it, Naomi.
Go. And the reason why is because they're very amenable to experimental manipulation.
So which number? This is 1122.
It's very easy to change the environment, both the parent's environment and the offspring's environment independently, but they are very similar to a lot of other organisms. We can compare some of the responses we see to pretty much every other animal that has a body temperature that really depends on the environment. So one of the things that we've been exploring is looking at how temperature affects your ability to tolerate heat. We've been doing this by manipulating the eggs early in development and looking at if they are able to cope more effectively with heat stress later in life. And from our experiments, those early experiences are not really going to help you cope with heat stress over the long term. Aside from our own observations, our team also conducts what we call research synthesis and meta-analysis. So every single experiment that we do needs to be replicated, and so what we're doing is we're relying on researchers from across the globe to do these kinds of experiments. And that's really important because we expect there to be a lot of variation, but what we don't really fully understand is why such variation exists and what might be the key factors that explain some of that variation. I've always been interested in how developmental environments are impacting how you behave. We see it in humans, and the same thing is true of any kind of animal, actually. So the kind of thermal environments, the social environments you experience early in development do set you up for life. That's what's sort of drawn me into this space. Receiving the Fenner Medal is recognition of the huge amount of work that my colleagues have been doing in this space and is encouragement to keep doing it.
Next is the Gottschalk Medal. This award recognises outstanding research in medical science and honours the contributions to science of the late Professor Alfred Gottschalk. Again, we have joint recipients. The recipients for the 2025 Gottschalk Medal are Associate Professor Amy Kane and Associate Professor Shom Gol. First, let me welcome Amy.
Congratulations, Amy.
Antibiotic resistance is one of the greatest threats to humankind. So by 2050, we'll be seeing around 10 million deaths per year due to antibiotic resistance. My name's Associate Professor Amy Kane, and I work at Macquarie University. Bacteria are really clever. They can pass antibiotic resistance genes between each other, and this results in a global spread of resistance genes. As soon as one antibiotic resistance is seen somewhere in the world, it will spread globally. About half of the antibiotic resistance-related deaths are caused by only 7 bacterial species. These are called the escape pathogens, and this is because they escape the action of the antibiotic. Some more experimentation. My group uses functional genomics techniques to study these escape pathogens. That's the most important part, right there. We ask how we can develop new antibiotics and new strategies to treat these really deadly pathogens in an effective way. But I also established and run the Galleria Research Facility here at Macquarie. These are the Galleria melanella wax moth larvae, and they're a really useful, ethical, and effective animal model alternative to, say, mice. This means that we can easily take drugs that have really promising data in the lab, what we call in vitro data, and get it into a body, what we call in vivo studies. And these are really the very first step to clinical trials and getting drugs closer to patient use. I used to work in Malawi in sub-Saharan Africa in a big public referral hospital for the Wellcome Trust. I set up a research program that was tracking hospital-acquired infections that are usually caused by these really nasty escape pathogens, and that program is still ongoing. Being on the ground in the hospital and looking at the infection control techniques and actually what's implemented in the hospital is a really important step for scientists to understand what the impact of their lab data has on the real world and on patients. I'm absolutely floored to be awarded the Gottschalk Medal. A lot of my scientific idols have gotten this medal, and I've seen them do great things with science, and I just humbly hope that I can also provide some big steps forward in my field of antibiotic discovery and drug development.
And now please welcome Professor Shom Gol.
Congratulations, Shom.
The idea that the research our team is doing in the lab could impact the lives of thousands of women and their families around the world is really the biggest motivator for me. My name is Shom Gol. I'm a medical oncologist. But in addition, I'm also a research scientist here at the University of Melbourne and the Peter MacCallum Cancer Centre. The focus of my research is on breast cancer, a very common disease that affects the lives of thousands of Australian women every year. The thing that I'm really interested in focusing on, working as a scientist, I think enables me to take better care of my patients as well, because it gives me a different perspective on their disease and the way it's behaving. In our lab, we specifically study a process known as the cell cycle. Cancer is a disease where cells are dividing too much. So our goal is always to try and kill cancer cells. But what we've come to realize more and more is that many of our treatments don't actually kill cancer cells, but rather just put them into this state of arrest. Some people call these cells sleeping cells. In the lab, we use the term cellular senescence. And some of the cells that don't have RB, is that right? Although senescent cancer cells are no longer dividing, they're still biologically very active. And we've focused a lot of our attention on trying to understand how these cells are different from normal cancer cells, and therefore to try and understand how we can target them. We've identified new vulnerabilities that these cells have, and that's allowed us to develop new drug combinations to treat certain types of breast cancer, through which we combine one drug that will help keep these cells asleep and a second drug which will target these new vulnerabilities. And in fact, one of the most gratifying things that's come from my team recently was the results of a large clinical trial where we found that the combination of treatments that we discovered in the lab is actually helping people live a lot longer. I think the part of me that has become a scientist is the part of me that loves to solve problems, that loves to identify what the core issue is that we've yet to understand, and then logically work through a process to solve that problem. And then thinking that you're able to apply your knowledge, your training, and your energies to finding treatments and to help the patients of the future. It's an honor to be able to do that, to be honest, and very motivating.
Next is the John Booker Medal in Engineering Science, which recognises outstanding research in engineering mechanics that addresses problems in the static and dynamic response of physical systems within engineering and applied mathematics disciplines. It honours the memory of Professor John Robert Booker, who worked in the area of theoretical geomechanics, was Professor in the Department of Civil and Mining Engineering at the University of Sydney, and held a personal chair in engineering mechanics at the University of Sydney. I'm pleased to invite the 2025 recipient of the John Booker Medal to the stage, Associate Professor Chen-Bing Zhang.
Congratulations, Chen.
Think about an earthquake or explosion. So those two extreme conditions will induce catastrophic failure in very short period. So it's important to understand those failures. My name is Chen-Bing Zhang. I'm current associate professor at Monash University. For my research, we focus on how geomaterials and structures fail. The sharing test. We do that by simulating catastrophic events. It's quite challenging because we cannot see them very in normal speed. So we have to use ultra high speed cameras and also using the facility we call Hopkinson bar. So that means we can see how materials are fracturing in a very slow motion and just in microsecond. This facility actually is a world first. We started building the facility since 2015.
The axial direction.
And building this facility is quite a challenge because there is no existing facility we can follow up with. So traditionally, the loading is in one dimensional only. So that's why we built these facilities to simulate the actual three-dimensional conditions. That is the world's first one. This helps when thinking about things located very deep underground. Construction, particularly for mining and infrastructure. Over the past few years, I have been closely working with industry. For example, for mining activities, we needed to explosively to control the directions of material fracturing, but also make sure that the structure itself will not be damaged after the explosive happening. I've also been working with the Northeast Link project. That area is quite challenging because there are multiple different ground conditions over there. If we're not properly designing the construction direction, the time of boring machine may not be properly driving smoothly and potentially induce a sinkhole or the collapse during construction. By using the techniques we developed, the project has been running smoothly. On traditional engineering, there is opportunities we can transform in traditional technology with AI-driven innovations. That is the main research we are doing at the moment.
Just different alignment according to the navigation system.
Receiving John Booker Medal is a tremendous honor for me. And I do believe we'll take this as momentum to keep moving my research to next level.
Next is the Lefebvre Medal for research in chemistry. It commemorates the work of the late Professor Raymond James Wood Lefebvre. Its purpose is to recognise outstanding basic research in chemistry. Please welcome the 2025 recipient of the Lefebvre Medal, Dr. Feng Wong Li.
Congratulations, Feng.
I do see a time that we do not need to dig into ground for new fossil fuels. Instead, we can recycle the use of carbon material. This way, we call it circular carbon. My name is Feng Wang Li. I'm working at the University of Sydney. My research is focused on using renewable electricity to convert carbon dioxide into something useful. For example, ethylene or ethanol. But there are many challenges for the carbon dioxide conversion.
Electrolyte is one molecule H.
Carbon dioxide is a very inert molecule. It is very hard to activate it. You need to find a suitable system that can efficiently utilize electrons. You will get quite sick catalyst.
We have developed a slim flow cell where we can convert up to 77% of carbon dioxide into the multi-carbon product. This was a world record for more than 2 years. There is a long way to go. We need to make the process more energy efficient. Almost half of our group is doing green hydrogen. The challenge for the green hydrogen is how you can produce it in a cheaper way. Actually, this is the reaction we are doing for splitting water into hydrogen and oxygen with the power from renewable electricity. We are collaborating so that we can develop some new reactor designs to make the water electrolysis for producing hydrogen in a more efficient way. We are also working on CO2 capture, again, using electrons as the driving force. Instead of capturing CO2 from the air, we are looking at capturing CO2 from ocean water, because ocean is the biggest carbon sink on the Earth. I feel passionate about this, and I feel excited about any new discoveries, because I don't think we have sufficient time to wait. We need to identify some way that can accelerate the process so that we can really convert these big discoveries into something that can really make a big impact on our daily life.
Next is the 2025 Moran Medal. The Moran Medal recognises the contributions to science of the late Professor Patrick Moran. It recognises outstanding research by scientists up to 10 years post-PhD in the fields of applied probability, biometrics, mathematical genetics, psychometrics, and statistics. Thank you to Louise Moran, Patrick's daughter, for joining us today and for her support of this and other Academy programs. Thank you. Congratulations to the recipient, Professor Margarita Moreno Betancourt.
Congratulations, Margarita.
I like to think of statistics as the science of science, because it's really about what it takes to create knowledge. My name is Margarita Moreno Betancourt, and I'm based at the Melbourne Children's Campus. My work in biostatistics aims to develop the methods that we use to analyze data from medical research studies to understand the extent to which factors from like taking a medication to like growing up in a disadvantaged neighborhood affect or may be detrimental to health outcomes. We call that causal inference. My research allows us to answer questions that were previously not possible to answer. For example, a method that allows us to look at whether intervening to improve preschool attendance and improve home reading can allow us to counteract the detrimental effect of socioeconomic disadvantage on children's development. We are also working to incorporate machine learning into our methods, meaning we can make the most out of the very rich data sets that we use. And more recently, there's a really big study called Generation Victoria, which is statewide and is going to really boost the numbers of participants relative to other studies and allow us to answer questions that we couldn't do before. Before I worked at the Melbourne Children's Campus, I was involved in studies at the other end of life, so looking at mortality registers in France and trying to look at the effects of different factors and socioeconomic disadvantage on mortality, not just the underlying cause of death, but to look at contributing causes, because people often live with chronic conditions at the same time. So in the end, assigning death to a single cause in many cases may not be sensible. And in addition, we have M1 and M2. I feel really honored to have been awarded the Moran Medal, and very thankful to my causal inference team, who are a bunch of really bright early career researchers. And I'm really excited about this medal in particular because it will really lift the profile of biostatistics, and in particular the area of causal inference in Australia.
Next, the Pawsey Medal. It recognises distinguished research in physics and commemorates the work of the late Joseph Pawsey. I would like to extend our thanks to Hastings and Liz Pawsey, who have been great supporters of the Pawsey Medal. Please congratulate the joint recipients of the 2025 Pawsey Medal, Associate Professor Claudia Lagos and Dr. Daria Smirnova. First, please welcome Claudia.
Congratulations, Claudia.
Right now in the world, we see a lot of conflicts. And I feel like when we consider the really big global challenges we tackle in science and the human endeavor involved, we are able to see above all those differences because we all have the same curiosity and the same questions. I'm Claudia Lagos. I'm an astrophysicist at the International Centre for Radio Astronomy Research in the University of Western Australia. What I do kind of on a day-to-day basis is to try to understand how galaxies form and evolve, basically using computer simulations to try to get to that understanding. The way we do that is by saying, okay, these are the laws of physics as we know them. So how can these laws give rise to the types of galaxies that we observe, like our own Milky Way, for example? So we feed everything we know into a computer to create simulations that we can then compare to observations. Does it look anything like what we actually observe? Our observations are extremely rich. We see galaxies come in all sorts of shapes and colors, and some are extremely big, some are very small. We are at a point in the simulations where we're starting to get some kind of diversity that looks like real galaxies. So once you get to that point, we can start questioning, what do we need to do now to answer some questions that are still open? What kind of observations we would need to do to resolve that type of question? So for example, I've worked a lot on supermassive black holes. So we know pretty much all galaxies that we observe have a supermassive black hole in their center. And although it's a tiny scale compared to the whole galaxy, we found that it can actually have a huge impact on the fate of that galaxy. We also look at how stars form throughout the universe. So we know that in the past, the universe was a lot more active in forming stars than it is now. And similarly, we can observe the gas content of galaxies. And there was a big problem there because we saw that the two were evolving very differently. But we know that stars form from gas. So it was, this question of why are these things not resembling each other? Turns out you need to trace the quite dense and very cold molecular gas. And the simulations predicted very nicely that, including my own simulations early on, that if you were to trace all that cold and dense gas, then you could see star formation much better. And that was actually observed years later. So simulations can really help understand star and galaxy formation.
And this is not strong enough, basically, but they're still running down the road.
I am very passionate about producing open source code that is free to use for the whole community. All researchers can benefit and everyone can see what's happening inside. And that's why we produced our semi-analytic model of galaxy formation called Shark. I feel extremely privileged, but also very proud to be recognized for work, that sometimes you're on your desk working the whole day, you never know if not someone notices, It's beautiful to see that acknowledgement of our scientific interest, but also of our tools that can be used by the community as a whole.
And now please welcome Daria.
Congratulations, Daria.
Research often requires persistence and self-devotion, and it's very rare to immediately solve a big, outstanding problem. That's why what is important is to develop your skill set, and also try to come up with new, fresh, original approaches that could just bloom one day. My name is Daria Smirnova. I work at the Australian National University. We live in the information era. That means we rely on the use of multiple devices every day. However, the electronic hardware in our devices is power hungry. The promising solution would be to develop light-based technology, and in particular nanophotonics. The fundamental challenge we face is how to control light and light metal waves at a small footprint most efficiently, so that it can be integrated in microchips of everyday devices. So basically I develop ideas that could have a transformative impact on the everyday technology of tomorrow. Measure both fundamental frequency, and I develop a toolkit that bridges the concepts of condensed matter physics with polaritonics and nonlinear optics. We can record the images in the real space, in both real space and Fourier space. Because this is like interdisciplinary approach, it is especially powerful, and this paves the way towards design of synthetic quantum materials in particular, and also new ways of diagnostics. My approach relies on mathematical methods, and in particular topology, which helps us to create special photonic states that could have superior properties. So this brings us to absolutely new level of controllability. So my career path has been challenging, and I think almost all female scientists face some challenges they have to overcome to be successful. And that's why being recognized with postdoc medal has a special meaning for me, and it encourages me to maintain high standards in my work.
And now to the Ruth Stevens Garney Medal. This award honours the contribution to human cytogenetics by the late Ruth Stevens Garney. It recognises distinguished research in human genetics, including clinical, molecular, population and epidemiological genetics, and cytogenetics. I'd also like to acknowledge the ongoing support of this award by the Garney family. A special thanks to Ruth's daughters, Sarah and Miriam, who are here with us today. Thank you so much. The recipient of the 2025 Ruth Stevens Garney Medal is Dr. Ira Devison. Unfortunately, the last minute, Ira was not able to attend, but let's hear more about her work.
Genomics has resulted in a huge advance in our understanding of fundamental gene regulation. It's been a total game changer for our understanding of biology and for potential applications in medicine. My name's Dr. Ira Devison. I work at the Garvan Institute of Medical Research. A really key resource for genomic medicine is to have this sort of map of what normal genetic variation looks like in the healthy population or the general population. And every person is going to be different. Every person's genome is different. Everyone has a different set of gene variants. But to potentially diagnose a patient with a genetic disease, we need to be able to take their genome and compare it to this reference. There might be one or two genes that are not functioning as they should, and if we can sort of pick those differences out from the crowd and find the really rare events, we can potentially tell that patient what the cause of their disease is, and more importantly, we'll be able to direct treatments that are sort of targeted to the specific gene variants that this patient has. And that's the whole idea, I guess, of personalised medicine. Part of the work we do is look at diverse populations because there are a lot of groups around the world that are underrepresented in this reference data, and none more so than Indigenous Australians. So what we set out to do in our recent study, which is just the first stage in an ongoing process, is to sequence the genomes of Indigenous people from around the country and basically try to fill the gap. And we found that there was a massive amount of uncharted genetic variation in these communities. So it tells us there's a lot of genetic diversity that's sort of unique to Indigenous Australians. So we really need to, cast the net wide and make these resources, span across the whole country and be as inclusive as possible if they're going to really work. We've built a lot of expertise in a particular area of genomic technologies called long-read sequencing. It's kind of the latest class of DNA sequencing technologies. And we're getting some good DNA.
There you can especially see that the peaks are nice and clear.
It enables us to sequence the most difficult, repetitive, mysterious regions of your genome for the first time. Just by being able to do that opens up a whole lot of possibility for discovery and exploration, which means you can, hopefully come up with a more accurate, more effective genetic test. Science is absolutely fundamental to shaping human knowledge and our understanding of how the world works and our place in the world, and also to coming up with the new technologies that are essential to advance human society, solve the challenges that we see around us in the world, and ultimately, improve the well-being of people and of the planet.
So now I'm delighted to present the recipients of the Aboriginal and Torres Strait Islander Scientific Awards for 2025. This important award recognises research by outstanding Aboriginal and Torres Strait Islander PhD students and early and mid-career scientists, and supports the growth of their research network and international knowledge exchange through visits to relevant international centres of research. The award covers the physical and biological sciences and allows interdisciplinary and socio-cultural research that can straddle the social sciences. Congratulations to the joint recipients, Dr. Mitchell Gibbs and Associate Professor Shannon Kilmartin-Lynch. Let's hear about their work.
Sydney's Botany Bay is one of the most industrialised parts of Australia. 2 runways sit on massive fingers of reclaimed land, while more than 1,600 container ships dock at the nearby cargo terminal every year. But for Uncle Robert Cooley, this is home.
Oh, this place, means everything to my family. I'm a son of a fisherman, a grandson of a fisherman. We relied on this ocean to supply food on the table.
The local Aboriginal people refer to Botany Bay as Gamay. Uncle Rob is one of the Gamay Rangers.
Our role as rangers is to work with the different decision makers, government, local environmental groups to keep this ocean as clean as possible.
And that's how he started working with the University of Sydney's Dr. Mitchell Gibbs.
Oh yeah, pull the top over.
A proud Dunghutti man through kinship, Dr. Gibbs is a geoscientist drawing on traditional practices to restore local marine ecosystems, particularly shellfish reefs. Less than 1% of shellfish reefs remain nationally, but combining traditional cultivation and management practices with Western science may hold the key to restoring shellfish populations.
For Indigenous knowledge, what you want to do is work with the community that lives there. You need to be able to go to those communities to then say, what do you know? How can we learn from you?
Uncle Rob says when researchers fail to ask those questions, research projects can be viewed as intrusive or exploitative.
So, Mitch, that trust was built up, and I think we're at the stage now with, Sydney Uni and others that we can be free to open a dialogue and ask each other questions, because I think we know that their work is around improving the oceans, which ultimately benefit us.
Dr. Gibbs now wants to extend that dialogue by collaborating with Indigenous communities around the world.
The project that we're doing, especially with the Gamay Rangers, is bringing people from the United States, Hawaii, Washington, hopefully people from New Zealand as well, to come to Australia and talk about the restoration projects that they've done.
And I'm not sure why, but it seems that every time, like every season, we get large blue swimmer crab numbers, we get large cicada numbers as well.
The project will be funded by the Australian Academy of Science after Dr. Gibbs was named one of the recipients of the Aboriginal and Torres Strait Islander Scientist Award. The award's other winner for 2025 is Associate Professor Shannon Kilmartin-Lynch from Melbourne's Monash University.
I'm a Yoel Ngillan-Balak and Ngadarak Balak man, a part of the Tangaroan people in Victoria's northeast Kulin Nations.
Associate Professor Kilmartin-Lynch uses his research to help care for country by reducing the ecological footprint of construction projects.
A lot of what I do is making sustainable materials more applicable within sort of cement and concrete construction, so anything that's got to do with a cement-based material.
And given that the concrete sector makes up almost 8% of global carbon emissions, there's a critical need for material substitutes that don't compromise strength or cost. He was part of the team that turned roasted coffee grounds into a charcoal-like substance called biochar, which can replace a portion of cement in concrete mixtures.
Melbourne, being a coffee capital of the world, has sort of got picked up by everybody, and that ended up in over 1,800 media outlets in 82 different countries. There's been times I've been at conferences and people come up, oh, you do the coffee.
Associate Professor Kilmartin-Lynch's new project explores the traditional use of plant resins by Indigenous communities.
Indigenous people surely haven't gone and put it into concrete before. It wasn't something that was used within Australia. It was definitely used in other materials and, in other processes, but we're going to take the knowledge behind those processes and try and incorporate that into concrete.
He says the work will honour cultural methodologies while addressing present-day engineering and sustainability challenges.
I think the most exciting thing, about this is, working with my community and at the end of the day, being able to help my community.
Amazing work, I'm sure you agree, and congratulations to both of you on the award. Unfortunately, Shannon cannot be with us today, but I'd like to now invite Mitchell to the stage.
Congratulations, Mitchell.
And now for our last but certainly not least award for this year, the Max Day Environmental Science Fellowship Award. This award is named in honour of Dr. Maxwell Day, who spent a lifetime championing entomology, conservation and forestry and helping other scientists. I'd like to acknowledge the ongoing support of this award by Max Day's family and friends. We especially thank Max's son, John, who is here with us today. Thank you, John, for your commitment and enthusiasm in ensuring the ongoing success of this award. This annual award assists PhD students or early career researchers with their research and provides funding, support towards the cost of travel, courses or research expenses. Congratulations to the joint recipients of this award for 2025, Rebecca Greening and Dr. Nina Wutan. Please welcome first onto the stage, Rebecca. We don't have an AB. All right, so now let's congratulate Nina. Would you like to come up, Nina? Thank you.
Congratulations.
Now that brings us to the conclusion of today's award ceremony. I invite you all to join me in a final round of applause to celebrate our remarkable award recipients. May I again extend our thanks to our donors, families and friends. We appreciate your generosity that makes these awards possible and in turn meaningful for their recipients. Thank you very much. And just before I comment on the celebrations tonight, I also want to take this opportunity to recognise a member of the Secretariat, Kira Scaife, who's here today, who does all the hard work behind the scenes on all these awards. So thanks, Kira. The celebrations will continue tonight for those joining us at the gala dinner. We look forward to seeing you at 6:30 at QT Hotel, which is just across the road. Thank you and good afternoon.